is dignity. Good morning to everyone gathered here. This is the one day international conference on ImmuneFest 2023, proudly organized by the Department of Biotechnology, School of Arts and Science, PONNAIYAH RAMAJAYAM Institute of Science and Technology, Vallam, Thanjavur. I request you all to rise for Tamil Thai followed by the founder's hymn. Proudly organized by the Department of Biotechnology, School of Arts and Science, PONNAIYAH RAMAJAYAM Institute of Science and Technology, Vallam, Thanjavur. I request you all to rise for Tamil Thai followed by the founder's hymn.
மொபைலில் ப்ளே பண்ணி விட்டு மைக்கை நேற்று சண்டை அதில் அப்படியே
சீராரும் மதனமென திகழ்வரத கண்டமிதில் எக்கணமும் அதில் சிறந்த திராவிடனல் திருநாடும் தக்க சிறு திரை முதலும் தரித்தனரும் திலகமுமே அத்திலக வாசனை போல் அனைத்துலகும் இன்பமுற எத்திசையும் புகழ் மணக்க இருந்த பெரும் தமிழனங்கே தமிழனங்கே உன் சீரிழமை திறம்பியந்து செய்ய மறந்து வாழ்த்துதுமே வாழ்த்துதுமே வாழ்த்துதுமே.
Please be seated. Light and brightness represents the positive, the good and the good spirits. Education is the movement from darkness to light. Yes, it's the time for the significance of lighting the lamp. I invite all our dignitaries on the stage to lighten the lamp. Light and brightness represents the positive, the good and the good spirits. Education is the movement from darkness to light. Yes, it's the time for the significance of lighting the lamp. I invite all our dignitaries on the stage to lighten the lamp.
ஆமாப்பா. அதுக்கு சப்போர்ட்டுக்கு அங்கே யாராச்சும் மணிமாறனையும் போட்டிருக்கணும். மணிமாறனையும் இம்ப்ரூவ் ஆகிடுச்சு.
Sometimes you will never know the true value of a moment until it becomes a memory. I welcome all the dignitaries to release the souvenir. Sometimes you will never know the true value of a moment until it becomes a memory. I welcome all the dignitaries to release the souvenir. Thank you all. It's time to honor our guest speakers. I request our VC sir to honor Dr. Natarajan Gopalan. Thank you all. It's time to honor our guest speakers. I request our VC sir to honor Dr. Senthil Nayagan sir. I request Kamraj sir to honor our VC sir and Dean sir. I request Kamraj sir to honor our VC sir and Dean sir. Thank you, sir. Thank you all. A small cheer and a great welcome make a merry event. I invite Mrs. R. Suganya ma'am, Assistant Professor, Department of Computer Science, to deliver the welcome address. Thank you all. A small cheer and a great welcome make a merry event. I invite Mrs. R. Suganya
A
ma'am,
smile is a
Assistant
welcome
Professor,
sight that
Department of
invites
Computer
people
Science, to
in.
deliver the
It's
welcome
my
address.
pleasant duty to bid you all in a genial welcome. A pleasant good morning to all. A good idea will keep you awake during the monitoring, and but a great idea will keep you awake during the night. Yes, it's time to have a great idea to be arranging you in this one day international conference on ImmuneFest 2023 at the right time in a successful way. By the blessings of our farmer, founder Chancellor Sir, PONNAIYAH RAMAJAYAM Institute of Science and Technology, deemed to be University Vallam, Thanjavur, Tamil Nadu. It gives me an immense warm and great pleasure to grace all your presence in the interest of the program. And it also gives me a tremendous pleasure to be presenting the welcome speech on behalf of PRIST University. I welcome our most honorable and dignified Chancellor Sir and Pro Chancellor Madam. Though they are not physically present with us, but their thoughts and prayers are always with us for our success. Now I welcome our honorable and respected President Sir, who has given me a support in this conference. I welcome you, sir. Next, I welcome our beloved and dignified Vice Chancellor, sir. Welcome, sir.
Hello.
It gives me an immense pleasure to welcome our Dean, School of Arts and Science, who has been backbone of this conference. I welcome you, sir. An open door is a welcome that makes a strange into a friend. From this conference, our guest will be familiar to our university. In this occasion, I convey my heartfelt welcome to the most extreme personality who has won accolades in their respective fields is none other than our chief guest, Dr. Nadarajan Gopalan, Professor and Head Department of Epidemiologist and Public Health, Central University of Tamil Nadu, Thiruvarur. Welcome you, sir. I am very grateful to extend my cordial welcome to our honorable guest, Dr. L. Sendil Nayagam, nephrologist, SB Hospital, Thanjavur. I welcome you, sir. It's been tremendous welcome to the professors and participants from various colleges and universities in various countries and states. I feel extremely glad to welcome our staff members and my dear students once again and welcome you all.
Thank you, ma'am. Success doesn't come from what you do occasionally, it comes from what you do consistently. I invite our Dean, School of Arts and Science, PRIST Dean to be University to deliver the keynote address. Sir, the dais is yours.
From the blessing of our former founder chancellor, Professor Dr. P. Murugeshan, PONNAIYAH RAMAJAYAM Institute of Science and Technology, deemed to be University, Vallam, Thanjavur, Tamil Nadu. I start my keynote address for this one-day international conference on ImmuneFest 2023. Our most honorable chief patron, the Chancellor, Mr. M. PONNAIYAH Nageswaran, Pro Chancellor Madam Mrs. P. Badmastri, President Dr. N. Ethirajulu, Vice Chancellor Dr. T. V. Christie, Pro Vice Chancellor Dr. S. Udayakumar, Registrar Dr. M. Kanigan, Special Officer Dr. S. Nidyanandam, Honorable Patrons Dr. N. K. Meera INSA Vice President, Dr. Taruna Madan Gupta, Indian Immunology Society President, Dr. K. Palakrishnan, Indian Immunology Society Secretary, beloved resource persons, Dr. Nadarajan Kobalan, Professor and Head Department of Epidemiology and Public Health, Central University of Tamil Nadu, Thiruvarur, Dr. L. Sendil Nayagam, nephrologist, SP Hospital, Thanjavur, Dr. D.S. Prabhakaran, Department of Radiation Oncology, Japan, Dr. K. Palakrishnan, Professor, Head Department of Immunology, Madurai Kamaraj University, Secretary, Indian Immunology Society, Director, Madurai FICA Center, most honorable directors, deans, professors, IT wings, including System Admin Seduraman team of PONNAIYAH RAMAJAYAM Institute of Science and Technology, deemed to be University, Vallam, Thanjavur, Tamil Nadu, and most welcomed participants from various countries and my dear students. Very good morning to all. It is a great honor for me to present the keynote address in presence of you all. About the institution, the parent institute was founded in 1985 by Professor Dr. P. Murugeshan. It was conferred deemed to be university status in 2008 by the University Grant Commission UGC under Section 3 of the UGC Act 1956. PONNAIYAH RAMAJAYAM Institute of Science and Technology, PRIST, is offering undergraduate, postgraduate, doctoral program in arts and science, agriculture, commerce and management, engineering and technology, law and pharmacy. Currently, it holds a total of 8 in renowned departments and 5 schools. About International Day of Immunology. The Day of Immunology, DOI, was established in 2005 by the European Federation of Immunological sometime in April 29th of that year. The purpose of DOI was to promote the importance of immunology and its role in maintaining the health and well-being of individuals. It was also meant to bring awareness to the public and the scientific communities about the significance of immunology. Highlights. The immune system is the key to the body's defense against infections. To signify the importance of immunity, April 29th is the marked as International Day of Immunology with the 2022 with the theme of vaccines. The International Day serves as a bridge between understanding the immune system and raising global awareness of immunology. About ImmuneFest 2022. The School of Arts and Science, PONNAIYAH RAMAJAYAM Institute of Science and Technology, PRIST, successfully organized a one-day conference in ImmuneFest 2022 on the occasion of International Immunology Day, including 12 faculty members from the field of biological sciences. The students presented scientific posters on appropriate themes, that is, vaccinology, autoimmune disease, infectious disease, COVID-19. The overall event was successful and admired in the university. It was beneficial to commemorate and update International Day about ImmuneFest 2023. The PONNAIYAH RAMAJAYAM Institute of Science and Technology, PRIST, and Immunology Society, Indian Immunology Society, IIS, are proud to announce the upcoming one-day international conference ImmuneFest 2023 in celebration of International Immunology Day. This conference will have the theme of immunology talks to the public health, and also all faculty members, academicians, and students are invited to submit scientific poster presentations related to this theme. This promises to be an exciting event, and all those interested in immunology are encouraged to attend. In this conference, we have received 258 abstracts from various colleges and universities. Out of that, we selected only 102 of them. The list of participants, academic wise, in the hybrid mode, that means both online as well as offline. That is academic wise, professors 7, associate professor 24, assistant professor 265, medical doctor 4, scientist 5, faculty 38, nurse 2, research scholar 68, then student 447, totally 860 are participated both online and offline. The list of participants state-wise: Andhra Pradesh 16, Andaman Nicobar Island 1, Assam 3, Bihar 14, Chhattisgarh 59, Delhi 6, Dayu Daman 1, Gujarat 11, Haryana 13, Jammu and Kashmir 8, Jharkhand 3, Karnataka 31, Kerala 27, Madhya Pradesh 15, Maharashtra 61, Mizoram 1, Nagaland 1, Odisha 4, Puducherry 30, Punjab 4, Rajasthan 9, Tamil Nadu 496, Telangana 4, Uttarakhand 2, Uttar Pradesh 22, West Bengal 18, total in India 845. List of participants in the country wise: United States 2, United Kingdom 1, Iraq 1, Egypt 1, Kenya 2, South Korea 1, Nigeria 1, Malaysia 4, Saudi Arabia 1, Sri Lanka 1, total 15 from the other countries.
Okay.
I mention our organizing secretary, Dr. R. Kamaraj. You can clap. Department of Biotechnology, School of Arts and Science has taken extreme effort to organize this one-day international conference on ImmunFest 2023. It has been a fortune for School of Arts and Science, PRIST deemed to be university, to be more academic functions like this in future. At last, my heartfelt thanks to the management and the higher authorities for providing all facilities and strong support. Thank you, one and all. Thank you.
Thank you, sir, for a brief keynote address. Every moment is a fresh new beginning. A wonderful inauguration is the great cosmic journey through the conference. I invite our Vice Chancellor, sir, PRIST deemed to be university, to deliver the inauguration address. Sir, you are pleased to be on the stage.
Before I formally go into the address, may I have the organizing secretary, Dr. Kamaraj, here on stage, please. Please. Yeah, he deserves a very, very loud applause. Thank you. Be seated. If we are here today, if we are able to have very notable, distinguished guests in our midst today, it is because of the efforts spent by Dr. Kamaraj. All of you must be knowing the university is now really in a very, very, tight moment. We are going to the National Assessment and Accreditation Council for accreditation, and at the moment, having submitted our SSR self-study report, we are now looking into the DVV queries. So we are really at a very, very tough time. But then, kudos to the Department of Biotechnology and the School of Arts and Science, led by Professor Chinnappa. I've been noticing, yes, he does deserve another loud appreciation from us. I've been noticing when it comes to organizing programs, somehow very meaningful programs are conducted by the school in spite of the so many restrictions that we have. Generally, we tend to give excuses, right? We tend to give excuses for not doing something. But then here I find a team led by Dr. Chinnappa, and today, in particular, by Dr. Kamaraj. I know there are so many faculty members and students who are behind him. I'm not able to mention each of your names, but this is really a very, very appreciable effort. Having said that, I would like to very specially welcome for this day our honorable guest, Dr. Nadarajan Gopalan, Professor and Head, Department of Epidemiology and Public Health, Central University, Thiruvarur. We are very thankful to you, sir, for being here. You know how busy and how engaged a professor and head of the department would be, and he has taken time to travel all the way from Thiruvarur to reach here, and he is going to be with us spending the entire forenoon. We are very, very thankful to you for your efforts. And we are very thankful to Dr. L. Senthilnayagam, who is here from the SB Hospital, Thanjavur. Let's give him a very big clap. He is a nephrologist. How many of you know about nephrons? Many a time we just run the life without really knowing what is happening inside of us. Do you know there is a 16 feet small intestine stuck inside our tummies? Just touch your tummy and feel it. It's a 16 feet large intestine. And then we have a 5 foot long, the small intestine is 16 feet. The large intestine is 5 feet long. Okay. For your information, 1 foot is a scale, right? 16 scales are tucked up inside our tummy. Okay. And kidney is a very, very small organ, right? I'm sorry, I'm taking a class on biology, but I cannot stop telling certain things, especially when you have gone through some of these things yourselves. About 30 years ago, sir, I was running to nephrologists for the sake of my father-in-law. So I know the pain. I know the pain. The nephrologist, when we met him, he said the kidneys are spoiled and he cannot live longer. And with the help of medications, he was able to live for about 3-4 years extra. And he died at the age of 53. And sharing the platform with the nephrologist today, I really have mixed feelings. I know how important you are to the society, doctor, and he is spending a half a day with us. You know the worth of his time? There'll be so many patients waiting for him today. But he has taken time to be here. We are very thankful to you, sir, for your presence. Okay. This is an awareness creation program, right? We are talking about immunology. And professor comes from the Department of Public Health, right? How aware you are about the Day of Immunology, which is celebrated as Day of Immunology? I'll have some gifts for you. Which day is celebrated as Day of Immunology? 29th April of every year is celebrated as the Day of Immunology. You must know about immunology. My dear friends, if you are sitting here with all health, it is because of your immune system. Your immune system, which is there inside, it is a complex mechanism of organs, cells, proteins, and whatnot. And they say the bone marrow, which is there inside our bones, that is where, the immune cells get generated. There is so much happening inside our body. If you just look into your own body, and then if you look into the planet Earth, you do not have a proof for God's existence. This is what I tell all of my students. Can you just imagine this place we are sitting now is revolving at a speed of how many kilometers? 1,650 kilometers per second. Okay, how are we getting day and night? Now the time is around 11 o'clock, and another 12 years it is going to be night. How is it happening? This planet Earth keeps revolving at a speed of 1,650 kilometers per second. And not just that, it goes around the sun at a speed of 1 lakh kilometers per second. Do you feel your chairs shaking? Do you feel your chair shaking? It's a wonder of creation. And so it is with your body, right? Your immune system is always alert to destroy any foreign, uh, virus or bacteria that gets in. And the beauty of the immune system is it recognizes which microbes it has killed. It is always alert. Next time when the microbe enters, automatically it gets killed. And if your immune system is going to falter, you will have all kinds of diseases, all kinds of disorders, metabolic disorders, cardiovascular disorders, and still fearful is the neurodegenerative conditions, which finally takes you to what's called Alzheimer's disease. Have you learnt about all this? Have you seen people suffering from Alzheimer's? I've seen a few of them. They are 70 years, 80 years old, but they behave like little children. They cry out of pain. They have forgotten everything. Just imagine if your memory is totally wiped out for a few moments. You just forget who you are. What would happen? Your system of immune system is responsible for keeping you going. So throughout this day, keep thanking immunology. Keep thanking your immune system, right? And keep thanking the Department of Biotechnology, Dr. Kamaraj, Dr. Chinnappa, and all of you others who are here to organize this function to make it a success, right? And it has come after a hard work. I remember about 3 months ago, Dr. Kamaraj coming to me and saying, "Sir, we want to have an international conference." I had my own doubts. International conference, it would cost a lot, and no management supports 100% the efforts of an international conference. And central universities need not worry. They are asking you, how are you spending the money? They give you the money and then they ask you, how did you spend the money? Things have changed. Okay, fine. Okay, so 3 months ago when Dr. Kamaraj came, I had my own doubts. But today we see the things happening. It's a beautiful example, right? There is one place where success comes before hard work. Where is it? Anybody? There is only one place where success comes before you do hard work. Where? Dictionary. Only in the dictionary you will find success coming before hard work. Friends, you have got to keep working very hard. Okay, now Immunology Day is being held, and you know what is a theme of Immunology Society this year? Anybody who answers will get a cash gift. The theme of 2023 Day of Immunology. Nobody? Sir, I give a cash gift if somebody answers this. I'll give you 1 minute's time. I see some of you looking into your cell phones. At least you take that effort. Okay. Okay, time up. Please be aware. See, awareness is required. Awareness is required. Towards the end of my talk, I'm going to give you all an assignment which will give you a greater awareness. Okay, it is not just today we talk about immunology. Is it okay your immune system works all right only today? Tomorrow your immunology, your immune system will go for a toss. Is it okay? No, right? So awareness has to be created. Immunology talks to public health. That's the theme. Immunology talks to public health. It is there at the front in the flex that they have displayed. Okay, it's all there. So the theme is important, right? Theme is important. That is what gives you focus. What is the theme of your life? If I ask this question, how many will answer? What is the theme of your life? To be a good wife, to be a good husband. Okay, it starts, charity starts at home. Don't say, I'll be a very good computer scientist. I'll be a very good pilot. Hello. In your home, first be a good son and good daughter. Okay. And the rest comes afterwards. The first circle. If you are not useful to your first circle, you are not going to be useful anywhere. So, immunology speaks to public health. By the way, what was 2022 theme? 2022 theme was vaccine. Okay, that is the time everybody was telling everybody, please go take vaccine. How many of you are vaccinated twice here? Hands up. Vaccinated twice against COVID. That is only COVID. Okay, please put your hands down. There are plenty of other diseases. There are plenty of other diseases, but mankind has really advanced a lot. Things like diphtheria, they say triple T, doctor. So when we are born, we are given certain vaccinations, right? Have you heard about smallpox? Smallpox, you would have seen, gory figures, people affected with smallpox. And what is that, sir? There's a recent virus. Ebola virus. Ebola virus. Have you seen pictures of people affected with Ebola virus? My God, you can't digest. You can't digest. Just imagine Ebola spreads to our country. Thanks to our immune system, it protects. Thanks to our immune system, it protects. Okay, now that brings me to something I wanted to tell. There is something called SDGs. Sustainable Development Goals. All of you must know, my dear students, your awareness is your success. What is this SDG? Sustainable Development Goals. In the year 2015, UN announced 17 SDGs. Okay, education is also, quality education is also an SDG. What is that? It's the third SDG right now. Fourth SDG 4 is quality education for all. And what is the SDG 3? You are all missing my cash prizes. SDG 3 is quality well-being, good health and well-being. Okay, now what is this SDG? SDG, finally it says, in the year 2030, there should be peace and prosperity prevailing in our planet. And poverty should be eradicated. So year 2030, all of us live in peace and prosperity. Think of our brothers and sisters who are now trying to flee from Sudan. You know what is happening in Sudan, right? Many Indians are caught up there, and our government is sending aircrafts to bring those Indians back. Same thing happened few couple of years ago in Afghanistan. So worldwide there are problems going on. So UN said in 2015, at least in another 15 years, 2030, we will have this whole planet living in peace and prosperity. Goodness and well-being is a third SDG. So when we talk about all these things, how important this particular conference is, how important that we keep our own immune system intact. You know, when you go to the doctors, they say your immunity is weak. Have you heard people, doctors say that to you? Your immunity is very weak. There are some of us who can take stress, any amount of stress. But there are some of us who can't, skip even a time's meal. They feel very weak and their immune system is very, very weak. So our own health has to be protected. Only then you can serve the society. So how do you protect your own health? Immune system. Okay, I will not take much time in closing. I just want to tell you two things. Number one, the biggest event on immunology is going to take place in Washington, D.C., May 11th to, I think, through 15th, if I am right. It's happening in Washington, D.C. The top minds in immunology in the world, they are gathering there. And it is available online. Please go watch these things online. What do you use your internet for? I'm putting a very serious question to you, my dear friends. We find today internet is spoiling the youth. You are watching things which are unnecessary on the internet. You are using social platforms for doing things which will spoil you, spoil your family, spoil your reputation. What are you using the internet for? Are you just using to watch some movies on Netflix? So on and so forth. People earn money out of your parents' blood, I tell you. You have sucked your mother's blood when you are born into this world. And today, when you are paying your fees, you are sucking the blood of your father. What are your eyes watching on the internet? Are you a person who go to sites which are not necessary? Let me put it plainly. Pornographic sites. It will kill your personality. It will kill your reputation. It will kill yourself and it will kill your family. Next time, when you want to visit such sites, please bring your father and mother, your brother and sister before your eyes. What are you watching on the internet? Please go watch from May 11th onwards. You can register for these programs online. Okay, the top immunologists in the world, they are coming together. The American Association of Immunology is hosting this program. You must get your hands on such information that will make you a good citizen. Okay, that is my first request. Second request is, I wish that this, uh, thing which has been started today, giving awareness about immunology, be carried throughout the year. And I would request Dr. Chinnappa to form a group, Immunology Awareness Creation Group. And with his permission, I nominate Dr. Kamaraj as the head of the group today. And volunteers, please join him. I do not have time now. Yes, let a group be formed. And this group will go to the other departments and conduct, small programs. We need not every day gather like this, spend time. You can walk into the classroom Tell something about maintaining your immune system for 10 minutes. Awareness creation is important, and not just today, throughout this year. Please keep doing things so that this good job that is started today will really yield fruits. I'm very happy. I think we have participants from the other institutions also today, and the conference has, uh, is having really an international flavor. That Shinapa was reading out the number of participants from across our country and from across the world. It is truly an international conference in its stature, and I hope what is started today will be carried on into the future, and all of us will be benefited. And finally, what is the point? We should be able to keep our own immune system intact, our own immune system getting stronger and stronger so that we can live longer and serve the society truly. Thank you. Thank you very much.
Thank you, sir, for your brief inauguration speech. Sir, that was really a fruitful to us, and even somewhere missed your cash prizes. Not today, but in forthcoming functions in future.
It's time for tea break. I request all the participants to refer. Sorry? Sorry?
The greatest scholar.
Sorry for the wrong information. I request all the participants to be seated.
The greatest scholars are usually the wisest people. To hear about the wisest person intro, I invite Mrs. R. Suganya, Assistant Professor of Department of Computer Science, to deliver the guest intro.
Scholars are gift. Scholars are gift. Saints are praises. Stages are blessings. And light is our reward. My immense pleasure to welcome our guest, Dr. Natarajan Gopalan, Professor and Head of Epidemiology and Public Health Department, Central University of Tamil Nadu, Thiruvarur. He has completed MSc in Pondicherry University, doctoral degree in Shivaji University, Gwalior, and postdoctoral degree in OUHSC, USA. His specialization is in the field of cancer biology, entomology, epidemiology, food biotechnology, herbal plant products. He has 39 years of experience in scientist and technical field. He has also acted as a guest editor for Defense Life Science Journal and DRDO Technology, focuses at Central University of Tamil Nadu as professor and head epidemiology and health public health department disease management. He has received many rewards like Best All-Rounder Scientist of the Year in 2016, Laboratory Scientist of the Year 2018, DRDO Technology Group in 2018, DRDO Spin-off Technology in 2018, National Science Day Oration in 2018, National Technology Day Oration in 2019, DRDO Second Best Paper in 2020. He is a member in BIS FAD 7 21 27 from 2016 onwards and FDA in 2018 analytical members. He is also a member in Task Force DRDO Committee for Cyborg Program. Still it is there, sir. I have been lot to study about you. Okay, sir. So we are eagerly waiting for your voice, please, sir.
Thank you very much. Very good morning to all of you. Uh, our VC from this university. I was thinking initially, the way in which he was talking about you is about the subject. It's a broad spectrum. He has given you the from the immunology to astrology and then education, whatnot. It's a clear cut about what you should do it, how should be a better human beings. That also he has pointed out. It's a really useful thing. Now the internet is you will from the internet which gives you the good things, bad things, all the thing, whatever you want it, you can take it now. Thanks to VC sir, because initially he was appreciating Dr. Kamaraj, but I should appreciate him initially because he has given all the things to Dr. Kamaraj and Dean to conduct such a wonderful event. Because since going to be tomorrow is a immunology day, and also the theme is really very very important. The public health, what is immune status? They wanted to know the public health because COVID period we have learned a lot because we all saved because of our immune system, which worked a lot. The food, what we used to take it, the traditional food, mainly from the Indian traditional food, that saved us in many of the things. They all became doctor, during the period. And also he was telling about the doctor because we gave so much, COVID period for the medico, paramedico, doctors, whatnot, because they only helped us to be here, live standing here because of those people. Thanks to doctors and then nurses and everyone. And then without any international participants for this conference, Kamaraj took so much pain. He has invited many people from this one, almost 850 participants. Without participants, it's very difficult. But he has arranged everything. Either it is virtual or from the offline, online, everything which he has taken from this conference. Thank you very much for everyone, your founder member, as well as the VC, Dean, the faculty, the students being here for this international one-day conference. It's general. I will be talking about how the vector-borne disease. What is vector-borne disease? When we know about the communicable, non-communicable disease, we are all facing it. The COVID has taught us everything, from where it is coming, how it is coming, from how we affected, how our immune system worked a lot, all the things. But things to be learned a lot. Every day we are learning, as he was mentioning about every day. I do learn from you people only, since the young students can read, they can get knowledge, they can get the memory power. The memory power is very high initially, because for this my age it's very difficult to read it. Even if we read it, I may recollect some of the things, but the young students can read it and then get more information. Those days when we were that's what I was talking to Dean in the morning. So when we wanted to do research, when we wanted to do conduct some of the part on the how the experiment going to be done, it was very difficult to get one publication. Those days we have to send a letter and then get the information, what type of experiment he has done, how he has done it. Now the cell phone is really which gives you within no minute you can come to know world what is doing it, what we should do it, what are the lacuna which we are facing. If I would have planned some of the experiment, but somebody would have done it. So what they have done it, how I can modify it, all the things you can get it from the internet, the cell phone. It's really the useful cell phone has given. Kindly take it as a positive way, do it, and then get the information, because every moment, the world is doing in a very good research. When we see the COVID period vaccine, we stood there first. We only produce, because we know that what are all the implications on the vaccine, what are the things which going to be, how the people will react, how the things, even the initially when this vaccine also made it, and then people were not taking it. They had so much fear whether they had done it properly, whether they had evaluated properly any of the vaccine. To get one vaccine, years together it will take. The first trial, second trial, third trial, all the things to be done. But they had done on in silico model. They have done it because we were not having the entire genome. We were having only the short piece of gene. From the gene only which the vaccine was made, how it will affect, where it is cloned, so many vectors, the virus which they have cloned, virus, how it will react. $1 million questions were there, but still we took it. We even when the first man to take vaccine in the Central University, I went personally. I said, okay, I will take it. I only educated people, because we educated people, our own faculty, our own students. Kindly go and take it. Nothing will happen. You may get fever for 1 or 2 days. You will be all right. The same way we took it. Now we saved it because we have to educate. That's what this year immunology topic is given. Clear cut topic, public health. We have to educate people. Most of the disease can be contained, can be controlled, or to be with you only all the things, how you educate the people. So when the simple example, the Aedes aegypti, which was transmitting the dengue virus, see, the mosquitoes are breeding with you only in your place, in your fresh water. So when you remove it, the fresh water, the mosquito will not breed, mosquito will not transmit. The mosquito doesn't want to transmit any disease. It needs blood. The female mosquito which needs to blood for the development of ovary, the egg development. But we are giving the parasite to the mosquito. The mosquito in turn, that's what some of the work which they have done. I will show you some of the things. So really, the some of the virus, once it is infected, the mosquito is infected, the progeny also infected. There's a few generation. That's called transovarial transmission. The virus can come to larva, the larva to again, the pupa to the next generation, egg, all the things which they have showed clearly by immunologist only. Because that's what the thing is. Since we know our immune system, what is the insect immune system, which are all the, but really they don't have the whatever B cells, T cells, spleen and other thing. Mosquito doesn't have. But mosquito has, the hemocyl part, the hemocytes, which are responsible. The parasite which has to multiply, the virus has to multiply, the transovarial transmission which is going means something which is protecting the death, prevention from the death of insect. So all the things which they have done, the recent development, science development, it's a very, 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 every day it's getting improved, all the things. So kindly see it, as VC Saab has mentioned clearly, the internet is really, really the model which really you can learn a lot. You can get all the information, the positive information. So I will talk about another 15-20 minutes. What are all the things which are mainly the vector-borne disease? Basically, I am entomologist. Thanks to madam, he was reading about my biodata. Basically, I am entomologist and turned to be a cancer biologist because I joined my service. If you see it, I am joined and just I am from the village. Tamil medium தான் நான் படிச்சதெல்லாம் till PUC, even the Tamil medium. When I went there, SLC last batch, PUC last batch, the principal சொன்னார் இதே மாதிரி தமிழ் மீடியம் தான் இருக்கு PUC-ல. I said okay, I'll join. What is wrong in it? So I learned it, and then BSc I did from ABC College, small college, MSc from Pondicherry University, first batch. Because I the hard work, that's the thing, always learn it. See, you have to do mistake. Always, by reading you can learn it a lot, and then, you can rectify yourself. But you can't get it success 100%, but we have to try it. We should not leave it anything which is possible. So when I did my doctorate mainly for the how the mosquitoes are getting resistance, because they were initially we have to do bioassay. You take the mosquito, the first generation, put the larva, 25 larva, the bioassay we have to do, which will take 24 hours. Then we thought that some of the things which they have done, the carboxyl esterase, one of the enzyme which overproduce when the pesticide is getting resistance, the enzyme is overproduced. So what we did, some filter paper, we put the beta naphthyl butyrate and then mosquito homogenate. Then you see the if the color intensity is more, the carboxyl esterase, we can say indirectly the mosquitoes got resistance. Because from where it is getting resistance, when we spray only it is getting resistance. The agriculture also important. Agriculture, they do spray all the insecticide, pesticides. So mosquito breeds on water. Yeah, generally we know that mosquito breeds on water. If the water bodies are eliminated, mosquitoes will not be there. But it's very difficult to know the urbanization, the semi-urban area, all the cultivable land which has gone to houses, factory, where we the sewage which we have, the close to sewage which are all the mosquito breeding will not be there. But it is very difficult to know to the population getting increased when we see it. When we are all overcoming the China tools, the population. We need the house, we need all the things. But the mosquitogenic place we can control ourselves. It is there in our hand. So almost we can educate the people how the sewage system should be there, agricultural land should be there, dry day, because dengue which they have controlled in Delhi and Singapore was one of the very good example. First they educated the people. So if the mosquito breeds in your house, how to close it. Close the water bodies in your house, the tank, the bucket, whatever water you store it. The mosquito, how it breeds, they showed. And then, when they go for second time, at least they show that this is the thing, kindly control it, close it properly. The third time they have fined it also, the $50 or something. Then, that's the thing, initially educate and then, some sort of policy, implementation, everything. They control the dengue. It's beautiful. But Delhi, the same procedure which they followed, they have failed it. Other places also, because the re-emergence of disease, so many places which are all coming. So one of the important how the disease was controlled in Singapore and other places, we also can do it. It's not a big job. We can do it easily. So many of the examples which are the how the vector-borne disease which they controlled it. And now Delhi they have taken, because the mosquito bites. This mosquito bites on daytime. You know that Aedes aegypti mosquito bites on daytime only. So many mosquito species, few thousand mosquito species are there. Each one has peak biting hours, different breeding places, different all the things. But the Aedes mosquito breeds on daytime. They made it in Delhi Municipal Corporation for week in a month, 1 day dry day. See to that all the people this one, there is no water bodies in and around, so that mosquitoes can be removed from that place. The same way, full hand shirt and all the things, when you close it, mosquito may not. All the things which they tried, they could do some other things, not 100%.
Biotechnology.
So I'll talk some few slides about a huge number of slides are there. I'll show some of the pass on some. And the important part is climate change. Climate change, which I have chosen for this topic, is climate change impacts on vector-borne diseases and immunology. I'll touch upon something, the climate part, as well as the insect, what all the disease which are all responsible. So this was the title which I have, going to cover is DFRL, CUT, and what is, because since I was scientist in DRDO, last few years I was in Defense Food Research Laboratory. That was an important place where we used to prepare for the food material for the soldiers, because soldiers are there, the border area, that's what we are all there here safe. Because why? Because soldiers can't carry the food material, they have to carry arms and other things. Food material is important. When we survey the patrolling, they can't take the large quantity of food material. The small quantity of food material with the high energy, they can survive for at least for few hours in the border areas. So that's what we used to do. It some of the food material, the high energy food material, and the storage of food material. So all the technologies are developed by Defense Food Research Laboratory at Mysore. So I'll tell about something, and then the importance of vector-borne disease, role of climate changes on vector-borne disease, the implication, the immune status of vector-borne disease, the vector disease containment. All we do have with us. So I have not taken from anywhere else. Some of the small experience which I had it on vector biology, as well as the publication. Many of the publication which are all there, you can download it. When you put it on the vector biology, you will get a plenty of papers. So I have this one thing which DRDO, you may be knowing. The DRDO, one of the important organizations. You should know about what all the organizations in India. You should know about CSIR, Indian Council of Medical Research, DRDO. DRDO, you can say that what is the role of DRDO on biology, because soldiers, we have to take care of few thousand soldiers are there. We have to take care of their health, their family members. So that's what DRDO, part of 9 labs which are all responsible for the biology lab. We do take care of the soldiers' health and everything, the food material. So the idea for all, what we have other university institute, they also have the different departments. And what are all the departments which are responsible? What are the food material? See, they wanted, army wanted the food material to have it at least minimum 2 years storage food material. We can store the food material for 2 years. Some of the food technology, whatever the retard pouch technology now we do have in the market, most of the technology transferred from the DRDO only. So the pouch, the food material, the processed, all the thing we can store it. But when we store it, the main thing is a contamination should not be there. The nutrient value should be there. Whatever the insult raw material which we prepared it without pesticide, all the things. So we have the set of standard we have to follow. So we used to follow all the things and prepare it the food material. And these are all the some of the technologies which are all available in DRDO website. You can see it. Any of the small entrepreneurship, if you want to develop your own, you can get the technology from DRDO and do it. That's the thing which are possible. Now the COVID period, many of the things we were sitting at home, we got it all the food material, all the drug, because of these are all the technologies. And Central University, we do have one of the a young university, I would say. We have 27 departments. Started 2011, almost 12 years old instrument. We have 27 department, 11 schools, and all the things. It's a big. If any chance comes, if you want to visit, you can visit us. And these are all the different schools, departments. And I'm basically looking after epidemiology and public health, because since I have I had some other experience on entomology as well as disease management. We have 19 students for the MSc, PhD, and all the people we do have it. So when we see the publication, the vector biology publication or disease related publication, see almost which is from 2000 to now it has gone to 220. So many of the publications, when you put on a website, you can check it up, because it shows that people are doing really the disease management, how the disease are transmitted, what are all the agents, what are all the pathogens, how we can control it, how it does, which varies place to place, environmental changes, many of the things, n number of factors, individual to immune system which varies, mine's immune system which varies from other people, so many things which are all. But we do overcome all the things, the B cells, the T cells, that's the common. But we may vary the polyclonal antibodies, all the thing, how it is produced in the body, but the basic nature is the same. So when we see about the vector-borne disease, almost every year many of the millions of people are dying with malaria, filariasis. In filariasis, not much mortality. The other vector-borne viral diseases that kills more. Mainly which shows about what all the responsible factors which are all main thing is how the insects are transmitting disease. But when we see about what all the insects, mainly the mosquitoes, different species. We have n number of species, the genera which transmit disease, mainly chikungunya, dengue, lymphatic filariasis. These all the things, and then what are the type of pathogens? When we have the fleas, lice, sandflies, leishmaniasis, many of the by insects. But insects, it's clear, as I was telling about, insect doesn't want to transmit any disease. We are all giving to insect. In turn, insect, multiplying those parasite pass on to us when it goes for the second blood feed. So this slide shows about the mainly the mosquitoes or disease, and then the transmission part, which mosquitoes, which insect which are transmitting, what is the global burden, and then prevention control. The prevention control vector-borne disease is very simple. When we different different ways of personal protection, drug, insect bite, when we prevent it, all the things, disease can be controlled. This slide shows about the vector-borne disease, how the global part, almost we do have all sort of disease in India. All the places we do have it, except some of the cold regions are very high temperature. The same way about the disease, the death, the mortality due to disease. So when the disease part, the causative agent, what is, whether it is parasite or whether it is bacteria, virus, what are the vectors, and then whether it is bites, how the mainly the bites which are all transmit disease. Some of the mosquitoes which bites, some sort of piercing and sucking, something which will make a hole such a way that the blood will come out, then the insect will feed. So when the things, the insect biology, when you say it's a beautiful insect, insects which are all evolved than human beings, which evolved first, which knows the blood clot should not, whatever the heparin, when we take it, the blood clot should not happen. The insect mouth, whatever the saliva which has that one. So see to that the blood is not clotted, the blood is properly which has gone inside. All the such a insect is a very good system which has. So the same way the disease causative agents and what all the flies which are responsible. The same way which different way which is given, what all the dengue, when we see, we do have the dengue 1, 2, 3, 4. The Plasmodium falciparum, initially we have 4 species. Now we have the 5 species also. The zoonotic, now the all the disease, the corona only we came to know all the zoonotic disease, everything which comes to on one health system. Now earlier it was not, that human disease is different, animal disease is different. Now the zoonotic is one thing from buffalo, cow, or animal, monkey, which comes to human beings, and then re-jump to the system, animal system. Again, it is all the mutations. Now you see the corona, which is jumping one to other, which we have, probably we don't have much mortality. That's what we don't bother about all the things, but still which will be with us as long as we are, the disease will be there. How to containment, we should know about all the things. So it is same way, different vector species. So overview, the some of the things, the main part is a mosquito borne, flies borne, lice borne, fleas borne, tick borne, all the things. We do have the dog with us, we do have the cat with us, we do have all the surrounding. Without animal we can't survive. So when we have the jumping disease one to other, the insects also bite one to other. So when we see about the what all the, we should know about the zoological name, the epidemiology, what is definition of epidemiology, morphology, life cycle. Unless we do learn it initially, the containment is very easy. So this is a simple way. One of the thing I'm not showing much about, but this is how the male female mosquito which looks like, how the larva looks like, how the egg looks like, where we should know about the where it breeds, how it breeds, all the things which shows about all the things. So when we see about the pathogen vector related determinants, the environmental condition, the economic condition, health, all which is interlinked any of the disease transmission. So mainly the demographic. See, when we talk about the climatic change, which shows about the entire thing which is responsible, demographic region, mainly the urbanization, agricultural, regional environmental changes. See, finally the disease emergence, all the things which are all related. When we compare everything, we learn it everything, or we educate the people, any of the disease can be controlled. So this climatic, experience from where it is by pollution, the water may not be slides, may not be clear because the photos are not clear. So all the things are responsible, the each and everything on the climatic change. Now the temperature is going up because it's not going up. We have made the environmental change everything. That's what the reemergence of the all the vector-borne disease. So it shows about the climatic change as well as the humidity, temperature. Finally, you see the things which is getting increased. The disease burden we are all facing it. Shows about the different pathways, the how the malaria, the insect, the mosquito-borne disease, or the one of the thing is the resistant part. The drug is getting resistance. See, the drugs are not used properly. We should know about the what drug to be used, when should be used, how should be used, all the things. When we are not taken properly, the drug or not used properly, bound to have all sort of the resistance status. So this is so not only now, which has been predicted long back. The thing, the temperature, the drought, flooding, all the things. Finally, the health status, where we are all only suffering, or our whatever the buffalo, cow, we do have with us those things also, which veterinary part, which we are all facing problem. So the climate change, mainly the range of abundance of animal reservoir, because of the what are all the things, really the increased part is due to climatic change. So the vector as well as pathogen in both part, the vector part also which has changed because we have given chance to develop the multiplication of vector. The pathogen, our immune system, any of the disease when we get it, what this is I was telling, the immune system, we should know about our immune system, what to eat, what not to eat, how the immune system we should develop ourselves. So when the immune system is very good, we may not get any of the disease. That's what all the things which shows about the uh, disease changes. Same way which all which are all interrelated. The climatic change which change to the vector pathogen as well as human population, poverty. So all wherever it is the climatic change which is responsible for the disease as well as management. So these are the just to show, picture about what are the insects which are responsible for the disease. How the classification. See, it's a simple way to tell about the how it is classified, the genus, phylum, class, and all the things. When the biology student should know about how the classified all the things. So then when we see about, first we should know about definition about entomology, parasitology, helminthology, all the things. As a student, you should know about, each and everything. The bacteria is given to you, the slide is given to you, or when we see on a microscope, the physical, chemical properties, all the things we should know where we should really fits on this. Then different classes, mainly the lice, the fleas, the bugs, as well as the mosquitoes. This is one of the mainly the important major diptera order. So this vector survival is important. The breeding place is important. How the temperature, the water bodies, the mosquito is one of the things. A beautiful system which we do have the sensor system. What we do take care of our children. The mosquito also the physiochemical BOAs which checks about the chemical property, physical property of the water. Then only which lays the egg. Otherwise, will not lay the egg. Even some of the mosquitoes, we check about the micro environment. What is the salt level, the pH level, all the things. The legs or the mosquito part of the abdomen, which has a wonderful system, the sensor. Then only which will lay the egg. See, even when we see one toxorhynchitis, you may be knowing it about some of the if you have entomologists. The toxorhynchitis is one of the mosquito species which will not transmit any disease, but it's a big mosquito. When we do it, some of the lab work only for the viral injection and other things. See, which will lay only one egg in the water. If we keep it 10 bowl, 10 eggs will lay in the 10 bowls, which will lay only one egg. So such a the second time chances are to get the disease. So this is a human cycle. This is what are the stages which are all there. What are the mosquito cycle? This all the things which are all showed here. What are the mosquito species? We do have almost different species, different sibling species. All the things we do have in India. Some of the species which we do have in plains. Some of the things which are all on the urban area, semi-urban area, forest area, and semi-forest area. All the species which are all there, but all the thing by science only, by learning only, different publication which we got it, that's what we came to know what are the species which are really transmitting disease. It shows about India, all the places where all the mosquito species which are really responsible for the transmission of malaria. There's another climatic change with the human to environment. This all the things. So it's a worldwide also. What are the mosquito species which are all transmitting? N number of species, some 286 mosquito species which are all transmitting malaria. It's about the 2050. What could be our condition? See the n huge number of malaria cases which we are all going to face with us, unless we control it early. So the another one is Kaisanur forest disease, KFD, the mainly which transmits by tick-borne disease. Generally, the cycle which is around monkey. When the deforestation, we used to go to forest. That's what we do get the tick bites. The tick also, the sensory system is very good. The larva which always stands on the leaf, the the end part of the leaf. When it touches you, or you are touching the leaf, immediately the ticks which bites. So how the sensory system is developed for the insects is really wonderful. Because of our body temperature, all the something is moving immediately which comes over there. So this another one is Lyme disease, mainly which transmits by tick as well as the rat. The how the Lyme disease, which are mainly the deforestation, because when we go to forest, all the disease which is not coming, it means we are only getting the disease when we go into forest area, or the deforestation. Same like the elephant corridor, they say that elephant is coming, it is not coming. We only made elephant to come to our own place. The same like all the vector borne disease, the insect, we are all bringing the insect to our place. So how the dengue, that's what we are all talking about, the mosquitoes, the Aedes aegypti, which is responsible, what is the temperature, what are all the studies which is done, the 4 species we do have it. The now earlier is different places where there now, even the south also, we are getting plenty of the dengue cases, especially this year, because all the thing mosquitoes, plenty of mosquitoes are the Aedes mosquitoes, and then we have given chance to bite, and then the disease which are all there. The same like the dengue, how the disease which is transmitted. The other one is JE. This generally the cycle JE, which are all there, Japanese encephalitis virus, mainly transmitted on birds, from birds to human beings, which all jump. Some of the species, the Culex bitorhynchus, trichorhynchus, some of the specific species, generally which breeds on the plantation, on the water bodies, on the rice field and other things. The other one is leishmaniasis. This is sandflies, because this also sandflies are with us only. All the things, the small fly which we can see on the corner of the wall of the things, either where which is a high humidity is required, which will be breeding with us only always, which will not go away from that place. Mainly the Patna area, Bihar, other places. Sorry, I think I have moved a lot. Yeah, this one, leishmaniasis is one of the disease by which transmit by sandflies. So the lymphatic filariasis is common in Pondicherry and Kerala. Some of the things we do have the two species. One is Pancreafti, another one is which Chicheria pancreafti. Another one is Malaya. This one, two species we do have in India. These are all the mosquito species which are transmitted. This is mainly the Pancreafti filariasis transmitted by Culex cuncutaceatus. mainly the sewage water. This mosquito breeds on highly polluted water. And there's Burgian filarias is this Mansonia species. This beautiful species, one of the mosquito species. This mosquito species, Mansonia, you can see the plant, mainly the water plant, which the larva will be there along with the plant only till the pupa, the mosquito is coming out till that period, which will be there on the plant only. Once you remove the plant, the mosquito will die. So such type of species, how they have evolved, you see that. You see, that's what the Kerala Sherthalai, that's a common Burgian filarias is. So when we went there, some of the field trials and all the things, when we removed each house used to have a pond. The pond has full of plants. The mosquito breeds. It was very high. So we have educated the people, when you remove the plants, the mosquito will die. Then they don't want to remove the plants. So we told we'll give the fish, the fish lingerings, and then we gave the fish. So you will get a earned also the fish once it is cultivated in that place. So they told, okay, we will remove the plants. So they got the fish also, plenty of fish over there, so that the mosquito also will not breed in other species. When we remove the plant, the mosquitoes were controlled, and in this way, the filarias is also controlled in that region. So what all the difference between the how the larva is how the Burgian larva or Wuchereria bankruptcy larva, how which varies from each species to species. So when we see about the immunology, I'll show some few slides about immunology. What is autoimmune allergies, asthma, cancer? The immunology part is very, very important. We should know about our immune system. So when we that's what Mrs. Abbas was asking. How many times you had a vaccine? First time vaccine, second time vaccine. What is your immune system? What are the upper polyclonal antibodies really which you developed or not? When I checked it for my polyclonal antibodies, it was around 39,000 when I got the second booster. So which shows about my immune system was very good, but other people also really which they have developed or not. But whatever the immune system which we developed during vaccine, somebody got it very, very, very short term only. Now time for see now also if you check it up, some of the people those who are sneezing cold, they may be positive. The chances are the immune system is not well developed. Those who got it really the first time infection, their immune system was very good, which developed along. Generally, any of the viral pathogen, same like he was telling about smallpox. When we had it, the smallpox injection, that is a lifelong. Whatever the chicken pox, once you get the infection, which goes for a lifelong. So all the things, but the COVID, because we were not knowing, still we don't know about the question how we got it, whether it is a cloned one or it is a lab developed or natural or jumping from animal to animal. We were not knowing it, but still our immune system is very good. That was we all have escaped for that one. So the this shows about the how the tighter between the as we were talking about the viral infection as well as COVID is, because it's a short-term immune system only which we all developed it. The missionary, actually the immunological missionary for the host understanding, we should know about the immune system, induction of humoral, what are all the things which really when we got injured. See why the malaria vaccine was not successful. It's a many five species, five mosquito species which are transmitting two cycle. It's really what to take it, whether it is sporozoite antigen we have to take it, or the heat killed we have to take it, which oocyst vaccine we should take. Many things which are all there. But natural infection really which gives booster for that one, so that you will get a complete, the memory cells will be with you, all the B cells which are all memory cells which are all there with you, so that lifelong you will not get an infection. That's what other beautiful studies which they have done. When we inject, I'll show you one or two slides. When we inject along with the needle any of the the vaccine, it's different from what the insect really injects. So insect along with the saliva injects, the parasites can be there for longer period, which can boost your immune system. So many things which they have published about all the things. So So it shows about what all the epitopes, immune responses, all the things, if we know about each and every basic science, how the system works, we can learn a lot, I think. The same way, the innate immunity, high variability because of, the factors of pathogen, different pathogen, different epitopes, different stages, how it is developed, the cloned one, all the things which mainly the genomic approach which will know. That's what the vaccine which developed COVID, we know about the genomic, the small sequence. That's what we could make it the things, the vaccine, in short duration. When we know about the whole genome, we know about really what are all the things which really responsible, how the which can boost our immune system. So we could have made a better vaccine. Another one is innate immunity, highly adaptable. As a modulation of, see, the insect immune system. Now it is these days people are working on insect immune system. We know about our immune system, insect immune system, which may be we are not knowing much about what is really which how the insect is doing it. How we can learn it from that one. People are working on it. The system which sustain the immune priming, that's what exposure pathogen. And they have studied very well in cockroaches, in a cockroach which they have exposed with pseudomonas species. And then some few days which they have, the system is developed. When they exposed with other bacteria, we showed some sort of positiveness, because the bacteria are not able to multiply for long. The same way other ticks they have done it, other species, Drosophila, fruit fly, which they have done it. The things, the same way, what is infected with the tick, and also the infected, not infected, because the saliva, which along with parasite or the virus which goes, the system is developed, the well immune system is developed in the system. And also what is which is blocked by Th1, as well as it's regulated the other interleukin systems. The same way, the insect which they have tried with the small interference RNA, how the small interference RNA which are working in the insect along with the exosome which they showed, and also transovarial transmission also which showed from the male female insect to the larva, egg, and all the things which they have showed it also. And also the food part is also important. These days people are working on the individual food, what is required for the individual food. The insect also, the individual food, what are the bacteria which are responsible, how the blocking the parasite or bacteria in the system, whether it is transmitting this type of the food material, this type of bacteria which are all there, those bacteria may inhibit the parasite development or parasite multiplication. So which showed about all the things, the recent past, the papers, the research part, and also the emerging re-emerging disease. Mainly we knew about, because of climatic changes, the disease which is spreading one place to other place. The case reduction management, how the early diagnosis, early warning, all the things, yes, it is possible. The human, the thing is, our role is responsible. The what all the disease which are all transmitted, what all the agents which are all there, how this is a situation is given, this is the environmental condition, and also this environmental condition persisting in other place. So the how the disease can be controlled or managed, or the forecasting of the disease, that's what our role, we have to do it. So all the things, risk part, which is important, where we should know about all the things. Other, the containment of disease, the integrated, the pest management, the vector management, the disease management, what are the things which really now the, in the machine learning or artificial intelligence which helps us. So when once we know about the data, the early diagnosis, the medical diagnosis, drug discovery, all the things which are all there, we can predict upon all the how fast we can manage the disease. The same way the as he was VC saw telling about the sustainable all the thing, one of the important goal, how we can the health monitoring system, they educate people all the things so that we can manage the disease easily. So integrated approach for the any of the disease, the planning, creating, establishing, implementing. That's what a few days back about you may be knowing the World Malaria Day that was fallen on April 25th. That's what this year theme they have given: innovate, implement, as well as to do the research, all the things. That's what that was the main theme which they have given for World Malaria Day. So some of the thing, collection, communication, awareness, mobilization, these are all the important part wherein we can control the disease in a better way. So first, when we see about, we should know about surveillance, what all the approaches, maintaining the responses, we should share our responsibility. So somebody is getting the data from the field, we should know about what type of data he has collected, how he has collected, whether it is really valuable. All the things when we do it, we can show in a positive way, in a fast way to control the disease. The control measures, different control measures, the larval control, people control, the male female mosquito control, pesticides, biological, chemical, all the things. So all the things where we should know about the where the really the disease which are all there, what type of technology we should use it in that place. Not like just like that, dengue is there, you take the fogging machine and go and fog it. No, we should not do it. So we should know about the entire disease about that particular place, then only we should react it. So diagnosis, treatment, cases, which are all very important. Disease surveillance, as we some of the things where we can go to the field, get the data, or some of the people, the disease people may come to you, we can get the data. So different way of control the larval fogging and other things. So now the tool of 21st century, which was put on few years back, the tool building, tool sharing, collaboration, training. We all the research, physics people were doing separately. Those days. Now all the things are integrated. Either physics, chemistry, maths, or the biologists, all the engineering people. They should know initially itself. That's what when we say that any of the project which you are taking, initially itself you plan it. Who are the people are involved? All the people who involve it. You want to make it any instrument for the diagnosis. Initially itself you involve. You will get a better result. You don't do research yourself and then bring to the engineer in the later period. So all the things initial itself, the planning is very important. Now everyone can contribute it for all the things, not only for the individual, that individual research has gone. Now we have to collaborate with everyone. We should know about each and every caliber involved initial itself, so that you will get a better result. So these are all the things when we see about the genomic surveillance, literature. So we know about, now we'll come to know easy to do it. Any of the gene, which gene is responsible, which from the bacteria, from the sand soil, initially the PCR, when we see it, we have to get the sample, do the PCR. No need to do the PCR also. Directly we know the sequence. These are all the sequence which are responsible for particular bacteria, are the pathogen, non-pathogen, what is existing, what are all the things which are all there. From the gene sequence, we will come to know without doing any other things. Not doing the elaborate way, the science which got improved a lot. So the conclusion part, I'll conclude almost took long time. So vector pathogen interaction, disease pathogen interaction, early information, and then classical part of all the things, National Institute as well as other colleges, university, we can control the disease easily. And also this last slide, which will show what to eat, what not to eat, when to eat, how to eat it. So we know the traditional food we have lost because of many other things, because of all, everyone has to go for job, the what's the junk food which we are all eating it. So we should stop it, all the things, so that our immune system will be the better. That's the immune system which will take care of all the things. Thank you very much for giving me a chance to do it, sir.
Thank you, sir.
if
Thank you, sir.
if
That was really a useful and worthwhile session to us. You are really a polite, So polite, actually, but your profile speaks a lot and shows a tremendous growth. Still, I want to say more about you and the funds that are being given to you by DRDO government funding of rupees 10 crores has been granted, and this is not to make you feel denim, but it makes us more inspiration and encouragement to us, or to all of us who take you as a role model from this conference. Once again, I thank you also.
Thank you, thank you, madam, for giving the things. And if you have any questions or anything, if I can answer for you,
Any
I'm
other
happy to answer.
questions? If there is any queries, you can ask now. Shoot out questions.
Sir, I have a question. I felt you should have spent a longer time on your last slide. It was about the eating habits. Could you please speak a few lines on what kind of eating habits we need to follow if we have to build a good immunity? Just a few words on that.
Can I, can I have?
Yes, yes.
Yes, sir. Because see, the traditional food, when we say it, because of COVID period, we also know the plant product, whatever the chile, tulsi, all the things we have taken it.
ஸ்லைடு ஆனால் முடிஞ்சிருச்சு சார் ஸ்லைடு முடிஞ்சிருச்சு
Hello.
ஸ்லைடு
Yeah.
பேசி முடிச்சு
Uh, which showed about even we have proved ourselves the see the way we were not having the skin cancer here in Tamil Nadu. If you see the south, manjal manjal தேய்ச்சி குளிப்பாங்க எல்லாம் skin cancer is no, there is no single case of skin. The curcumin, which is really is very good, sir. That's what the COVID which it used to take care of. We have showed, we have proved, we have published also the cancer cells. When you put the curcumin, the immediately within 24 hours all the cancer cells died, different cancer cells. We used about HD29 or other breast cancer cell lines, many of the cancer cell lines. Where the normal cells, the 83 cells, when you put it, there was no this one death on that one. So it shows about the clear cut, the traditional food, whatever the sambar masala, any of the all the masala which you used to have the, that's what they followed. Because COVID period we followed the same thing, what we should eat monthly, what are all the things. Because meat, I don't think anyway, I am pure vegetarian, I don't know about the meat, but still we used to work on meat and everything. Still we do the improve upon all the things. So you do have it everything on the plant products, what is required. The seasonal biotechnological, we have cloned it everything nowadays. We have seasonal fruits, seasonal vegetable used to be there. Then natural, which really in the summer season you do get plenty of pushinika, parenka, velerica, ardum, umarugaka. So what is required, each one has a meaning of that one natural has given. But we cloned it round the year. Manga, it's some other thing which has a pacha manga has a minerals, plenty of minerals that is required on summer only. So round the year we have changed the nature. That's the internal nature has changed in that one. That's the one thing. Then weekly, what to eat, what not to eat. It's rare, but anyway, the things with the fish now, the omega-3 which are all there, but I don't say the omega-3 is very good. Even you can take it whenever possible, because that's a system will not be there for long. That's what they have put it in that place. Another one is daily. What are all the things vegetable we should eat daily? It shows about, but things are all the pesticides now, the fertilizer now, because large quantity is produced. We have used the fertilizer, pesticides, all the things. But when you clean it, take it everything, that will be very good. And then the daily physical things, the rice, other things, yeah, which are all there daily, the physical. See, including my son, if I tell my son, you go and bring milk, he first question he will ask, where is a key? Means he wanted scooter. They don't want to walk. If you can able to walk, still wherever, I used to walk at minimum 10,000 steps per day. Wherever possible, if you can able to walk, first thing you do it. Wherever you be able to go by cycle, kindly go by cycle. The third part only use it for scooter, car, and everything. So when we do it, all the things, the immune boost. See, all the thing naturally we are all boosting. See, vitamin D deficiency, you will not get it. See, those who are we have studied some of the things. Those who are the building construction people never had any vitamin D deficiency. The school used to have the 1 day PT period. Now we don't have. So all the time when you walk on the evening, see, when you wanted to purchase milk, when you wanted fish milk or flour, I used to go by walk in the evening. Go there for half an hour. If I'm able to go there in the evening hours, go and bring it half kilometer, 1 kilometer, all that. That's what I am sorry. Don't take it otherwise. Any of the temple constructed those days, the temple is a big temple. You go in the walk in the temple in the name of God. You go there and then you will get a vitamin D. There won't be any vitamin D deficiency. That's what each and every step which is given, sir, natural. Sir, we have spoiled the natural intern. Natural is in the nature is taking care of us. But anyway, we may overcome. But COVID period, we have learnt a lot. Still, people started it. That our immune system is developing well, sir. Thank you very much, sir. I don't know. I have answered your all the things. Because everything is scientifically proved. Those days, my grandfather period used to tell, don't bring the chapel, don't wear the same dress, wash it, put it on the this. We followed everything. That's what we could contain the disease. So we have learned it. I think we have revert back everything the same old fashion, old thing, everything which will come to us, so that disease will not be there. Thank you, Mr.
Thank you, sir.
Thank you, sir. Kindly be seated, sir. Um, I invite Mrs. R. Suganya. Okay.
Now I invite.
கேக்குதா? இன்னைக்கு பார்த்துட்டு இருக்காங்க மேடம். இதுதான். So, good morning, everyone. It's a really honor to be here. So I thank the Vice Chancellor, the Dean, Dr. Kamaraj, everyone for inviting me. And this is the first lecture I'm addressing non-doctors. Most of the time we go to medical conferences only. It's the first time I'm giving a lecture to non-medical students. So it's not going to be a long lecture. It's short and sweet. So you can just give. It's just introduction. It's autoimmune disease is a very big topic. So what I'm going to say, just introducing you to what is the concept of autoimmune disease. So it has only 20-25 slides. So I won't. It will be interesting. Just keep your eyes and ears open. So VC sir has just given an introduction about the functions of immune system, how it is defending our body against many diseases. Defense is not the only function of our immune system. So of course you know that immune system defends our body against bacteria, viruses, and so many things. So it also does regular surveillance even before just a bacteria enters it. The surveillance system immediately just like police go for surveillance. Our immune cells also go for surveillance and removes this bacteria and toxins. And also it removes the tumors, not only against immune system acts against infections, it also acts against cancer cells also. So even for action against cancers, we need our good immune system. And so we know that immune system not only acts against these tumors and cancers, It plays multivarious roles. There is, you said that 16 foot intestine is there. The intestinal movement is also controlled by our immune system. The nervous system also plays, immune system is controlling it. So, how much energy intake we take every day, how much energy we spend every day, it's all controlled by the immune system plays multiple roles. So when we say defense, so it so we know that it defends against microbial bacterial pathogens and everything. So how this bacteria, this immune system identifies our own cells. So immune system should not attack our own cells. We have many proteins in our body. We have insulin, we have actin, myosin, so many proteins are there in the body. So our immune system does not act against our own cells. That should not happen. So our immune system develops tolerance towards our own self antigens. That is known as tolerance. Discrimination between self and foreign antigens is the most crucial step in our immune system. That's the most important step. If that system fails, our immune system will attack our own body. That is what we call as autoimmune disease. So when our immune system is not able to identify our own cells as our own cells, it thinks this is a foreign antigen. It attacks our own system. Then it is known as autoimmune disease. The tolerance refers to the ability to recognize self and limit autoimmune responses. So whenever this tolerance fails, we develop autoimmune disease. So what is immunological tolerance? There are two types: central tolerance and peripheral tolerance. So I think you have all learned some basic immunology. So there are some central lymphoid organs such as bone marrow and thymus, and peripheral lymphoid organs such as spleen and lymph node. So I'll try to not to use so many medical terms. So I'll try to use some simple immunological terms. I think most of you are aware of these simple immunological terms. So central tolerance, there's an organ called thymus. You should learn about human organs. There's an organ called thymus. Thymus is present in our chest, just behind this bone. That's an organ called thymus. That's a central lymphoid organ where our lymphocytes are produced and they are tuned to attack the attack the pathogens. So in this central lymphoid organs, uh, there is these lymphocytes are exposed to our self antigen. Well, in well, we are very small age, or even now, our own antigens are presented to the lymphocytes. So when these lymphocytes are presented to our own antigens, some of these lymphocytes undergo cell death. Apoptosis means cell death. The lymphocytes which are exposed to our own antigens, they die off. So they can't attack our own cells, our own antigens. So this is a very important step. So if this does not happen, this is regulated by various transcription factors. If there is mutation in these transcription factors, our own lymphocytes can't recognize our own antigens, they go and attack our cells. So this is central tolerance taking place in central lymphoid organs. So if the cells they don't recognize, some cells don't recognize our own antigen, so they won't attack our own cells. So they go to the periphery, they migrate to the periphery. So central and peripheral tolerance. Peripheral tolerance happens in peripheral lymphoid organs. So I told us bone marrow and the thymus are central organs. There are peripheral organs also. Peripherally, lymph nodes, spleen is there. You should have heard the name spleen. That's a peripheral lymphoid organ. These T lymphocytes and this go to the periphery. Some cells they don't, and they are not exposed to these self antigens in the central organs, so they need to be eliminated in the periphery. So this happens in the peripheral lymphoid organs. There are various mechanisms. I'll just go through one or two of the mechanisms. So in the periphery, because the lymph cells go to the periphery, they are exposed to self antigens, and these cells again they're eliminated by various mechanisms. The common mechanism is Fas and Fas ligand pathways. So whenever there's a cell is activated, presented to a self antigen, they gets activated. They secrete this Fas and Fas ligand. These cells they undergo apoptosis. Apoptosis means they undergo cell death. So our defensive lymphocytes, when they are exposed to our own antigens in the periphery, they undergo cell death. So if they don't undergo cell death, they'll go and attack our own cells. And ignorance. So there are certain organs in our body, they are not exposed to the lymphoid system itself, such as our lens. So if you see our eye lens, lens is there in our eyes. So they are not exposed to the lymphoid system itself. So our own lymph cells go, they can't recognize our lens antigen. So whenever there is injury, whenever there is injury to one eye, so they will develop redness in the other eye also. That's because this lens antigen enters the blood system because of injury. our lymphoid system is not exposed to this lens antigen right from our childbirth. So they think the lens is foreign antigen, they go and attack the lens. The lens is there in other eye also. They go and attack the lens also. Similarly, there are many these privileged sites. They what called immune privileged sites because they are privileged because these cells they can't replicate. So these cells are protected from our own immune system itself. Like testis, there's a disease called mumps. You would have heard of. There's MMR vaccine we put in childhood. Mumps attacks our mumps virus attacks our testis. Testicular antigens also not exposed to the lymphoid system. Whenever mumps attacks our testis, these testicular antigens are exposed to the lymphatic system. The lymphatic cells go and attack the other testes also. So this is an autoimmune reaction. So this brain, placenta. So pregnancy, this entire human body, which is different inside the mother's womb. But the immune system is not going to attack because whenever we get, so as a nephrologist, we do kidney transplant. We take a kidney from other people and transplant into the newer person. But what happens is our immune system goes and rejects the kidney. But our immune system does not reject. Mother's immune system does not reject the newborn fetus. இதே அந்த மதர் வந்து கிட்னியை கொடுக்குறாங்க அந்த குழந்தைக்குன்னா ரிஜெக்ட் பண்ணிடும் அதுக்கப்புறம். But the entire kidney, the human fetus, is inside the mother for 10 months. But the human system is not attacking the fetus because of this placenta is immune privileged site. So placental antigens cannot be recognized by the mother. So this is known as ignorance. Our immune system ignores certain antigens. Whenever this fails, we develop autoimmune disease. And again, this is another mechanism. I won't go into details of energy. So and immune regulation. Not only there are immune regulatory cells also. These regulatory cells they prevent autoimmune reaction through secreting interleukins or to cell inhibition. So these are all some. This is summarizing the peripheral tolerance. So the cells can be inactivated through energy or through apoptosis or through various mechanisms of cytokines. So this tolerance mechanism is antigen specific. In the antigenic tolerant and the antigen மட்டும்தான் tolerant இருக்கும். So it's antigen specific. We know that there are two types of lymphocytes, T and B lymphocytes. Tolerance develops for both T and B lymphocytes. It lasts longer in T lymphocytes. And so how does autoimmune disease? Some people develop autoimmune disease, and so there is a genetic predisposition. Certain families are genetically predisposed to develop autoimmune disease. So family experience autoimmune disease ku nammalukku varala autoimmune disease. So because of basic some HLA systems and cytokine pathways, environmental triggers. நம்ம இப்ப கோவிட் பேசிட்டோம். கோவிட் itself can trigger an autoimmune disease. நம்மளுக்கு ஏற்கனவே autoimmune predisposition இருக்காது. But sometimes an infection can trigger our immune system to act against our own cells. So various infections. இப்ப நம்ம ஓசோன்ல climate change சொல்ற ஓசோன்ல water. So those ultraviolet radiations can trigger an autoimmune response. They can have a genetic damage that can trigger an autoimmune response. And so these autoreactive T cells, and then there's a breakage of immune tolerant self antigens, and these cells go unregulated and they cause tissue damage. The antibodies and the T cells, they go and attack our own cells and they cause tissue damage. The kidney attack பண்ணிச்சுனா அது பேருக்கு kidney disease வரலாம். And there are various mechanisms how this gets triggered. இப்போ இந்த கோவிட் வந்துச்சு autoimmune, some COVID antigen can mimic our own antigen. So ஒரே மாதிரி இருக்கும்போது sometimes our immune cells they think that that's a similar foreign antigen இருக்குதுன்னா they go and attack foreign antigen along with our own antigen. So there are various mechanisms by which our own cells can attack our own cells. So attack பண்ணும்போது it can cause various symptoms. Stimulate பண்ண அந்த organ, can organ damage பண்ணிரலாம். So it can damage the insulin secreting pancreas by damage from autoimmune antibodies. So we develop diabetes. Insulin receptor damage, insulin இருக்கும் but insulin போய் act பண்ண முடியாது. So that cause insulin resistant diabetes. So various autoimmune diseases can happen by these mechanisms. So na kidney disease mechanisms மட்டும் ரெண்டு மூணு disease name சொல்றேன். So this is just the timeline of autoimmune disease. This initiation because of triggers, then there's a propagation of disease where entire disease happens, then resolves. Once autoimmune process சரியானோட it resolves. This is the timeline. The timeline over disease over மாதிரி இருக்கலாம். So it can may last for few days to few years வரைக்கும் இருக்கலாம். So எல்லாரும் பயாலஜி படிச்சிருப்பீங்கன்னு நினைக்கிறேன். சார் சொன்னார் there are kidney is a very small organ. There are this organ called glomerulus is in the kidney. There are 1 million glomerulus in each kidney. So 1 million இருக்கு. So every minute our blood is almost cleaned 5 times. Our entire blood is cleaned 5 times every minute. So by this organ called glomerulus, blood in the உள்ள போது clean ஆயிட்டு வெளில போயிடும். So this is the glomerulus. அதோட அல்ட்ரா ஸ்ட்ரக்சர் தான் அது. This is the blood which goes inside the glomerulus. இதுல இருந்து யூரின் வெளில வந்துரும். So இதுல இருந்து யூரின் வெளில வந்துரும். So 180 liters of urine is produced every day. 180 liters. அதில் வந்து 1 liter தான் நம்ம போகிறோம் வெளில. Everything is reabsorbed by this. So it's a very complex mechanism. So blood goes into this thing and it gets filtered. எல்லா waste productலாம் filter ஆகி normal blood goes back to the system. So it almost takes place every second of our life till our death. So என்ன ஆகும்னா what happens in autoimmune disease, இந்த செல்ஸ்லாமே வந்து நிறைய ப்ரோட்டீன்ஸ் இருக்கும் இங்கே. ஸோ அந்த நம்ம செல்ஸ் வந்து autoimmune ப்ராசஸில் வந்து இந்த செல்ஸ் கேன் கெட் டேமேஜ் ஆச்சுன்னா what we get is renal failure. So the antibodies can then this gets deposited. இதில் பார்த்தீங்கன்னா இந்த இடத்துல எல்லாம் நிறைய பேஸ்மெண்ட் மெம்பரேன் சொல்லுவோம். This is another podocyte, is capillary cell. So the antibodies can damage this sites. The antibodies வந்து இங்கே டெபாசிட் ஆயிடலாம். There can get antibodies can deposited. Antibodies can deposit in the basement membrane or between the basement membrane and the epithelial cells. This is on the blood cell. இங்கே அந்த இடம் இருக்குது. So this is the so antibody can attach to the basement membrane. In the moon disease, but they just share a few slides. These are all typical autoimmune disease affecting the kidney. So first we'll go to the antibody attached to the basement membrane. So first I'll talk about the SLE. SLE is a classical autoimmune disease. So multi-system disease. Autoimmune disease பொறுத்தவரை ஒரு single organ affect பண்ணலாம் இல்ல multiple organs affect பண்ணலாம். Multi-system இருக்கும். So SLE is a classical multi-system autoimmune disease. There is genetic factors, ultraviolet rays ரொம்ப important factor சொல்றாங்க. So என்ன ஆகும்னா damages our cell and nuclear antigens are released. So ஒரு செல் எவ்வளோ செல் நியூக்ளியஸ் இருக்கும், நியூக்ளியஸ் ஆன்டிஜென்ஸ் தான் இருக்கும். அந்த ப்ரோட்டீன்ஸ் தான் can get released into the system. அதுக்கு against antibodies டெவலப் ஆயிரும். So antibodies develop ஆயிடுச்சுன்னா antibodies go and gets deposited at here. The antigen antibody complex is going to get deposited in the kidney. So once the deposit ஆயிடுச்சு kidney இல்லைன்னா the kidney function gets damaged. அந்த glomerulus damage ஆயிரும். The antigen antibody complex damage ஆயிரும். So என்ன ஆயிரும், deposition immune complexes triggers inflammation. Inflammationனா அது வீங்கி அடிப்பட்ட வீங்கிருன்னு சொல்றோம்ல, அது மாதிரி இன்ஃப்ளேம் ஆயிரும். அந்த இடமே கிட்னி இன்ஃப்ளேம் ஆயிரும். ஸோ கிட்னி டேமேஜ் ஆயிடுச்சுன்னா எப்படி தெரியும், நம்ம பேஷண்ட்டுக்கு எப்படி தெரியும்னா ப்ரோட்டீன் லாஸ் ஆகும், கால் கை வீங்கிருச்சுன்னு வருவாங்க. இந்த மாதிரி டிசீஸ் ஆக்சுவலி அக்கர்ஸ் இன் யங் ஃபீமேல்ஸ். ஜென்ரலாக இந்த 20s 30s ஆக்சுவலாக அந்த ஏஜ் ஆஃப் ஃபீமேல்ஸ் அதிகமாக காமனாக இருக்கும். ஹார்மோன் ட்ரிவன் டிசீஸ் சொல்லுவோம். ஸோ திஸ் டிசீஸ் இஸ் காமன் இன் ஃபீமேல்ஸ், பர்டிகுலர்லி இந்த சைல்ட் பேரிங் ஏஜில். சைல்ட் பேரிங் ஏஜில் கிட்னி டிசீஸ் ஆயிடுச்சுன்னா ஈவன் pregnancy is difficult for these females. So அந்த protein loss ultimately lead to renal failure. The renal failure ஆயிடுச்சுன்னா ட்ரீட்மெண்ட் இஸ் அந்த இம்யூன் சப்ரஷன் சொல்லுவோம். அண்ட் இம்யூனோ சப்ரசிவ் ட்ரக்ஸ் கொடுப்போம், இம்யூனிட்டியை குறைச்சிரும். ஸோ நார்மலாக வந்து நம்ம இம்யூனிட்டி தேவைன்னு சொல்கிறோம். பட் ஆட்டோமின் டிசீஸில் இம்யூனிட்டி அதிகமாகிடுச்சுன்னு வச்சுக்கோங்களேன். இம்யூனிட்டி அதிகமாகி நம்ம பாடியை அட்டாக் பண்ண ஆரம்பிச்சு. அப்போ வந்து we have to suppress that immunity. We suppress immunity. இம்யூனிட்டி சப்ரஸ் பண்ணுற மூலமாக we regain the organ function. சில பேஷண்ட்டுக்கு ஆர்கன் ஃபங்க்ஷன் ரீகெயின் ஆகும், எல்லாத்துக்கும் ரீகெயின் ஆகாது. ஸோ ஒன்ஸ் ரீனல் ஃபெயிலியர் ஆச்சு தென் we have to do dialysis. டயாலிசிஸ் கேள்விப்பட்டிருக்கோம், டயாலிசிஸ் அண்ட் டிரான்ஸ்பிளான்டேஷன் போயிருவாங்க. அதுக்கப்புறம் this is one type of autoimmune disease where immune complex are formed in the body, gets deposited in the kidney. So வேற இடத்துல கிட்னிலே ஃபார்ம் ஆகிறதில்ல, வேற இடத்துல ஃபார்ம் ஆகி then it gets deposited in the kidney and leads to kidney disease. And பட் இது இன்னொன்று ஆன்டிபாடி வாஸ்குலைட்டிஸ் சொல்லுவோம். இது ஆட்டோமியூன் டிசீஸ். இந்த ஆட்டோமியூன் டிசீஸில் ஆன்டிபாடி நியூட்ரோஃபில்ஸ் கேள்விப்பட்டிருப்பீங்க, நம்ம டபிள்யூபிசி கேள்விப்பட்டிருப்போம். டபிள்யூபிசி நிறைய டைப்ஸ் ஆஃப் இருக்கு. நியூட்ரோஃபில்ஸ் டபிள்யூபிசி இருக்கு, ஒயிட் பிளட் செல்ஸ். நியூட்ரோஃபில்ஸ் நம்ம நார்மல் ரொட்டீன் இம்யூனிட்டிக்கு தேவை என்ன, அது ஒரு நியூட்ரோஃபில்ஸ். நியூட்ரோஃபில்ஸ்ங்கிறது வந்து இப்போ எல்லாமே ப்ளஸ் டூலேயே வந்துருச்சு. ஆல்மோஸ்ட் ப்ளஸ் டூ புக்கை பார்த்தோம்னா ஆல்மோஸ்ட் மெடிசன் புக் மாதிரி தான் இருக்குது, படிச்சிருப்பீங்கன்னு நினைக்கிறேன். நியூட்ரோஃபில்ஸ்லாம் நியூட்ரோஃபில்ஸ்ங்கிறது வந்து இந்த அதில் ஒரு ஆன்டிஜென்ஸ் தான் இருக்கும், இந்த புரோட்டீனேஸ் 3 மைலோபெராக்சிடேஸ்லாம். அதுக்கு ஆன்டிபாடிஸ் டெவலப் ஆகி அந்த நியூட்ரோஃபில்ஸ் கெட்ஸ் ஆக்டிவேட்டட். Activated neutrophils go and attack our own vasculitis na vascular na blood vessels, itis na inflammation. So blood vessels நம்ம போய் அட்டாக் பண்ணோம். நம்ம blood vessels போய் நம்ம neutrophils நம்ம ஃபாரின் ஒரு பாக்டீரியா அட்டாக் பண்ண neutrophils வந்து it goes and attacks our own blood vessels. Blood vessels in the kidney, in the lungs லாம் அஃபெக்ட் ஆகலாம். So patient அதே மாதிரி தான், they come with ரத்த வாந்தியோட வருவாங்க, they come with hemoptysis, urine blood in the urine and renal failure. So இது வந்து ஆன்டிஜன் ஆன்டிபாடி காம்ப்ளெக்ஸ் கிட்னியில் ஃபார்ம் ஆகிறதில்ல, டபிள்யூபிசியில் ஃபார்ம் ஆகி அந்த இது போய் கிட்னி அட்டாக் பண்ணுது. ஸோ நம்ம கொலாஜன், நீங்கள் கொலாஜன் படிச்சிருப்பீங்க, கொலாஜன்லாம் இருக்கு. கொலாஜன் இஸ் அ ப்ரோட்டீன் சீன் இன் அவர் பேஸ்மென்ட் மெம்பரேன். அதுக்குமே வந்து அதுல நிறைய டைப்ஸ் ஆஃப் இருக்குது. அதுக்கு ஆன்டிபாடிஸ் ஃபார்ம் ஆகலாம். கொலாஜன் நம்ம குளோமருலசன் படிச்சோம்ல, பேஸ்மென்ட் மெம்பரேன் காட்டினேன் உங்களுக்கு. ஸோ இந்த பேஸ்மென்ட் மெம்பரேன் பார்த்தோம். இந்த பேஸ்மென்ட் மெம்பரேன்ல அந்த ஒயிட் கிரீனிஷா இருக்கு பேஸ்மென்ட் மெம்பரேன். தே ஹேவ் லாட் ஆஃப் ப்ரோட்டீன்ஸ். அந்த ப்ரோட்டீன்ஸ் கேன் கெட் எக்ஸ்போஸ், பர்டிகுலர் ஸ்மோக்கர்ஸ். நம்ம ஸ்மோக்கிங் பண்ணக்கூடாது அடிக்கடி சொல்லுவோம், ஸ்மோக்கிங் பண்ண கெட்டது. ஸ்மோக்கிங் கேன் ட்ரிகர் மெனி டிசீசஸ் ஆக்சுவலா, லைக் ஹார்ட் அட்டாக், அண்ட் ஸ்மோக்கிங் கேன் காஸ் ஆட்டோஇம்யூன் டிசீஸ் ஆல்சோ. ஸோ ஸ்மோக்கிங் பண்ணுறது கரெக்டாக ஸ்டாப் பண்ணுங்க. ஸ்மோக்கிங் கேன் காஸ் ஆட்டோஇம்யூன் டிசீஸ். ஸ்மோக்கிங் கேன் டேமேஜ் இந்த மாதிரி லங்ஸ்ல இந்த மாதிரி இந்த மெம்பரேன் இருக்கும். இந்த லங்ஸ்ல உள்ள இந்த மெம்பரேனை போய் ஸ்மோக்கிங் வந்து டிஸ்டர்ப் பண்ணிச்சுன்னா அந்த ஆன்டிஜென்ஸ் வில் கெட் எக்ஸ்போஸ் டு தி பிளட் சிஸ்டம். நம்ம சிஸ்டம் வந்து இந்த ஆன்டிஜென்ஸ்லாம் பார்த்துருக்காது. இந்த லிம்போசைட்ஸ் சடனாக பார்த்த உடனே ஃபாரின் ஆன்டிஜென் நினைச்சிட்டு தே கோ அண்ட் அட்டாக் திஸ் பேஸ்மென்ட் மெம்பரேன். ஸோ when they go and attack this basement membrane and they get pulmonary hemorrhage and rapidly progressive renal failure. ஸோ லங் லைன் டேமேஜ் ஆகும், சைமல்டேனியஸ் ஒரே நேரத்தில் டேமேஜ் ஆகும். பேஷண்ட்ஸ்லாம் பார்த்தோம்னா நம்மளுக்கே பயமா இருக்கும். They'll spit out blood, at the same time they'll have blood in the urine also, and they have renal failure. So this is known as good pastor syndrome சொல்றோம். Goes both to renal failure and lung hemorrhage. So இந்த மாதிரி நிறைய ஆட்டோஇம்யூன் டிசீஸ் இருக்குது. ஸோ தே ஹேவ் வேரியஸ் பிரசன்டேஜ். எந்த ஆர்கனை ஆட்டோஇம்யூன் டிசீஸ் அஃபெக்ட் பண்ணுதோ அந்த மாதிரி அந்த பிரசன்டேஷன் வேரி. பேங்க்ரியாஸ் அஃபெக்ட் பண்ணி டயபெட்டிஸ், கிட்னி அஃபெக்ட் பண்ணி ரீனல் ஃபெயிலியர். Autoimmune disease can affect the brain also. Can affect the brain, lungs, heart, என்ன ஆர்கன்ஸ் அஃபெக்ட் பண்ணலாம். ஸோ ப்ரிவென்ஷன் சொன்னபோது நத்திங் ஸ்பெசிஃபிக். டயபெட்டிஸ் எப்படி ப்ரிவென்ட் பண்ணலாம் சொல்லலாம். ஹெல்தி டயட்டை பற்றி மென்ஷன் பண்ணாங்க. 2-3 பாயிண்ட் நம்ம ஹெல்தி டயட்டுக்கு இது பண்ணி பண்ண லைஃப் ஸ்டைல் சேஞ்சஸ் சொல்லுவோம். ஸோ ஃபிசிக்கல் ஆக்டிவிட்டி இட்ஸ் வெரி இம்பார்ட்டன்ட், அண்ட் மெயின்டைனிங் ஹெல்தி வெயிட். இந்த ரெண்டு வெரி இம்பார்ட்டன்ட் டு மெயின்டைன் நார்மல் இம்யூன் சிஸ்டம் ஆல்சோ. ஸோ மெயின்டைனிங் ஹெல்தி வெயிட் அண்ட் ஆக்டிவ் லைஃப் ஸ்டைல். அட்லீஸ்ட் ஒரு half an hour of brisk வாக்கிங் இப்போ நிறைய பேர் போவோம், பட் ஃபோன் பேசிட்டு மெதுவாக நடந்துருப்பாங்க. தட் இஸ் நாட் சஃபிஷியன்ட். ஸோ brisk வாக்கிங் அண்ட் மெயின்டைனிங் ஹெல்தி வெயிட் இஸ் இம்பார்ட்டன்ட் ஃபார் குட் இம்யூன் சிஸ்டம். அண்ட் டயட் என்ன சாப்பிட்டா இம்யூன் சிஸ்டம் கூடுன்ட்டு நிறையா இருக்கிறதோட என்ன சாப்பிடக்கூடாதுன்னா வெரி சுகரி டயட்ஸ், அதை மட்டும் அவாய்ட் பண்ணணும். நான் டயபெட்டிஸ் இருக்கோ இல்லையோ, சுகர் இன்டேக், சிம்பிள் சுகர்ஸ் பர்டிகுலர்லி ரொம்ப நம்ம கோக், பெப்சி அந்த மாதிரி ஃப்ரூட் ட்ரிங்க்ஸு, சிம்பிள் சுகர்ஸை நம்ம அவாய்ட் பண்ணுறது ரொம்ப முக்கியம். ஸோ காஃபி டீலையுமே வந்து போட்டோம்னா 2-3 கரண்டி சுகர் போட்டு சாப்பிடுவாங்க. அதெல்லாம் தவிர்க்கணும். ஒரு கரண்டி அல்ல ஹாஃப் கரண்டிலே பழகிடலாம். ஸோ மினிமைசிங் சிம்பிள் சுகர்ஸ். சிம்பிள் சுகர்னா நம்ம இந்த வெல்லம் இதெல்லாம் சொல்லணும். நிறைய பேர் நாட்டு சக்கரை நல்லது, நிறைய அள்ளி போடக்கூடாது. நாட்டு சக்கரை நல்லதுனாலுமே லிமிட்டடாக தான் இருக்கணும். சிம்பிள் சுகர்ஸ், brisk activity, maintain a healthy diet, will have a very good immune system. But அது autoimmune disease prevent பண்ணுமா இல்லையான்னு சொல்ல முடியாது. Autoimmune disease has various other triggering factors like environmental factors, other triggering factors தான் இருக்குது. So once you develop an autoimmune disease and that needs medical treatment. So kidneys are the most common organ involved in autoimmune disease. நிறைய ரீசன்ஸ் இருக்குது. கிட்னி நம்ம 20% ஆஃப் த கார்டியக் ஹார்ட்லேருந்து வர பிளட் ஃப்ளோ கிட்னிக்கு தான் போகுது. கிட்னி ஸ்மால் ஆர்கன் தான். பிகாஸ் கிட்னி இஸ் இம்பார்ட்டன்ட் ஆர்கன்ஸ். பிகாஸ் நம்மளோட கழிவுப்பொருள் எல்லாமே கிட்னிஸ் எலிமினேட் பண்ணிட்டே இருக்கு எவ்ரி செகண்ட்ல. ஸோ மோஸ்ட் ஆஃப் த பிளட் என்ன சாப்பிட்டோமோ என்ன இன்ஜெக்ட் பண்றோமோ ஃபர்ஸ்ட் கிட்னிஸ் தான் போகும். ஏதாவது அதில் வேஸ்ட் ப்ராடக்ட் இருக்கு எலிமினேட் பண்ணணுமா என்னன்னு லிவர் அண்ட் கிட்னிஸ் தான் மோஸ்ட்லி போகும் ஃபர்ஸ்ட் எடுத்த உடனே. ஸோ அதனால கிட்னிஸ் ஆர் இம்பார்ட்டன்ட் ஆர்கன்ஸ். ஸோ டார்கெட் ஆன்டிஜென்ஸ் ஆர் தேர் இன் தி கிட்னி. So once this autoimmune process triggered, kidney failure can result in kidney failure. That may need to replacing the kidney transplant therapy. So this is just an introduction to autoimmune disease. As a biotechnology student, you'll be working on so many projects. Autoimmune disease is a very exciting topic. Various research possibilities in this areas also. Thank you.
Thank you, sir. Is there any queries, students, participants? Thank you, sir. Thank you for giving a detailed explanation about autoimmune kidney disease. Still more about our guest, Dr. L. Sendil Nayagam, nephrologist, SB Hospital, Thanjavur. I'm really very happy to share the profile of our guest in front of you. He has completed his MBBS in Madurai Medical College in the year 1999. He has completed his MD in PGI, Chandigarh in the year 2002. He has completed his DM Nephro in the year 2006. He has lifetime membership in International Society of Nephrologists. Indian Society of Nephrologists, PDSI, Indian Society of Organ Transplantation, American Society of Nephrology, Transplantation Society. He has done routine internship in Madurai Medical College, Madurai from 1998 to 1999, and from 2006 he is a consultant nephrologist in transplantation physician in SB Hospital, Thanjavur till now. He has published 4 scientific papers. He got Rajkumari Amrit Karoor Gold Medal Achiever in MBBS. Thank you, sir, once again. Thank you for coming with us. Next.
It's time for photo session. Kindly walk forward near the flux. I invite all the dignitaries to be there, followed by all the participants, staff, and students. Dear online participants, we will be resuming after 15 minutes.
My dear students, parallel பக்கத்தில் என்எஸ்எஸ் சார்பாக டென்டல் முகாம் நடைபெற்று கொண்டிருக்கிறது. அங்கேயும் ஒரு டாக்டர் வந்திருக்கிறாங்க. உங்களுடைய வாய்களை எவ்வாறு சுத்தமாக வைத்துக் கொள்வது என்பதை அவங்க வந்து இப்போ அறிவுரையாற்றினாங்க. இங்கே இம்யூனாலஜி இஸ் கோயிங் ஆன் பேரலல் டு த ஹவு டு ப்ரொடெக்ட் அவர் மவுத், நாட் ஓன்லி இன் டாக் பட் ஆல்சோ இன் கிளீன். அதனால் வாயை எப்படி சுத்தமாக வச்சுக்கிறது அதற்கானது நீங்கள் போய் உங்களுடைய என்ன ட்ரீட்மெண்ட் எடுத்துக்கலாம், அட்வைஸ் பண்ணுறதுக்கு ரெடியாக இருக்கிறாங்க. என்னென்ன ட்ரீட்மெண்ட் வல்லத்தில் உள்ள டாக்டர் தான், நீங்கள் ஆன்தவே எப்போ வேணாலும் செக் பண்ணிக்கலாம். அடுத்து ஏ பிளாக்ல தான் நடந்துட்டுருக்கு. அதனால் நீங்கள் அந்த இது இன்டர்வலில் வந்து அந்த முகாம்லையும் போய் உங்களுடைய வாயை செக் பண்ணிக்கோங்க. வாயை எப்படி ஒழுங்காக வச்சுக்கிறது, பேசுகிறது மட்டும் இல்லை, சுத்தமாக எப்படி வச்சுக்கிறது பற்றியும் அதில் சொன்னதை ஒரு செய்தி மட்டும் நான் சொல்லிடுறேன். பல்லெல்லாம் நீங்கள் எப்போ வளர்க்குறீங்க? காலையில் தானே. அதாவது இன்றைக்கி காலையிலேருந்து சாப்பிட்டு பல்லு இடுக்குள்ள எல்லாம் போய் அதை அழுகி எல்லா ஃபேக்ட்ரியும் உட்காந்து அது சாப்பிட்டு நம்ம ஈரலை பதப்படுத்தின பிறகு தான் பல்லு விளக்குறோமா. ஆனால் அவங்க சொன்னது நைட்டு பல்லு விளக்குங்கன்னு சொன்னாங்க. அந்த செய்தியை கொடுத்துட்றேன். பல்லு எவ்வளோ நேரம் விளக்குறீங்க? ஆமாம், நான் கிராமத்துலேயே வந்தால இவ்வளோ பெரிய குச்சியை வச்சுட்டு விளக்க ஆரம்பிப்பாங்க. இந்த வயக்காடெல்லாம் சுற்றிட்டு அந்த குச்சி முடிகிற வரைக்கும் விளக்கிட்டு இருப்பாங்க. ஸோ பல்லு என்னாமல்லாம் அவங்களுக்கு போயிடும், அப்புறம் பல்லும் போயிருக்குது. அதனால் 1 நிமிடம் தான். நீங்கள் ஸ்டார்ட் பண்ணிக்க. பண்ணிவிடுங்க, போக சொல்ல. நீங்கள் இது முடிங்க. நீங்கள் ஏற்பாடு பண்ணிகிட்டே இருங்க. ஜஸ்ட் டு என்கேஜ் தம். நீங்கள் அதை ஏற்பாடு பண்ணுங்கள். நீங்கள் ஏற்பாடு பண்ணிகிட்டே இருக்கலாம். ஜஸ்ட் ஐ வாண்ட் டு என்கேஜ் வாட் ஐ ஹேவ் டாக்ட். அங்கே என்ன பேசணுங்கிறத உங்களுக்கு அந்த செய்தியை போகாமலே கொடுத்துட்ருக்கேன். அவ்வளோதான், சரியா? அதனால் அவங்க சொன்ன செய்திகள் அப்ஸ்ட்ராக்ட் மட்டும் 2-3 சொல்கிறேன். பல்ல எப்போ விளக்கணும்? 1 நிமிடம் விளக்குனா போகிறோம். பல்ல இப்படி விளக்கக்கூடாது. இப்படி விளக்குனா இல்லை என்ன செய்கிறீங்க, இந்த பல்லு இடையில உள்ள நல்லா குத்தி உள்ளே வைக்கிறீங்கன்னு அர்த்தம். பல்ல இப்படி ரவுண்டில் கிளாக் வைஸ் ஆண்டி கிளாக் விளக்கணும்னு சொன்னாங்க. அந்த செய்தியும் எடுத்துக்கோ. என்ன சாப்பிட்டாலும் உடனே வாய்க்கு பிடிச்சி உள்ளே சாப்பிட்டுடு, இல்லைன்னா துப்பிடு. அது இஷ்டம். ஒரு 3 தடவை செஞ்சிரு. உள்ள பல்லு நடைபெற்று இருக்குது. அதே சமயம் அங்கேயும் வந்து இம்யூனாலஜி பற்றி தான் அங்கேயும் நடத்துகிறாங்க. அதனால கொஞ்சம் அதுவும் பயனுள்ளதாக தான் இருக்குது. ஸோ வி ஆர் ஹேவிங் ஏ பேரலல் ப்ரோக்ராம். ஓகே, வாய்ப்பு கிடைக்கும் பயன்படுத்தி.
Kindly arrange there for photo session. Participants also can be there.
Hello. 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 தம்பி ஒன்றும் இல்லை, இப்போ யாரும் அட்மிட் கேட்டால் மட்டும் அட்மிட் கொடுத்துக்கோ. கொடுத்துக்கோன்னா அவங்களே இது பண்ணிப்பாங்க. அவங்களே ஃபஸ்ட்டு வரவங்க அவங்களே பிபிடி ஷேர் பண்ணிடுவாங்க. செகண்ட் வரவங்களுக்கு தான் பிபிடி வந்து இங்கே நான் கொடுத்துட்றேன். சார் சொல்லுங்கள் சொல்லுங்கள். சாரை பார்க்கவா? இந்த வரேன். கூகுள் மீட் ஐடி வேறு ஐடி வேணுமா? இப்போ வரப்போது. சரி சரி. இங்கே தான் நம்ம அட்மிட் கொடுக்கணும். அந்த பையன்கிட்ட சொல்லியிருக்கேன். இல்லை அட்மிட் எனக்கு வரலையே, அவரை ரெக்வஸ்ட் கொடுக்க சொல்லுங்கள். ரெக்வஸ்ட் கொடுத்தாருன்னா இங்கே அட்மிட் கொடுத்துருவாரு. அவரை அப்படியே ஸ்லைடை அவரே ஷேர் பண்ணி ப்ரெசென்ட் பண்ண சொல்லுங்கள். லாஸ்ட் பர்சனுக்கு மட்டும் சொல்லுங்கள், நான் அந்த பிபிடி போடுறேன். லாஸ்ட் பர்சன் பிபிடி ஹெட்டிங் வந்து இதானே ஃபைனல் ஹெச்எல்ஏ. ஓகே ஓகே ஓகே, அதை நான் தம்பிகிட்ட சொல்லியிருக்கேன். இதை இவர் நீங்கள் எனக்கு அட்மிட் கொடுக்க சொல்லுங்கள். சரி சார். மதியத்துக்கு தான் சார் வேறு ஐடி கேட்குறாரு. லஞ்சு முடியணும். லஞ்சு கேப்பில் நான் வேறு இன்னொரு கூகுள் மீட் ஐடி கிரியேட் பண்ணி நான். ஓகே சார். கர்சரு கர்சரு அந்த பிளிங்க் ஆகுதில்ல, தெரியுதா? மவுஸ் மாற்றினா அந்த வருது இல்லை, கர்சர் வராமல் இது மட்டும் வருது. மணிவரன் எங்கே இருக்கீங்க? இல்லை பார்த்துட்டு நீங்கள் இங்கே வந்துடுங்க. ஸ்மார்ட் கிளாஸ் வந்துடுங்க. எனக்கு நிறையா மேலே நிறையா கால் வந்துட்டு இருக்கு. ஸ்மார்ட் கிளாஸ் வந்துடுங்க. அந்த பையன் பக்கத்துக்கு உட்காந்துருங்க. கூகுள் மீட் நம்பரா? இந்த வாட்ஸ்அப் பண்ணி வராங்க. நம்ம ஹார்ட்வேர் டீம்லேயே இருக்குமே. ஹார்ட்வேர் டீமில் அந்த கூகுள் மீட் ஐடி இருக்குல்ல, அந்த அந்த லாஸ்ட்டு அந்த கோடை மட்டும் எடுத்து போடுங்க. அந்த அந்த குரூப்பில் ஓப்பன் பண்ணுங்களேன். ஹார்ட்வேர் டீமை ஓப்பன் பண்ணுங்களேன். ஆமாம், அந்த ஜேவிஓன்னு ஆரம்பிக்குதுல்ல, அந்த ஜேவிஓலேருந்து எடுத்து அந்த கூகுள் மீட்டில் போட்டு கனெக்ட் பண்ணிக்கோங்க. சரி. இன்னும் வர்றது எல்லாமே ஒரே விண்டோ தான். சரி. கூகுள் மீட்லேயே தான் எல்லாம் ப்ரெசென்ட் பண்ணுவாங்க. ஃபஸ்ட்டு வர பார்ட்டி வந்து கூகுள் மீட்லேயே இந்த ப்ரெசன்டேஷன் ஷேரிங் இருக்குல்ல, கர்சர் வர மாட்டேங்குது. நம்மளாக தான் கொஞ்சம் மேனேஜ் பண்ண வேண்டியதாக இருக்குது. சார் இந்த வரேன். நீங்கள் லைப்ரரி வச்சு பேசிகிட்டு இருக்கேன், நான் அந்த வந்துடுறேன். அவங்க அருள் சார் வச்சு பேசிகிட்டு இருக்கேன். இந்த நான் வரேன், இருங்க ஒரு 10 நிமிஷம். இல்லை என்ன வரேன் சார். சார் கொஞ்சம் இருங்க சார், இந்த வரேன். நீங்கள் சொல்லுங்கள் சார். ஆமாம், வரேன் மூவ் பண்ணுறேன். அட்மிட்டுக்கு ரெக்வஸ்ட் கேட்குறாங்களா? அந்த ஒரே நிமிஷம் நான் ஃபஸ்ட்டு உள்ளே வந்துக்கிறேன். எங்கே இருக்கிறாங்க சார்? சி பிளாக் செமினார் ஹாலா? அந்த யூடியூப் யூஆர்எல் தூக்கி போட்டுருங்க, இல்லைன்னா கூகுள் மீட்டில் அங்கே இருக்கிறவரை கூகுள் மீட்டில் வர சொல்லுங்கள். ஆமாம் 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 குரு. ம். ஓகே ஓகே சார். ரெஃப்ரெஷ் பண்ணுங்களேன். ரெஃப்ரெஷ் கொடுங்க. இல்லை அந்த யூடியூப்பில் அந்த லிங்க்கை ரெஃப்ரெஷ் கொடுங்க. மூணாவது பலமுறை நான் அந்த லிங்க்கை ஷேர் பண்ணேன்னே.
Good afternoon.
சார் குட் ஆஃப்டர்நூன் சார்.
Good afternoon.
Sir,
Am I audible?
5 minutes, sir. We will make the
Okay,
program, sir.
no
Yes,
problem.
yes, sir.
Previous speaker போயிட்டு இருக்காரோ?
இல்லை முடிச்சிட்டாங்க சார்.
சரி சரி.
ஓகே. அங்கே அவங்களுக்கு கால் பண்ணி கொடுங்க சார். நான் கால் சார். இப்போ இந்த மீட்டிங் வந்து அங்கே லைவுக்கு வரணும், அப்படி தானே? அந்த ப்ரெசன்டேஷனில் வரணும். அந்த கொடுத்துட்டு அந்த மானிட்டரில் கட் பண்ணிட்டு அந்த ப்ரொஜெக்டர் இதை இதுக்கு மாற்றி விட சொல்லுங்கள் சார். யுவராஜ் இருக்காரா? யுவராஜ்கிட்ட கொடுங்க. யுவராஜ், இப்போ அந்த ப்ரொஜெக்டருக்கு தனியாக மெஷின் போட்டிருந்தீங்கல்ல, அந்த விஜே கேபிள் எடுத்துகிட்டு இதுக்கு கொடுத்துருங்க. கூகுள் மீட்டில் வரீங்கள்ல, அதில் விஜே கேபிளை கொடுத்துருங்க. ஆமாம், அதுலேருந்து எடுத்து இதில் கொடுத்துருங்க. இல்லை இப்போ நான் சொல்கிறேன் மட்டும் செய்யுங்களேன். ப்ரொஜெக்ட் கனெக்ட் பண்ணிட்டீங்களா, அதில் கொடுத்துக்கோங்க. நீங்கள் எல்சிடி ஸ்க்ரீனை பார்த்து நீங்கள் ஒர்க் பண்ணிக்கோங்க. இனிமேல் எல்லாமே வெறும் கூகுள் மீட் மட்டும்தான், கேமராலாம் வேண்டியதில்லை. இவர் வந்து அவரே பிபிடி ஷேர் பண்ணிடுவார். அடுத்த இதுக்கு நம்ம தான் பிபிடி ஷேர் பண்ணலாம். ஷேர் பண்ணுறதுக்கு இங்கே இருக்க ஆப்ஷன் ஃபஸ்ட்டு பிபிடியை ஓப்பன் பண்ணி வச்சுக்கணும். நான் ஃபைல் இங்கே ஓப்பன் பண்ணி வச்சுருக்கேன். இந்த இடத்துல இதில் போயிட்டு விண்டோவில் போயிட்டு அந்த பிபிடியை வந்து இதில் இருக்கணும். இப்போ வச்சுருக்காங்க பிபிடியை ஓகே கொடுத்துட்டு ஷேர் கொடுத்தீங்கன்னா நான் பிபிடி ஷேர் ஆகும். அதை பார்த்து அங்கே இருக்க ப்ரெசென்ட்டாக பேச ஆரம்பிச்சுப்பார். அது அப்படியே லைவில் போயிட்டு இவருக்கு தான் லாஸ்ட்டு பீப்புளுக்குன்னு சொன்னீங்க.
சார் ஸ்டார்ட் பண்ணிடலாமா சார்?
Yes, you can proceed.
We have in our presence Dr. K. Balakrishnan. We welcome you, sir. It's my pleasure to introduce you, Dr. K. Balakrishnan, who formerly was the professor and head, Department of Immunology since 2015. He has completed his PhD from the same department, Madurai Kamaraj University, in the year 1992. Now, he is working as a director and consultant immunologist in Madurai HLA Center, care of Madurai Kidney Center and Transplantation Research Center. Cleared UGC NET JRF examination in 1986 and awarded Robert Cochrane Youth Overseas Travel Fellowship from Royal Society of Tropical Medicine and Hygiene, London, UK, for training in microsatellite mapping in leprosy families at Wellcome Trust Center for Human Genetics. His research is HLA immunogenetics. Currently, Professor Balakrishnan is working on immunogenetic polymorphism and their susceptible and protective associations in a number of diseases such as type 1 diabetes, type 2 diabetes, adult and pediatric kidney failure, coronary artery disease, ischemic stroke, autoimmune thyroid disease, rheumatoid arthritis, cervical cancer, Parkinson disease, and other selected neurological diseases. Dr. Balakrishnan has published more than 55 research papers in reputed international journals, including Nephrology, Nature Genetics, PNAS, Journal of Infectious Diseases, Genes and Immunity, Biochemical Genetics, Immunogenetics, Diseases, Infection, Genetics, and Evolution, to name a few. Several workshops and conferences. He is offering PCR SSP based on tissue typing and donor matching services to more than hospitals for life and cadaver kidney transplantation under the industrial consultancy from 2014 to 2019. As on March 2021, an amount of rupees 1.5 crore has been generated through this service. Dr. Balakrishnan has held a number of administrative positions at Madurai Kamaraj University, such as Special Officer, Dean, Faculty of Science and Director, International Quality Assurance Cell, to name a few. He has successfully guided 15 PhDs and one student are working currently. In addition, Dr. Balakrishnan is the Secretary, Indian Immunology Society, New Delhi since 2022 to till date, and previously he acted as Vice President of Indian Immunology Society, New Delhi since 2017 to 2018 to date. He has visited countries such as UK, France, China, USA, and Singapore to undertake workshops. He has organized annual conference for Indian Immunology Society, ImmunoCon 2014, at Madurai Kamaraj University, in which 250 participants from 20 states and 5 countries have participated. He has contributed popular writings on immunology, HLA immunogenetics in national and regional newspapers. He has delivered more than 100 invited guest lectures and acted as a member in board of studies, zoology, biotechnology, microbiology, biotechnology, human genetics, forensic science in various universities and colleges. He has more than 25 research and 32 years of teaching experiences. With this introduction, I welcome Dr. Balakrishnan to present his talk on HLA in infectious diseases.
A very good afternoon to everyone. And outset, I really thank the management and administrative authorities of PRIST University, Thanjavur, and the organizing secretary, Dr. Kamaraj, for making these nice arrangements in this university and for the benefit of students and faculty members who registered in a big way. So thank you one and all at the outset. So, as and also I thank the introducer who gave really elaborate introduction about me. That is not needed in such situations because there is a crunch of time in making these sessions to complete in timings. So, with this introduction, I really wanted to go into my topic. Can I have the next slide? The topic of my today's talk is HLA and infectious diseases. I will try to. Am I audible? Someone please let me know. Okay. So the today's topic is HLA and infectious diseases, and you please closely follow me. The person who is operating the computer. I'll tell you the next slide. Can I please have the next slide? Yeah. So our health is being controlled by the genes which are present in 23 pairs of chromosomes as well as a number of environmental factors which includes food, infection, hygiene, environmental hygiene and other conditions. So genes and environmental factors really controls the human health. Next slide. So I need the slides quickly, please. So there are a lot of communicable diseases. To name a few very important communicable diseases which are affecting global populations includes malaria, TB, diarrhea, acute respiratory diseases, AIDS and others. Next slide please. Yeah, so this is an elaborate list, but of course this is not a comprehensive, complete list. To name a few diseases, tuberculosis is caused by Mycobacterium tuberculosis, which is a bacteria. Similarly, leprosy, M. leprae, by leprae, and AIDS by HIV, which is a virus, and malaria, which is a parasitic, infested diseases caused by Plasmodium falciparum bacteria, and smallpox again by a virus, and anthrax, which is a bacteria caused by a bacteria called Bacillus anthracis. So, by which one can able to understand all the microorganisms. Be it there are fungal diseases which I have not included. So, all the microorganisms really cause the disease. Can I have the next slide? So this is a picture, very complex picture. You can able to see several diseases like monkeypox, anthrax, dengue, yellow fever, cholera, Ebola, Marburg hemorrhagic fever, plague, Nipah virus infections, H5N1, and we are having about SARS. So these are all the outbreaks, global outbreaks in different countries across continents last 20, 30 years, which is because of the human activities again, because of the human intervention, we are destroying the nature. So these microorganisms causing these diseases are normally present in variety of wild animals, which they jump into human species, which is called as a species jump. That's what really lead to the present day COVID-19 pandemic, which is similar to SARS-related viruses. Can I have the next slide? So this is a beautiful point I want to stress in this lecture. So the viruses are and the receptors in humans are thrilling, really. You see, in HIV virus, which is a virus which is having human proteins, CD4 proteins or CCR5 protein, CXCR4 proteins as receptors for the re-entry. Similarly, for dengue, that is mannose binding receptors. This is a sign, and similar proteins are acted as a human proteins. These are all proteins present on the human cells, right? Cell membranes. So these proteins are acted as receptors to attract infection or attach the viruses onto the cell membrane. Similarly, you can able to see a variety of other viruses like polio, HPV, EBV. Say, for instance, human papilloma virus, which is affecting women predominantly in underdeveloped and developing countries, which is having alpha 6 integrin cell membrane on human epithelial cells, really attracts the virus. Similarly, for Ebola, it is a TIM protein present on cell membranes. And you all pretty well know that the recent coronavirus infection, human the protein which is attracting the virus to attach with the cell membrane is angiotensin converting enzyme receptor 2, ACE2. So these proteins which are present in humans really acted as a receptor for these viruses. So there is a correlation between the viral infection and the proteins which are expressed predominantly in specific cells of the human tissue. Can I have the next slide? And this is another slide in which there is a correlation between the bacterial and fungal and as well as the viral infections with reference to specific antigens. So, for instance, if you take ankylosing spondylitis, which is a disease affecting the cervical, vertebral column, including several bones and neurons, which is associated with the specific type of antigens, especially B27 and type 1, 2, 4, and collagens. And there is a similarity between these antigens and the epitopes induced by the organisms such as Klebsiella pneumonia and chlamydia. So the molecular mimicry of the antigens present in these pathogens as well as the host antigens really makes the individual susceptible whenever the individual is getting the infection from these pathogens. So you are similarly having variety of host antigens like myelin basic protein in the case of corona, measles, and mumps, EBV, herpes viral infection, as well as beta 2 glycoprotein 1 in case of Haemophilus influenza, Neisseria gonorrhea infections. So these slides, the past two slides, one can understand what is a tight linkage between the host proteins as well as the viral attachment sites as well as viral infection favoring agents. Can I have the next slide? Next slide. These are the other host factors which favors or which promotes, which accelerates the pathogen transmission. Age of the individual, immune status, illness or infirmity, host genetic factors, pregnancy, nutritional status, demographic factors, and social and behavioral traits all influence the pathogen infection as well as transmission. Next slide. Next slide, please. So the universal fact with reference to immune response and infection is not all infected got the disease, right? So if you travel, if 100 people travel in a train or 1,000 people travel in a train, next day some 100 individuals or 50 individuals may get the fever. Or if you travel in a bus, some 50 people, some of you get a fever and many of not getting the diseases. So that is a very important dictum or theory behind the infection immunity and protection concept. Not all infected got the disease. It is because of the variation in the immunogenetics, genetics, and other host physiological factors. Or if you study about the pathogen genetic factors, human genetic factors, and other important environmental factors, all these called collectively being called as infectomics. very recent area in which people study in a multi-pronged approach the infection and immunity. Can I have the next slide? So, if you take the closer look at the immune response in humans, the cells of immune response are broadly divided into myelin progenitor cells. Cells are originated from myeloid stem cells and lymphoid stem cells. In lymphoid stem cells, the cells which are developed out of lymphoid progenitor cells or stem cells includes natural killer cells, T helper cells, cytotoxic cells, B cells, and dendritic cells. Whereas in the other stem cell, like myeloid progenitor stem cells, leads to the development of cells such as dendritic cells, monocyte macrophage lineage cells, neutrophils, eosinophil, basophil, and platelets. So this is basically, these are all the two broad category of important cell types involved in immune response in humans, which are broadly divided into cells originated from lymphoid progenitor lineage cells as well as myeloid progenitor lineage cells. Can I have the next slide? So this is just to tell what is the immune response which is happening immediately after the antigenic exposure. Whenever an individual got infection, antigen first exposure, which is engulfed by the macrophages which are present in the tissues, right? And which becomes antigen presenting cells later. So the macrophages engulf the antigen or engulf the bacteria, which becomes the antigen presenting cell, present the antigen to the helper T cell and stimulates the cytotoxic T cells. Similarly, I'm getting some problem here. The icon whenever so the helper T cells stimulates not only the T cells but also it stimulates the B cells to produce antibodies, right? So it is so the whole thing explains how starting from the infection, B cell mediated antibody immunity as a humoral immunity as well as a cytotoxic T cell based cell mediated immunity are initiated and orchestrated due to the infection and various other cells which are involved in the immune response. Next slide, please. So now we are just moving into the other, slowly into the other area to understand a little bit about the immunogenetics basis as well as the HLA and infectious diseases. So it is a nature's mystery that some of the closely related genes are highly diverse, highly polymorphic. Say, for instance, if you take in chromosome number 16, the MHC, major histocompatibility complex genes, which encoded the class 1 and class 2 genes like HLA-A, B, C, DR, and DQ are all highly diverse. In HLA-A allele alone, A loci alone, there are 2,500 different alleles. B, around 3,000 different alleles. In that way, each and every different loci is having highly diverse number of alleles. And say, for instance, only 2 alleles from A loci are transferred to from the parents to the siblings or the SIBs or the children. So 2 allele from mother and 1 allele from father and 1 allele from mother. So 2 parental alleles are transferred to from each loci into the human into single child. So 2 from A loci, 2 alleles from B loci, 2 alleles from C, in that way. If you put permutation combination of any 2 allele from A loci and B loci, so this will come this much one combination out of this many. 71 lakhs 30,475 means that only 2 alleles out of 2,500 and 2 alleles out of 3,000. If you multiply these two numbers, you will get this number. So the permutation combination, the diversity of alleles and permutation combination of the 2 alleles going together by simple Mendelian inheritance into the children is highly diverse, highly enormous, highly unpredictable. And if you take another loci, A, B, C, the possibility of 2 alleles out of this many going into a single child will this many, out of this many. One set out of this many possibility. So imagine if you add A, B, C with the DR, and there are other loci, DP and DQ. So it is a highly complex system. So the diversity is because, so the important, which is a mystery, but answer is the diversity of the HLA alleles in humans is this much. It is because the pathogen is highly diverse in the nature. Pathogenic variations are highly diverse. Variations are enormous in nature. So we know pretty well that pathogens are highly mutating again and again, maybe due to various factors, also due to their drug variations. So the more we take, more we consume drugs, pathogens definitely mutate, right? So there are beautiful examples how the pathogen mutates and how it correlates with the HLA data. One or two slides are there in the downstream. Let us see in the course of time. Can I have the next slide? Can you please go back to the previous slide? I missed one point. Previous slide. Yeah, like the HLA alleles in the MXA, T cell receptors are also highly diverse, which is present in T cells. And VDJ is nothing but variable region, diversity region, and joining regions of antibody molecules, which is also highly diverse. And also KIR receptors, killer cell immunoglobulin-like receptors, which are present on the cell membrane of the nature killer cells, are also highly diverse. So all these molecules are very much involved in your immune response, which really tells you how important is the immune response with reference to protecting you from the infecting pathogens. Can I have the next slide? Next slide. Yeah, so this is a T helper cell and this is an antigen presenting cell. This is a membrane. So these are class 1 molecule, which is this is a class 2 molecule and this is a class 1 molecule. So this is a separate diagram and this is a separate diagram. So the class 1 molecule which is presenting the peptide and the T cell receptor is recognizing this peptide and sends signals into the cytoplasm. So T cell receptors are the molecules which are present on the T cells which recognize the foreign derived peptide presented by the HLA molecules or MHC molecules and accordingly sends a signal into the cytoplasm to the genome to produce respective cytokines and accelerate or induce the respective cells in order to protect from the invading pathogens. This is true with the cancer cells. Whenever a new cancer antigen is coming, the HLA molecules present the peptides or cancer peptides or cancer antigens to the T cell receptor. So the T cell receptors then send signals into the cytoplasm. They produce cytokines, mount anti-tumor immune response. Next slide, please. So this is a diagram showing the chromosome number 6, showing the MHC class 1 location. From telomeric region, it is a class 1 and class 2, class 3 and class 2 towards the centromere. And you can able to see variety of blocks genes in between the presence of heat shock proteins, properdin factor, drug metabolism genes like B450 and complement genes and leukotriene B, tumor necrosis factor. So the chromosome number 6 and MXA complex harbors very important gene complex other than the MXA class 1 and class 2, class 3 molecules. So the complement factors are also present and very important cytokines, tumor necrosis factor and heat shock proteins are also present in the chromosome number 6. So that shows the very important importance of this complex, and hence the chromosome number 6 and MXA complex is being called as a mini genome of humans. Can I have the next slide? So this is Professor Jean Darcy, who got Nobel Prize in the year 1980 for the discovery of HLA, and who discovered this HLA, role of HLA, which is very important for transplantation matching, accepting the donor's graft in kidney failed patients. He discovered these concepts during 1940s, late 1930s and early 1940s, but he got Nobel Prize after 40 years. So 36 to 41 is the period of, I think it is World War II. He worked as Army headquarters chief medical practitioner. So he started working on side by side the animal model experiments. What are all the genes which controls the transplanted organ? And because he was involved in the team who is having transplantation surgeries in wounded warriors, in the soldiers in the Second World War, and that really forced his teammates to find out the genetic basis of rejection of organs while they were doing these practices during the wartime. So he was awarded Nobel Prize in the year 1980, and we transferred the technology from Professor Jean Gosset from Paris to Madurai Kamaraj University in the year 1983, the HLA typing technology. And when I visited during 1996, for International Histocompatibility Conference at Paris. I met him at the age of 87 at that time, but he is no more. 10 years ago, he passed away. Can I have the next slide? So these are all the time-bound manner even one can appreciate. Starting from 1987 onwards, scientists got counting the number of HLA class 1 alleles and class 2 alleles. Class 1 comprise of HLA ABC alleles and class 2 comprise of HLA DR, DQ and DP alleles. So year after year, so in the international histocompatibility workshop, you can able to see the number of alleles keep on rising. As far as class 1 alleles, the number of alleles are much bigger. So shows that the more the viruses are mutating, more the newer HLA alleles are appearing in populations. So whenever they study new populations, whenever they discover new technology, whenever they use new approaches to study the HLA, they identify newer alleles, and hence the class 2 alleles and class 1 allele number in global populations are increasing. So why the alleles are increasing over the years? This is a very important question for the youngsters to understand. The answer is because of the newer pathogens which are coming into the picture, coming into the societal, exposed population exposed to the pathogens, as well as the newer alleles are being identified by newer technologies. And whenever new population and tribes living in jungles and forests and isolated areas are typed for their HLA, newer alleles are being identified. Can I have the next slide? So this slide shows the population specific variations of allele frequencies. Say for instance, if you take a class 1 HLA-A, A1 is one allele, A2 is another allele. So there are a variety of such alleles. In A1 allele alone, if you take and compare with the European Caucasians, Ashkenazis, Jews, Mexicans, Negroids, and Japanese, see the difference. In European Caucasians, 27.5, Jews it is 20, Mexicans it is 18, and Negroids it is 6, and Japanese it is 1. So you can able to see graded variations, gradients of allelic distribution. So it is a percentage distribution, right? So out of 100, 27 were in European Caucasians, 27 were HLA-A1. Whereas in Japanese population, it is only 1. So the differential distribution of HLA alleles in population tells you the important story behind how important this distribution of alleles in different populations, differential distribution of different HLA alleles in different populations. It is because of the pathogen prevalence in that environment really selects the alleles in different populations in different way. So likewise, if you take A2, you're able to see considerable quantity of this presence of these alleles, 45, 40, 44, 27 and 43%. So that means the A2 is highly important allele which is present throughout in all populations in a higher frequency. So this A2 is a very important allele and extensively studied HLA alleles. Similarly, different alleles are having different significances. Can I have the next slide? So coming to the molecular mechanism, how the viruses coming into your cells and how the antigens are being processed. When the HIV virus or any virus for that matter attaches with the receptor, so the receptor really attracts the virus to bind with it. Engulfed, the virus is being engulfed and internalized, and its nuclear material is transferred into the nucleoplasm. If it is an RNA, it is converted into DNA by using reverse transcriptase enzyme. The molecule which is also present here, which converts RNA into DNA. Then the viral DNA, after conversion, get integrated into the viral, host DNA, eukaryotic DNA, and viral DNA is converted into viral mRNA, that is by transcription machinery, move into the cytoplasm where the mRNA is translated into viral proteins. Viral proteins are then sent to a structure in cytoplasm called proteasome, where the bigger proteins are choked and cut into pieces called peptides. Then peptides sent to the endoplasmic reticulum, where your HLA molecules are standing as vehicles or trucks waiting to be load the cargo. So they pick up the peptides derived from the pathogens and move to Golgi, fuse with the Golgi membrane, and fuse with the membrane, present cell membrane, present the viral derived peptide outside by the HLA class 2 molecules. So class 1 or class 2 molecules. So this is a mechanism by which the viral proteins are coming into the cytoplasm and being presented outside by the HLA molecules for the T cell to study about the peptide and produce cytokines to orchestrate the immune response. Can I have the next slide? So this is a molecular structure. I don't want to explain this in detail because all the students they might have studied. This is a class 1 molecule, which is a single chain molecule, which is attached with the beta to microglobulin, whereas the class 2 molecule is two chain made of alpha 1, alpha 2 domain alpha chain and beta 1, beta 2 containing beta chain. Can I have the next slide? So this slide shows exactly how the MXC molecule one side. So this is a MXC molecule and the T cell receptor on the other side. So in between, the trapped is the influenza peptide. So they crystallized, they eluted the peptide from the cell membrane of the infected animals, infected animal cells in the cell culture environment, and eluted this MXC antigen T cell receptor complex and crystallized and crystallographic structures. From the crystallographic structures, they elucidated that MXC presented the antigen and T cell receptor recognized the antigen by using its T cell receptors. So this is called MXC restriction phenomena. So the class 1 restriction, class 2 restriction, two important restriction phenomena are being decoded by the scientists long back, some 3 decades ago. And this is called as a ternary complex. This is very important stage immediately after which the cytokine production accelerates the immune response in the humans as well as any living organism. Can I have the next slide? So this is just a molecular structure of the class 1 molecule, which is having 2 alpha helices on both the sides and beta sheets at the bottom. So there is a groove, a cavity-like structure. Only in this cavity-like structure, the pathogen-derived peptides comes and binds. So the critical amino acids in these locations, maybe here or here or here, what type of amino acids are present, whether it is a acidic amino acid, basic amino acid, aromatic amino acid, aliphatic amino acid, type of amino acids and other clusters of amino acids really study the peptides which are coming and binding into this peptide binding groove. So after studying the peptide, only it sends a signal into the cytoplasm. The T cell receptor and T cell receptor accordingly send the signal. Can I have the next slide? So this is a same diagram in a crystallographic this thing. Next slide, please. So coming to the other aspect of my talk, the host genes and course of infectious diseases. If you take the time and the number of genes involved and their type of role in immune response. Say for instance, if at the starting of infection, one particular gene called A, this is just for explanation, just given A, B, C, D, right? Genes name may be different. If you take one particular gene called A, starting from the exposure at day 1 to till the disease is progressing towards the end stage, the gene is getting activated. Okay. Whereas the other genes are exposed, are produced, the product of the gene is produced, are subclinical infection, right? Mild and moderate infection, you can able to see only 1 or 2 genes which are expressing. Whereas in severe stages, only 1 or 2 genes are expressing, right? So the diagram really illustrates the differential expression of different genes during different time course of infection. So different genes act at a different time. So only if all the required genes are produced in humans in an effective manner, orchestrated manner, only then the infection being controlled and the disease being eliminated. Can I have the next one? Next slide. So coming to closer look at how the different diseases, infectious diseases and prevalent diseases are present and their mechanisms and their HLA association. Say for instance, if you take malaria, it is a tropical disease present in all the region which falls close to the Tropic of Cancer, right? So Africa, northern part of this South Americas, and almost all the part of South Asia, right? Starting from Iran, India, and Burma, Indonesia, into the Austronesian islands, right? So this is a very important disease which is being spread by the vector mosquitoes and caused by plasmodium, as we have seen in some of the previous slide. Can I have the next slide? Next slide, please. So the genes involved in malaria includes maybe for severity as well as mild form of disease. Reduced severity of disease is being orchestrated or modulated by hemoglobin type, RBC proteins like HPS, HPC, alpha thalassemia, beta thalassemia, Duffy genotype, G6PD, erythrocyte band 3. So all these proteins of host orchestrate or modulate or decide the severity of the disease. And also the HLA alleles are also being studied by scientists. Like they identified one allele, HLA-B53. The individual with B53 in Africa are highly protective and towards the malaria in Africa. Likewise, the HLA-DR2 also offers protection in African context. And there are other genes, INOS 954 promoter C allele. Increased severity of the disease is associated with the TNF alpha 308 promoter alpha homozygosity as well as ICOM. One coding region T allele presence increase the severity of the disease in malarial parasite infected individuals. Can I have the next slide? So this is Professor Jean Dawsey, Adrian Hill, who was a senior professor at the time when we collaborated with him. And incidentally, he was my external thesis evaluator during early 90s. And we collaborated for 6 years, and we came out with a Nature's paper in a Nature Genetics journal, in which we demonstrated there is a one-to-one strong association between the genes in the chromosome number 10, and since this is not HLA but microsatellite marker genes with the leprosy in India. So we collaborated with him. We collected DNA samples from 250 families of leprosy families and studied the microsatellite mapping, genome-wide mapping with the help of Oxford University Professor Adrian Hill. Can I have the next slide? Actually, the Adrian Hill is a professor, scientist behind the AstraZeneca vaccine, which is being extensively used for COVID-19. And he published a paper in the year 1991. Only based on this paper, we collaborated with him. We contacted him for my thesis evaluation. He accepted, and his collaboration went very well. And still, my Professor Pichappan is still having contact with the Professor Adrian Hill. And he studied extensively and identified that HLA-B53 individuals are producing high protection, affording high protection against the malaria in Africa, in particularly in Gambia. That is true with most of the African populations in most of the African countries. Can I have the next slide? So, by molecular dissection and methodologies approaches, Adrian Hill et al. Identified specific epitopes from the malarial parasite Plasmodium falciparum derived epitopes, and they used the CTL epitopes. Epitopes is nothing but 10 amino acid length small protein segment, and which is isolated from the cytotoxic T lymphocyte peptide binding groove of the HLA molecule. They sequenced the peptide and they used the Plasmodium falciparum derived liver stage specific antigen LSA1 they called as a peptide candidate peptide to produce the vaccine against malaria. And of course, they launched this vaccine last year, and it is in the market for introduced into the malaria endemic regions. Can I have next slide? So that's the same thing. The other group studied sporozoite derived epitopes, CTL epitopes of liver stage antigen 1 and circumsporozoite protein P, and they gone in for vaccine production based on HLA derived peptide elution and then vaccine. This is a process called reverse immunogenetics. Reverse immunogenetics is a process by which they are approaching the vaccines, developing vaccines from the peptide eluted from the HLA bound peptide, and then sequence the peptide, and then based on that, the synthesized peptide, use it for vaccine production. Can I have the next slide? So there is another important area to cover, TB and HIV, which is called as a double trouble. And people with HIV infection face a greater risk of also developing TB. And can I have the next slide? Next slide, please. and Professor Luc Montagna received a Nobel Prize for Physiology in the year 2008 for his discovery that HIV-1 virus is the causative agent for previously unnamed disease, that is AIDS, acquired immunodeficiency syndrome. Only after they elucidate the role of HIV-1 in the AIDS, the AIDS is being named as AIDS. So can you have the next slide? So this is just epidemiology map of India is having moderate epidemiology, right? So the Africa, the more the color intensity, red color, and the prevalence is more. In Africa, it is 4.4, whereas our Indian color, it is Southeast Asia, it is zero point 0.3 only, right? Zero point 0.3 to 0.4, whereas in Africa, it is 4 to 4.5. So it is very high. It is moderate, whereas the yellow color shows the HIV moderate prevalences. Here it is moderate, here it is very low. Can I have the next slide? So in Indian states, which is really our curiosity, which are all the states which are having more HIV patients, though it is data is a very old data, some 20 years old data. Interestingly, but the data is not much difference, much differed with the present day data. So the NACO, National AIDS Control Organization data of 2004 revealed that Maharashtra is a top leading state having more HIV patients, followed by Tamil Nadu and then Karnataka and Andhra Pradesh. And there is one more northeast state also having high prevalence rate. Can I have the next slide? And that is data in a different form, which is acquired in 2004 by NACO. Tamil Nadu, different time point. Tamil Nadu was topped with 40,000 patients HIV positive cases, Maharashtra with 12,000. Even today, the race is between the Tamil Nadu and Maharashtra in having the highest number of HIV individuals in any given state in India. Can I have the next slide? So we need to understand the why this much infection is there in South Indian states, southern states, but not in north states. Lot of data, lot of points to be discussed, but this is not the forum to discuss all those points. But very importantly, there are environmental factors, socio-biological, behavioral factors, and also genetic factors, which determines these drastic regional differences of north and south divide of genetic basis of infection in HIV. So which is one of the deadliest pandemics in the human history, and extremely high rate of mutation. The high rate of mutation rate only determines the HIV disease, high rate of viral diseases in populations. Millions of distinct variants are generated each day. Imagine the virus mutates every day, and if the pharmaceutical industry come with a drug working for 10 years, and the virus escapes the drug within a day, right? Within a day or a few days. So in a single patient, which is called as quasi-species. So the virus with a new mutation are called as quasi-species, which is highly individual specific, that adapts and destroys the immune system. This is called as escape mutants. Can I have the next slide? So they studied in two individuals. One is the donor of the virus and another is the recipient of the virus. So over a period of time, they studied what are all the different clades of viruses present in the two individuals, donor and recipient. In during the 8 month of exposure or 8 month, eighth month of infection identification of the HIV positivity. So this is a clade or gene sequence of the virus. After that, 20 months they collected the blood and studied the sequence. 42 months. So in donor, three different time duration testing and sequencing of viruses gives three types of viral clades. Similarly, in recipients, 20 months, 42 months, and 52 months, the DNA sequencing of virus revealed three different clades. So here, the donor is the husband and recipient is the wife. He is the one who contracted the virus from because of his behavioral this thing, and he donated the virus to the recipient. And you can able to demonstrate here the virus, though it is being given by the husband to the wife, even in the wife itself or the recipient itself, over the period of time, again and again the virus mutates and newer clades are being identified within the patient. So when a pregnant woman gone for a normal routine clinical checkup, she was identified positive for HIV, and then they started testing the husband. He also turned to be positive, and this study was conducted, and beautifully they demonstrated that virus really makes a mutation again and again within a very short span of time, and in a single individual itself you can able to find out quasi species circulating at a given point of time. Can I have the next slide? Of course, I'm skipping, because these are all some of the basic slides. The HIV virus discovery was related to Haiti, which is a small island close to America. Those who during 60s and 50s, people mysteriously died of some unknown disease, and later it was identified the causative organism is HIV virus, and many of those professionals or business people who visited Haiti got that mysterious disease, and they contracted the disease when they visited the brothel houses. So it is a disease of social behavior as well as very unique biological characteristic features. Can I have the next slide? So there are other genes which are associated with HIV with resistance, HLA CCR5, and disease progression is associated with the type of HLA, CCR type, SDF, MBP proteins, vitamin DR, vitamin D receptors, IL-10, interleukin 10, and tumor necrosis factors. And also it was shown in HIV case that based on the type of HLA of the individuals, the vaccine against HIV will be much efficient in individuals with specific HLA. So these are all some unique points when we plan for a vaccine development as well as vaccine administration in a community at large. Can I have next slide? So of course I'm skipping a couple of slides. Next slide. So this is a very classical paper, Nature, published in Nature by Capella et al. 2004, by which they demonstrated that individuals susceptible with HIV are predominantly having alleles called HLA B58 and B18, whereas protective individuals supposed to be protective are having predominantly B57 and 5801. Here it is 5802 allele, here it is 5801 allele. Can I have the next slide? But sequencing studies, they identified that HIV proteins called NEF protein and GAG protein, or when they sequence, the variations in HIV NEF and GAG variations are similar to the HLA B alleles, or the means that when they analyze the data based on the number of mutations accumulated in HLA B restricted allele manner as well as HLA A allele restricted manner, more HLA viral polymorphism were encountered with the HLA when they analyzed the data based on HLA B alleles, right? So there is a one-to-one correlation between the more diverse nature of HLA B alleles and more diversity of the HLA NEF and GAG proteins of HIV. And similar to the correlation that the more the police developing technologies, thief to develop technologies. So similarly, it is being demonstrated in biology as well as by using molecular tools that viruses are evolving fast in order to cope up with the drugs as well as the other immunological threat goes inside exposed into the human host. Can I have the next slide? So these are all the alleles which are associated, HLA alleles associated with the slow progression of HIV disease, and these are all the alleles associated with the fast progression of HIV alleles. So by studying a very large cohort across continents, they come to a conclusion that there are alleles which are favoring slow as well as fast progression with reference to HIV. Can I have the next slide? Maybe another 10 slides may be there. I will finish off in 5 to 10 minutes. So I'm skipping a couple of slides. Next slide. Of course, we discussed a lot of many other points. The only important point I want to discuss is for vaccine response with reference to HIV infection, enhanced immune response or enhanced vaccine response is observed with reference to individuals who are having HLA B57 27 allele with reference to canary box HIV vaccine. And for drug reactivity, this is well established fact that people are HIV positive individuals who are given abacavir, which is one of the RT reverse transcriptase inhibitor drug, abacavir, and individuals who are having HLA B57 develop allergy and lead to hypersensitivity reaction, and they ultimately die of the abacavir induced hypersensitivity reaction. So beautifully, the HLA B57 individuals respond heavily to the abacavir drug and develop more hypersensitivity, more immune response, lead to cytokine storm, as we observed in COVID-19 patients who died during a huge number 2020-21. Similarly, it happened here, and hence last 5-10 years, Apakavir was withdrawn from the market in US and other developed countries. Can I have the next slide? I'm skipping a couple of slides. Skip the slide. Next slide. So this is TB incidence global data. Of course, South African countries are having a very huge prevalence rate. India is in median stage, but of course, India is one of the leading country having huge number of TB patients. Can I have the next slide? These are all the genes associated with the TB infection and immune response genes, like HLA-DR and DQ genes are shown to be associated strongly with the HIV infection. Interleukin 1 R receptor, interleukin 1 receptor A, interleukin 1 alpha receptor alpha, and myelin binding leptin MBL proteins are also shown to be associated with the disease. Other genes which are associated with the TB includes vitamin D receptor, which is present in 12q chromosome 12, and NRAMP1 gene, which is present in chromosome 2, and X chromosome, some genes are present in X chromosome, and locus present in chromosome 15 are shown to be associated with the TB. Next slide, please. And these are all the interesting studies showing the genetic link between the Mycobacterium tuberculosis and the human genes, different genes. I'm positive time, I'm not explaining all this in detail. Can I have the next slide? So this is a paper we published in 1991 from our lab, Association of Pulmonary Tuberculosis and HLA in South India, in which we demonstrated that HLA-DR2, one important allele, which is serological allele, later it is divided into HLA-DR15 and DR60. DR15 further subtyped into DR1501 and 1502, which are showing high positivity rate among the tuberculosis patients. Means that those individuals who are having DR2 or its subtypes like DR1501 or 1502 are prone for developing the tuberculosis when they get exposed to the Mycobacterium tuberculosis. Can I have the next slide? So coming to the leprosy story, I'm not explaining too much about it. So India is a hotbed of leprosy for hundreds of years, like some pockets of Africa and some pockets of South America. Can I have the next slide? Next slide, please. We identified and published in Nature Genetics. This is what I told you, Adrian Hill, Professor Adrian Hill collaboration. We published this paper in Nature Genetics, a major susceptibility locus for leprosy in India maps to chromosome number 10. And a role for the genetics of the host is variable susceptibility to leprosy has been identified by family clustering, twin studies, complex segregation analysis, and human leukocyte antigen association studies. So it has a lot of genes associated with infectious diseases. HLA is associated with several diseases, and we identified in this study a genetic loci, which is a microstructure marker in chromosome number 10p13 position, which is highly strongly associated, but this is non-HLA linked loci. So other than HLA, also genes also associated with infectious diseases. Can I have the next slide? So these are all the other studies, international studies, in which they demonstrated genes HLA-DR13 is associated with the HPV clearance. Individuals with HLA-DR13 clear the virus very quickly, whereas HPV persistence is noted with the DR beta 11 and 12 individuals. DQ beta 301 is also induced the HPV persistence. And HCV susceptibility is associated with the DR3 and 7 alleles. Can I have the next slide? So this is a slide showing the classical definition of supertypic alleles, which was published in Journal of Immunology long back, 1999, by Chang et al., in which HLA A3, A11, A31, A33, and 68 alleles are called as A3 supertypic alleles, and the alleles B7, 35, 53, 54, 51 are called as B7 supertypic alleles. So individuals who are having any one of these A allele or any one of these B allele are together. One A locus, this any one of these allele, and B locus, any one of these allele, are more protected because of the quick viral clearance from the blood. So these alleles are called as HLA supertypic alleles, very important for human protection and survival. Can I have the next slide? So these are all some of the slides showing that HLA alleles are associated with infections. HLA-B35 shown to be with chlamydia infection leading to coronary artery disease. Can I have the next slide? Not only HLA-A35 allele, A3, A35 two locus haplotype and B35DR1 haplotypes revealed a strong association in chlamydia pneumonia infection mediated coronary artery disease. Similarly, HLA-B51 revealed a strong association with respiratory infection mediated stroke. Next slide, please. Yeah, an association of herpes simplex virus with the type 1 diabetes and type 2 diabetes is also reported. Next slide. And it has been demonstrated beautifully by a group of scientists long back, but of course the same Potts et al. Last 10-15 years also did some work, and they demonstrated that in animal model the MXC heterozygosity presents the more diversity in the populations, not only in humans but also in animal this thing. The mating pattern is determined by the HLA alleles, right? So say for instance, they studied in extraterritorial sites, and the animals with heterozygous HLA type moves far away from their native point, and they mate with the individuals which are having different HLA types, right? So this work was done in animal model, but it exactly replicates what is happening in human system. So the mating is also influenced by, to some extent, of course, it is influenced in humans in a different way, but in animal model, they demonstrated the MXC drives the mating behavior in animals in order to make the population with more heterozygous individuals, in order to protect the population from the innumerable number of pathogens. Can I have the next slide? So this is a slide, maybe the last slide, I hope, or one or two slides prior to the last one. Polymorphism in MX genes of the cheetah is very limited, presumably because of a bottleneck in breeding that occurred in the not too distant past. It is assumed that all cheetahs alive today are descendants of a very small breeding pool. It is because of the humans more population of human population, I mean, becoming bigger and bigger. We are making the forest for utilization, more areas of forest for utilization, and hence we are reducing the available space for the animals, so they become highly homogeneous. The gene pool becomes more homogeneous. Heterozygosity got dwindled. That is why some of the cheetahs which are being translocated from Africa to India very recently, they are one after another getting sickness in Indian environment, because the cheetah population present globally are not genetically diverse. MXC based diversity is not there in animal model because of thousands of years we humans destroyed their environment and destroyed their diversity. Can I have the next slide? So this is another, it's a very big story, but I don't want to go into the story. We acquired one gene from viruses, right? Viruses are having a gene called syncytin. Somehow during the infection of some of the viruses into the human ancestors, human vertebrate ancestors, the virus gene syncytin is transferred into the human genome. Like initially I said, many genes, there are receptors. Similarly, we acquired many genes from the viruses, right? During the course of infection. So one gene is syncytin. So the early viral infection contributes the syncytin gene to the human ancestors, and hence the syncytin is a gene which makes the cells to fuse together. So the fetus in the developing womb of mother got attached with the mother's endometrial wall. It is because of the genes that is acquired from the virus called syncytin. So without the syncytin, from the virus, we humans may not develop as humans, right? We may be another vertebrate, like egg-laying vertebrate or some other vertebrate type, right? So marsupials, something like that. So we acquired syncytin. So our ancestors developed the intrauterine development of the fetus because of the viral donation of the gene. So some good things are also happened, but of course most of the viruses are really friends to humans, really friends to the many of the living organisms. The more we make the wild animals, I mean, more give more problem to the wild animals. The viruses jump into humans and they do unexpected harm like COVID-19. Can I have the next slide? So the genes and drugs really matters when the genes are really matters when you consider to develop a drug, and we all need to take into the racial factors or genetic factors, particularly in different populations, before finalizing any drugs. Of course, drugs are being now designed based on the genetic type of the individuals. Say, this has been true for many of the warfarin like drugs, in which some warfarin type of drugs or quantity, some population they need a little drug dose, whereas in some population the warfarin dose must be little more. That slide I eliminated, which has not been given, which has been beautifully illustrated in global populations. Can I have the next slide? uh, Probably the last slide. So maybe I may need your attention for 30 seconds. In USA, see the population 33 crores. In India, what is the population? 140 crores. How many times? Maybe 4 times or 5 times. Right? Now you just look at the total cases of HIV. It is like 10 crores or 10 lakhs. Yeah, 10 crores. Out of 33 crores of population in US, 10 crores got the infection. How many death? Close to 12 lakh death encountered. It is a very recent data we got collected from the global epidemic COVID-19 database. 12 lakhs death documented. Come to India. It is 140 crore population country, whereas we are having cases of total cases of 4 crores, 4.5 crores, and death will be close to 6 lakhs only. So, a very big country, infection rate is very less, half of US, half of US, whereas death rate is also half of the US. What is the reason? So, though technologically developed country US, having much smaller population than India, still having much higher case rates as well as death rate, right? Some intriguing facts are there, and not only the environment, not only the genetics, not only the behavioral, not only the lifestyle factors. Many things really protected Indian population from the upcoming epidemics and endemics. With this, I'm stopping here. Can I have the next slide to check if anything is there? Yes, we are all humans, we are all relatives, we are all one. With reference to genome, 99% of our genomes are similar, except the HLA, like some diverse genes are, we are varying in only that aspect, which is in order to handle different pathogens differently in different environment. Can I have the next slide? So thank you very much. Thank you one and all for your patient listening at this very odd time. Thank you very much. I thank the organizers, all the management staff, all the faculty members, all the students, participants, online, offline. Thank you very much. Thank you one and all. Couple of questions I'm really, if interested, if some of you are really interested, you can ask, or else you can mail me. I will answer your queries.
Thank you, sir, for your nice presentation. Audience, you may raise your questions.
If it is not
Thank you, sir, for your nice presentation.
okay, okay.
Thank you. Dear participants, if you have any queries or questions, you can ask. Okay, thank you, sir.
Thank you for patient.
Okay, thank you, sir.
As a secretary of Indian Immunology Society, I invite those interested faculty members and research scholars to join Indian Immunology Society in a big way to make strength, give strength to the society. It is a 50-year-old society, and we are celebrating this year 50th year of inception of Indian Immunology Society, right? So that is what we granted 50 different CMU programs and conferences this year. So you are one of the luckiest center to organize this event. Thank you very much. Thank you one and all for your support and patient listening. Thank you. Thank you. Thank you. I'm just leaving. Thank you.
Okay, thank you, sir.
சரி சார். அவங்க அந்த இது 40 கேமராக்கு ஆர்டர் ப்ளேஸ் ஆகிருக்குன்னு மறுபடியும் சொல்கிறாங்க சார். ஆர்டர் ப்ளேஸ் ஆகிருக்காங்க சார். அவங்க 40 கேமரானா அப்புறம் மறுபடியும் வேறு பிளான்லேயே அனுப்புகிறாங்க அப்படின்னு சொல்லியிருக்காங்க. நம்ம மெடிக்கல் கேம்ப் பண்ணணும் சார். எல்லா பேருக்கும் சேர்த்து பழசு அந்த ரூல்ஸ் ஒரு புதுசு ஒரு 40 கேமரா இருக்குது சார். அவங்க ஓனர் சொன்னாங்க சார். ஆர்டர் ப்ளேஸ் ஆகிருக்கு. சார் சார் ஓ சரி சார். நான் அந்த என்ன சரி சார் லைப்ரரி மெட்டீரியல் வாங்க இருக்காரு. சரி சார். ஓகே சார். நான் வரேன். சார் பாடி சாரை கூப்பிடுறீங்களா? சொல்லுங்கள் சார். என்ன சொல்லுங்கள்.
Hello, can anyone hear me?
Yes, sir.
Okay. Which one is which one their serial PPT? Yes.
Hello. Hello.
Can anyone tell me who is the first presenter?
Hello, hello.
Yes.
This is Rashmi Singh. I
Yes,
would
ma'am.
like to present my PPT.
Okay. See-
I just want to ask how to share my PPT.
Uh, in your options have a arrow like arrow option is here.
Yes,
So
yes.
click yes.
Yeah.
So yes. So you can click on this, and after that you share.
Okay, okay.
Hello.
Yes.
Sir, I want to present my slides.
Okay. Okay.
Yes.
First of all, who is the? Yes, Rasmi Singh. Can you share your slide or not?
Sir, I'm trying, but I have to click on your entire screen or a window.
In your window, you minimize your screen, and after that, you select your presentation and select that. Okay.
Sir,
Yes.
is it
Yes.
showing here?
Yes. Yes. Okay.
Okay, sir. So when we have to start?
Okay. Start the
Can we
presentation.
start now, sir?
Uh, First of all, you all
Or we have to
participants are
wait for some
here.
time?
Hello. Hello. Good afternoon, sir. Hello. Good morning, sir. Sir, is there any list for the participants? Means when I am going to present my paper.
I don't know because I am also a student. So please don't.
So is there anyone who is from the organizing team?
I don't know who one is here or not.
Good afternoon, organizers. I request at what time the presentation will be started. So, can you please tell me, please?
May I know also same doubt because 30 minutes.
Yes, sir. That's true, sir.
Hello? Hello? Can anyone please contact?
Sir.
Can anyone please contact the organizer?
So
Yes,
I
sir.
will
Yes,
make
sir.
a
Yes,
contact.
sir. I
I
will
will make a
contact him.
contact.
Yeah. Anyone please contact because it is already half an hour when we are going to start the presentation. And the valedictory is already at 4 o'clock. So it is going to be very difficult to continue with the presentation.
Hey, போடா, போடா, போடா. அப்பாட்ட சொல்லு, அப்பாட்ட சொல்லு.
Participants, kindly be in line. The session will be starting soon. Participants, there is a technical issue. Kindly be in line.
சிஸ்டம் ரீஸ்டார்ட் ஆகிடுச்சு நீங்கள் இன்னொரு சிஸ்டம் நான் கொடுத்து வந்தேன் சீட்டில் அது சொன்னாங்க நீங்கள் வந்து மேலே இந்த ஆஃபீஸில் ஒரு சிஸ்டம் கீழே வேணும்னு சொல்லிட்டு அது நாங்கள் எங்கள் பிரிண்டர் ஒர்க் ஆகலை நெட்டு வரல சார் ஒரு லிங்க் ஒன்று அனுப்பிச்சிருக்கீங்களா சார் அது ஓப்பன் சரி சார் சரி சார் இல்லை சார் இப்போ சிஸ்டத்தில் ஓப்பன் பண்ணணுமா சார் சரி சார் சரி சார் சரி சார் சரி ஓகே சார் சரி சார் ஆமாம் சார் சரி ஓகே சார் சரி ஓகே சார் லிங்க் ஒன்று அனுப்பிவிட்டாங்க அதை ஓப்பன் பண்ணி கூகுள் மீட்டில் ஓப்பன் ஆகலாம் பாருங்க அப்படின்னு ஓப்பன் பண்ணி இந்த இது நமக்கு வருதா என்னன்னு பார்க்க சொல்கிறாங்க இந்த லிங்க்கு ஓகே வருது எங்கே வருது உன் பேர் வேணா ஐயோ கேன்சல் பண்ணுவேன் பைத்தியம் லெஃப்ட் ஆனது அவங்க அதில் ஒன்று ஏன் பைத்தியம் ஏன் அப்படி பண்ணேன் அதில் எண்ணெய் மட்டும் எடுத்து விடு இது என்னது எண்ணெய் மட்டும் எடுத்து விடுங்க இன்னும் ஒன்று காணுமே கீழே வா கீழே வா கீழே வா கீழே வா கீழே வா கீழே வா ஃபஸ்ட்டு வா நான் சர்ச் பண்ணி பாரு யுவராஜ் இருக்குது ஆடெல்லாம் இல்லை ஓகே இல்லை நினைக்கிறேன் உங்களுக்கு ஜாயின் பண்ணாதான் நான் இது யார் மேல் என் மேலியா ஐயா எங்கே இருக்குது சாரோட ஆன் பண்ண மைக் ஆன் பண்ண ஆன் பண்ண அது பண்ணல அது பண்ணல சாமி ப்ரெசன்டேஷன் பண்ண க்ளோஸ் பண்ண
Committee? Anyone from the organizing committee?
Sir, I'm in line, sir.
Ma'am, can I ask you two, three questions?
Sir, there is a technical issue in streaming in your.
If the session is recorded, can we start our presentation so that it is going to be recorded? If the
Let me ask my.
Okay, you can ask that the session is recorded. If the session is recorded, so then we are going to start the presentations.
Yeah. I could understand, sir. Please be in line, sir. I'll be
And ma'am,
asking you.
second, one more thing, ma'am, we are not having any means list of the participants who is going to present first and second and third and fourth. So, is there any list, or can it is going to be a random that who is going to present?
Sure, sir, but it has to be streamed, no, in YouTube. That's why we are waiting for it, sir. I'll talk to
Okay.
my higher official and talk to you, sir,
Then
soon.
please make sure that it is going to be. Okay,
Please
thank
be
you.
in line.
ப்ரெசென்ட் பண்ண சொல்லிடுவோம் இதை
இது இல்லை நீங்கள் அப்படியே
இது இப்போ ஓரல் ஒன்று ஃப்ரீயாக இருக்குண்ணா சார் அது ஓடி இருக்கும். இது வந்து நம்ம இது பண்ணிகிட்டே இருப்போம்.
சரி
இப்போ நம்ம ஃபர்ஸ்ட் லிங்க் இருக்குன்னா சார்
ஃபர்ஸ்ட் லிங்க்
அதில் வந்து கனெக்ட் பண்ண சொல்லிடலாம்.
வேறு
அது கொரியாவா?
சரி
இதை வந்து அதுக்கு அடுத்தது பண்ணிக்கலாம்.
சரி
இல்லைன்னா இது கூட பார்த்து
அப்போ நம்ம இந்த லிங்க்லேருந்து வெளில வரணும்.
இதுலேருந்து வெளில
ஆமாம்
அது வந்து அவங்க வரல உள்ளே இருக்கட்டும் சார்.
சரி நம்ம வெளில வந்துடுவோம். ஏதாச்சும் ஒரு நியூஸ் கொடுத்துட்டு வெளில
வரும் சார். அதில் போட்டுருவோம் சார். நவ் டெக்ஸ்ட் ஸ்டார்ட் ஆகும். நவ் the oral presentation is going on. I'm going on. I'm full stop with after oral presentation. Oral presentation. We will inform you all about the poster presentation schedule. Yes. Okay sir, send me. Okay sir, இதை போட்டு விட்ருவோம் சார்.
சரி சார், இதுலேருந்து நம்ம எல்லாம்
நம்ம வெளில வந்துடுவோம் சார்.
மார்னிங் போனால் லிங்க்கில்
அதிலே போயிடுவோம் சார். இப்போ நம்ம அங்கே ஒன்று வாங்கி வச்சு இது அக்ரியில் ஒன்று வச்சுருந்தோம்னா சார்.
ஆமாம் சார்.
ஹலோ அதை மாற்றிட்டேன். இப்போ இது வந்துடும். நான் இங்கே தான் இருக்கேன். இப்போ கொரியா வந்துடும். ஓகே.
இது மார்னிங் கொடுத்த லிங்க் சார். இப்போ இது யூடியூப்பில் போகணும்.
இது யூடியூப் சார்.
யூடியூப்பில் போகணும். சேவ்லாக் செமினார் ஹாலில் இதை காமிக்கணும்.
ஓகே
காமிக்கலாம். யுவராஜுக்கு ஃபோன் பண்ணி
இப்போ இவர் அட்மிட் ஆக சொல்லிடுவோம் சார். புரியாது. ஹலோ இல்லை இருக்காங்க எல்லாம் இருக்காங்க. நீங்கள் இது பண்ணிடுங்க. நான் கனெக்ட் பண்ணிடுவேன்.
இப்போ இருக்க கூகுள் மீட்லேருந்து டிஸ்கனெக்ட் பண்ணிக்கோங்க. மார்னிங் ஒரு கூகுள் மீட் பண்ணியிருந்தாங்க இல்லையா, அந்த கூகுள் மீட்டில் உள்ள வந்துக்கோங்க. அதை வந்து ப்ரொஜெக்டர் ஸ்க்ரீனில் டிஸ்பிளே கொடுத்துருங்க. அதாவது நேற்று நைட்டு ஒரு கூகுள் மீட் ஒன்று போட்டிருக்கேன். அந்த கூகுள் மீட்டில் உள்ள வந்துக்கோங்க. உள்ளே வந்துட்டு அது ஸ்க்ரீனில் டிஸ்பிளே ஆகிட்டு இருக்கணும். கூகுள் மீட்டில் என்ன நடக்குது, ஆடியோ வீடியோ எல்லாம் அதில் வந்து ஓகே. வச்சு கரெக்டு கரெக்டு ஆ கேமரா ஆடியன்ஸ் என்ன திருப்பிங்க என்ன திருப்பிங்க அதான் இப்போ என்ன எம்பிஏ மட்டும் தானே பெண்டிங் எம்பிஏ அக்ரி மட்டும் தான் அது எப்போ நல்லா பார்த்துக்கிட்டீங்களா சரி நாளைக்கு வர சொல்லியிருக்கேன் நாளைக்கு 3 நாள் அடிச்சாங்கன்னா அக்ரியில் வந்து எம்பிஏல 2 அடிக்கணும் எம்பிஏல எந்த ஹால் அடிக்கிறதுனா தெரியல மேலே எம்பிஏ கன்ஃபார்ம் தான் கன்ஃபார்ம் பண்ணி சொல்லிட்டாங்க எந்த ஹால் அடிக்கிறதுனா தெரியல ஒரு இடத்துல பார்ட்டிஷன் இருக்குது ஈவினிங் போவோம் ஈவினிங் போய்ட்டு என்ன பார்ப்போம் ஒர்க்கு எதுவும் உங்களுக்கு இருக்கா நீங்கள் கோஆர்டினேட் பண்ணிக்கிறீங்களா சாரை பார்த்துங்க நான் ஒரு ½ hour போய்ட்டு வந்துடுறேன்
ஒன்றும் இல்லைம்மா அப்படி வர்றது வந்து நீங்கள் அப்போல்லாம் கேமரா அந்த ஸ்டேஜ் இது காட்ட சொல்லிட்டு செஷன் வந்து ஸ்டார்ட் பண்ணிட்டு நான் உங்களை அட்மிட் ஆக சொன்னேன் இன்னும் வரவே இல்லை உள்ள நீங்கள் அதே டாக் கொடுக்குறது தான் இப்போ போஸ்டர் இல்லை டாக் கொடுக்குறது தான் அதாம்மா வரட்டும் நீங்கள் ஃபர்ஸ்ட் அட்மிட் ஆயிருங்க அவங்க வரும்போது நீங்கள் இது பண்ண முடியாதுல்ல நீங்கள் அட்மிட் ஆயிருங்க இல்லைம்மா நான் சொல்கிறது மைக்கை நீங்கள் ஒரு இதில் மியூட் போட்டுட்டு வீடியோவும் இது நீங்கள் ஃபஸ்ட்டு அட்மிட் ஆயிருங்க உள்ளே வந்து அந்த லிங்க் வழியா அட்மிட் ஆயிருங்க அப்புறம் அவர் கொரியர் பர்சன் வந்துச்சோடனே நாங்கள் இப்போ அது பண்ணிக்கிறோம் ஏன்னா நான் இங்கே இருக்கும்போது நீங்கள் அங்கே இன்ட்ரோ கொடுக்க முடியாது நீங்கள் ஒரு ஆள் இருக்கணும் அதுக்குள்ளே ஓகே இங்கே வரலையே நீங்கள் நான் கொடுத்த லிங்கில் வரலையா ஆமாம் அதில் தான் இருக்கேன் நீங்கள் வரவே இல்லையா இங்கே இருந்த கேட்குமே இந்த ஒரு பொண்ணு கூட வந்து வந்தாச்சு ரிக்வெஸ்ட்டு வரல இங்கே வந்தால் நான் கொடுத்துருவேன் நான் அங்கே தான் இருக்கேன் வரலையே ஆமாம் ஆமாம்மா ஆமாம் ஓகே சார் ஓகே ஓகே அனுப்பிட்டேன் சார் அனுப்பிட்டேன் கொஞ்சம் வாங்க ஏன்னா அங்கே போட்டு நம்மளை போஸ்டர் ப்ரெசன்டேஷன் கொள்றாங்க இதில் வீடியோ இருக்கா சார் வராது இதில் வீடியோ இருக்கு ஓகே இப்போ அங்கே ஓடுமா சார்? அப்போ வீடு இவருக்கு அங்கே ஓட ஆரம்பிச்சிருங்களா? ஃபோன் பண்ணி கேளுங்க. அங்கே இது கிளியராக தான்.
Hello. Hello. Hello, can you hear me? Hello.
ஹலோ ஹலோ
ஹலோ
Hello, am I audible? Hello.
ஹலோ பிரபாகரன் கேக்குதா?
கேக்குது. உங்களுக்கு கேக்குதா?
ஹலோ
ஹலோ.
ஹலோ
ஹலோ, கேக்குது எனக்கு கேக்குது. ஹலோ.
யா ஆடிபிள் ஒன்லி.
ஓகே. என்னோட ஸ்லைட்ஸ் விசிபிளா இருக்கா?
ஓகே ஓகே எல்லாம் கிளியரா இருக்கு.
ஓகே. ஸ்டூடண்ட்ஸ் வந்துட்டாங்களா? இன்னும் இல்லையா?
யா
Sir, shall we start,
எவ்ரிதிங்
sir?
ஓகே.
Sir, shall we start the session?
இந்த வந்துட்டு இருக்காங்க. இப்ப சாப்பிட்டாங்க. இந்த வந்துட்டு ஓகே.
ஹலோ இந்த வந்துடுறேன் சார்.
Sir, shall we start the session? Sir, shall we start the session, sir? Shall we start the session, sir? Prabhakaran, sir, can you hear me?
Hello. Yes, madam. Shall we start?
Sir, shall we
Yes,
start the session, sir?
Yes, madam. Please.
Okay, sir, thank you. I invite Swati to give the guest intro about Prabhakaran, sir.
I am happy to introduce Dr. D. S. Prabhakaran, who is a post-doctoral research at Shangbuk National University, Republic of Korea. He has completed his PG diploma in fermentation technology, Master of Science in Biotechnology at Jamal Muhammad College, Tiruchirappalli, and Doctor of Philosophy in Medicine, majoring in radiation oncology at Changbuk National University, Korea. Moreover, he is a proud alumni of PONNAIYAH RAMAJAYAM Institute of Science and Technology, Thanjavur. His research experience include working as a visiting postdoc researcher at National Institute of Radiological Sciences, Japan, and research assistant in Bharatdasan University, Tiruchirappalli. Moreover, he is also currently working as a visiting faculty at Ayya Nada Janaki Ammal College, Sivagasi. His key area of research focuses on a radiation-resistant X-ray C-ion, EGFR signaling, Notch signaling, epithelial mesenchymal transition, cancer stem cells, cancer immunotherapy, radiation production. He has submitted his thesis on a functional study of FTS on EGFR and not signaling in human uterine cervical cancer. His fellowship and award include Fellowship for Pursuing PhD Research from prestigious Brain Korea 21 and National Research Foundation of Korea, founded by Ministry of Education, Science and Technology, Government of South Korea. Postdoctoral Fellowship Award from the National Research Foundation of Korea since March 2017. Junior Research Fellowship in UGC project entitled Improving the Production of Aristological Acid from Aristolochia indica and A. bracteolate through in vitro culture, a potential antivenom and anti-cancer drug in Department of Plant Science of 1 lakh AOW at the Gazan Medical Conference held on 13th November 2015 at College of Medicine, Changbuk National University, Cheongju, Republic of Korea. Furthermore, he has a total of 22 publications in reputed journals. With this introduction, I welcome Dr. Prabhakaran to give his talk on role of radiation therapy in cervical cancers. The session is yours, sir.
Shall I start? Shall I start the session, madam? Hello. 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 Hello, am I audible? Hello. Hello.
பஸ் சரி அவங்க பேசுகிறது உங்களுக்கு கேட்குதா இல்லையா? அவங்க பேசுகிறது உங்களுக்கு கேட்குது, நீங்கள் பேசுகிறது அவங்களுக்கு கேட்குதா?
Hello?
Hello.
Hello?
Am I audible?
Hello?
Shall I start? Shall I start? Hello.
Hello? கேக்குதா?
ஹலோ கேக்குது.
பிரபாகரன்?
கேக்குது. ஹலோ. ஸ்டார்ட் பண்ணவா? ஹலோ.
ஹலோ?
ஸ்டார்ட் பண்ணலாமா?
ஸ்டார்ட் பண்ணுங்க, ஸ்டார்ட் பண்ணுங்க.
ஓகே. எது இன்ட்ரப்ஷன் இருந்தா எனக்கு மெசேஜ் பண்ணுங்க வாட்ஸ்அப்ல. ஹலோ.
வந்துட்டாங்க ஸ்டூடண்ட்.
ஓகே, ஓகே.
ஸ்டார்ட் பண்ணுங்க.
Okay, thank you. Thank you for a elaborated introduction about me. Before starting my talk, I would like to thank and remembering our beloved chairman and founder of our PRIST University, the man who introduced quality and disciplined education in the collegiate level. His ideas were followed by several other institutions across the states. As alumni of our PRIST University, I witnessed the growth and the quality of education given by our beloved faculty members. I wish the institute to reach its greater heights in the upcoming years. At this occasion, I am honored to be a presenter at this ImmunFest 2023 International Conference, and importantly, sharing the dais with the eminent persons like Professor and Head Dr. Nadarajan Gopalan, Department of Epidemiology and Public Health, Central University of Tamil Nadu, and Dr. Senthil Nayagam from SB Hospital, and Dr. Balakrishnan Sir, Professor and Head Retired. Also, I thank the present Vice Chancellors, Chancellor, Pro-Chancellor, Registrar, Director of Research, Dean, Head of the Department, faculty members, and my dear young minds. A very good afternoon to you all. Here, I have a question to our student participants. Does anyone know what is cervical cancer and how it causes? Dr. Kamaraj, please pass the mic to the student participants. I want this session to be an interactive session with the students. Hello. Please pass the mic to the students. Does anyone know what is cervical cancer? Hello. Please unmute the audio and please.
Sir.
Yes. Does anyone know what is cervical cancer and how it causes?
1 minute, sir. I'll pass on to the
Yes.
students, sir. I'll pass it to the students, sir.
Yes, please, please.
Hello, sir.
Yes, please.
It's, hello.
Yes.
Uh, yes. Cervical cancer is cancer in the cervix region of the female reproductive system.
Yes, yes. How it cause?
Um, Cancer is usually due to the uncontrolled growth of cells.
Yes, that is cancer. But what is the main cause of cervical cancer? Is there any? Do you have any idea about it?
Mutations.
Do you think that virus causes cervical cancer? Do you know HPV, human papilloma virus? No idea.
Yes.
Okay. Today let's discuss. Okay, thank you. Thank you for your response. I will move on to the next slide and explain what is cervical cancer. Cervical cancer, which is the third most deadliest cancer in the women worldwide. The cancer which occurs in the cervix, which is the lower part of the uterus, which connects the vagina, where the various strains of human papilloma virus, which plays the main cause, and it is a sexually transmitted infection which causes cervical cancer. When individual who exposed to human papilloma virus, typically our body immune system which prevents the virus from doing any harm. But in a small percentage of people, the virus survives for several years and contribute the process which causes the cervical cells to be the cancerous cells. And here there may be a question arises to you, is there any way to reduce the risk of causing cervical cancer? Yes, there are some ways to reduce the risk of getting cervical cancer. As per CDC, Center for Disease Control and Prevention, they have recommended the women aged 30 years above should undergo a cervical screening test, which is said to be a pap smear test. I think in India we can do at the cost of 1,000 to 2,000 rupees. CDC highly recommended the women aged 30 years above must undergo a routine check 3 years once. It is nothing but a pelvic exam where a swab of culture will be taken from the clinicians and observed under the microscope and check the normal as well as the abnormal cells. Based upon that, they will start the treatment at the earlier stage. It is a very important thing. And there are some signs and symptoms which include abnormal vaginal bleeding and during menstruation as well as during intercourse, as well as the pain in the pelvic region. And there are signs and symptoms which are seen in the more advanced disease stage, such as swelling of legs, problems while urinating in the bowel movement, as well as blood in the urine. There are more than 120 different types of HPVs. Among that, 40 can infect the epithelial lining. As I said you that most of them are harmless, but there are about 30 types of HPVs which are put you at the risk of inducing cancer. These HPVs which are classified into two types. The one is the low risk HPV which causes genital warts, and here the picture which shows how the warts which looks like. It is a very common in most of the individual. It will be on the face as well as the neck region. And the high risk HPV which leads to cancer in the cervix as well as vulva, vagina and anus in women. In men it leads to cancer in the anal as well as in penis. The low risk HPV which are classified two subtypes, namely HPV6 and HPV11, and the high risk HPV which is HPV16 and HPV18. These are the two subtypes which causes cervical cancer. Among these, there are set of genes which plays a major role, where here the early genes are said to be E5, E6 and E7, which plays the key molecule in initiation as well as the progression of cancer, and the early genes E1, E2, E4 and E5 which are involved in the viral replication. And here the E2, which is the major regulator which regulates the E6 and E7 and causes cancer. Here, this picture which clearly represents when individual who infect by the HPV, which typically affects the basal epithelial layer of the cervix, it will sustain in the human body for several weeks, and it will do its viral replication. It is just a small circular double-stranded DNA which releases the viral capsid into the healthy cells. Upon several days, it will replicate from 10 to 30 years, and then if it left untreated, it will be in invasive stage. And this figure which shows you very clearly what are the stages of cervical cancer. Here in the top image, if you see, when a HPV which infects the basal epithelium, which releases the viral capsid into the cells, and then the viral DNA, which consists of a set of early as well as the late genes, each of the genes which have a several role. In the first stage, which is said to be SIN1, which is cervical intraepithelial neoplasia 1, where it affects the basal layer and multiply at the epithelium, and the stage 2, which is said to be SIN1 to SIN2, which is cervical intraepithelial neoplasia stage 2, where here it affects the lower half of the epithelium. Upon entering, the virus which first targets the tumor suppressor protein P53 and degrade its actions, as well as the E7, which targets retinoblastoma, which is the tumor suppressor protein, and inhibits its action and hijack the entire genome and takes the genome machinery and passes the viral replication cycle. At the later stage, which is said to be SIN3, which infects the entire thickness of the epithelium and leads to cancer. Here, how the normal cervix which looks, and the stage 1 infected, which is SIN1, and the third image which shows the stage 2, which is SIN2, and the fourth image which shows the SIN3, how the infected cervix which looks in the pictorial representation. And what are the mechanisms of E6 and E7 carcinogenesis in cervical cancer? Upon infection, as I told you, that E6 which downregulates the tumor suppressor protein P53, and the E7 which downregulates the tumor suppressor protein RB, and it ultimately leads to inhibition of immune responses, immortalization as well as transformation, inhibition of apoptosis and inhibition of differentiation, and it gathers several accumulation of mutations which leads to cancer. And here I would like to share our research. We are working on fused dose homologue. Fused dose homologue, which is a gene initially identified in the mouse mutants, due to the several hundred kilobase per deletions in the region, which is said to be FT1 or FTS. Importantly, this FTS, which identified the 96% homology and the interaction screen with the AKT1, which is said to be a cell survival marker, and to control the AKT phosphorylation by PDK1. Importantly, FTS is a member of E2 variants, which shares an extensive sequence homology to the ubiquitin conjugating domain and lacks the critical cysteine residue for the enzymatic activity. For the first time, our laboratory identified the FTS in human cancer cells. My professor is a MD PhD clinician come radiation oncologist. He first screened the set of genes which are overexpressed from the radiated patients. Two more cervical cancer patients who underwent radiotherapy. He have taken some of the tissue specimens and screened the markers which are overexpressed after radiation therapy. And then he wanted to know whether the overexpression which is involved in the progression of cervical cancer among the set of cervical tissue specimens, as well as in EGFR signaling, NOT signaling, and epithelial mesenchymal transition. This is a very important slide which I want to show various stages of cervical tissue specimens from the patients. The first one, which is the normal cell, and here this is the normal to the SIN1 stage. Here, if you see, the tissue specimens were stained with the FTS. Here you can see a gradual increase of FTS expression, the brown color, at the SIN1. And from SIN2 to SIN3, as well as SIN3 to carcinoma, you can see a gradual increase of the FTS protein. We speculate that FTS is involved in cervical carcinogenesis, but the molecular level understanding we don't know. And then what I did, I have chosen Casky and Siga, two cervical cancer cell lines, which are HPV-16 positive. Here we identified the E6 and E7 markers which are highly expressed, and then we wanted to check whether silencing of FTS by siRNA mediated approach having any significant effect. But silencing of FTS greatly reduces the E6 as well as E7 oncoprotein in Casky as well as SIGA cells. Importantly, silencing of FTS, which increases the tumor suppressor protein as well as the P53 and phospho-RB. And then we wanted to check the reverse change, whether E6 silencing or E7 silencing, which affects the FTS protein. Interestingly, we observed that E6 silencing as well as E7 silencing reduces the FTS expression and gradually increases the tumor suppressor protein P53 as well as the phospho-RB, which shows that FTS is involved in the cervical carcinogenesis, and FTS is necessary for the activation of E6 as well as the E7 oncoproteins. And then we wanted to check whether there is any change in the mRNA expressions, and the results shown there is no significant change at the mRNA level. And then we wanted to check the co-localization study by using immunofluorescence assay, and the results shown in controlled, you can see the green color, which is the FTS, which stained using the green color marker, and the second one, which is using red color marker E6 as well as E7. In the merged image, you can see the yellow color, the green and the red one, the combined yellow color, which shows FTS is co-localized with E6 as well as E7. For the progression of E6 and E7, FTS is the major factor. And then we have performed immunoprecipitation assay to see the interaction between these molecules. The results shown in the controlled E6, which is interactive with the FTS, as well as E7 also interact with the FTS. And then we have proved by using the cervical cancer patient tissue specimens, the green and brown color, which shows the clear co-localization of FTS and E6, and here FTS and E7. Taken together, the results which shown, FTS affects both HPV-16 E6 as well as E7 oncogenes in cervical cancer, and we propose that FTS as a target for the prevention of cervical cancer in development and progression. This work, which we have recently published in the PLOS One journal. Move on to the next topic. Here, radiotherapy, which plays a major role in treating several widely used to treat any kind of cancers. And the slide which shows Rongton, who discovered the X-rays at 1895 and got the Nobel Prize in Physics in 1901. And these are the history of radiation from the 18th century to the 2000th century. Usually, cancer will be treated by using three ways. The first, they go for a surgical therapy, the second one radiotherapy, and third one chemotherapy. In some rare cases, clinicians choose the combination therapy such as surgery, combined radiotherapy, or radiotherapy combined chemotherapy. Depends on the case and the severity of the disease. Here, radiation, which targets the DNA of the cell and causes single and double-stranded irreparable damage, which leads to cell death. Here, the major problem what we face, whatever damage which occurred by the radiation therapy, there are a set of markers and genes which are involved in the DNA repair pathway. This will repair the radiation-induced damage and nullify the radiation-induced effect. So our research is focused on how to increase the radiation sensitivity in the patient tissue specimens. Here, the slide which shows the radiation-induced effect, which are classified into two types. The one is direct effect, and the another one which is the indirect effect. Where here direct effect directly hits the DNA by electrons and affect it, and the second one, which is the indirect effect by the free radical formation and induces irreparable damage. As I told, some of the markers which are involved in DNA damage repair, when the cell is induced by IR, there are set of markers which is said to be ATM, ataxia, telangiectasia, mutated, as well as Ku70 and Ku80, DNA protein kinase. These are involved in repair the damaged DNA. This pathway is said to be non-homologous end joining. and the other pathway, which is homologous recombination repair. This repair pathway is recruited by the 53 BP protein. In day-to-day life, how we face the radiation. For example, if you travel on aeroplane from Hong Kong to Northern America, we will be exposed to radiation like 0.08 millisieverts of dose. Chest X-rays, they have used 0.05 millisievert of dose. Mammography as well as CT scan. As per the for the radiation workers, the annual dose limit, which is said to be 20 millisievert for a healthy individual. And then these are the slide which shows what are the types of radiation and how it prevent by the particles. Alpha rays, which are prevented by the piece of paper, and beta rays, which are prevented by a piece of wood, and gamma rays, which can be prevented by a block of concrete wall. X-rays as well as gamma rays, which will be blocked by the metal lead as well as the concrete walls. And move on to the next topic, notch signaling. Here we wanted to know the role of FTS on notch signaling. Notch signaling, which plays a major role in activation of stemness, and the stemness which leads to the treatment resistance, where here the notch signaling molecule, which consists of 4 receptors, namely notch1, notch2, notch3 and notch4, and the known ligands, which are JAG1, JAG2, delta like 1, delta like 3 and delta like 4. Upon interaction with this ligand and receptor interaction, the signal get activated. When the notch1 receptor which get activated, which in turn translocate to the nucleus by the set of molecules called gamma secretase complex, which activates the notch target gene S1, and it ultimately leads to activation of cancer stemness, which regulates the gene expression as well as differentiation, proliferation, and importantly maintaining the stem cell population, which ultimately leads to cancer. Here we hypothesize that FTS would be a target for treating EGFR as well as in notch signaling molecules and increase the effect of radiotherapy in cervical cancer. First, here we perform the experiment by using the X-rays. Here we have chosen 4 cervical cancer cell lines, namely ME180, HeLa, SIGA and Casci. First we check the expression pattern of all the notch signaling molecules by irradiation treatment. We found a time-dependent increase of notch signaling molecules as well as the gamma secretase complex molecules. And then we wanted to know whether FTS silencing have any effect on reducing these molecules. In lane 2, you can clearly see the increased expression of the notch signaling molecules, and further silencing of FTS combined irradiation treatment greatly reduces the all signaling molecules. And then we have seen the interaction between these 2 proteins by immunofluorescence assay. Silencing of FTS reduces the notch1 as well as cleave notch1. And then we check the interaction pattern by using cervical cancer patient tissue specimens. The results shown a clear interaction between FTS as well as the notch1 signaling molecules. And then we wanted to check whether is there any effect in the transporter molecule gamma secretase such as persilin1, persilin2, nicastrin and pen2 and the notch target gene S1. The important results we got: radiation induced increase of this set of molecules which are greatly reduced in FTS silence treated group. And then we documented the interaction between notch1, cleave notch1 and S1 by immunoprecipitation assay as well as in silico analysis. And then we wanted to check the spheroid formation assay. These spheroids, which are said to be the in vivo tumor models, which will be grown in ultra low attachment dishes, it will mimic the in vivo tumors. This is a hallmark to check the cancer stem cell proportions as well as the stem cell markers. In figure B, if you see in control as well as in radiation treatment, which does not affect the spheroid formation, but in FTS silence treated group as well as combination of irradiation treatment, which greatly reduces the spheroid formation as well as importantly the cancer stem cell markers NANOX, OX2, OX4, OX4A. Taken together, we conclude that FTS silencing significantly inhibits the radiation-induced stem cell marker as well as the spheroid formation. And we propose that FTS as a potential target for the Notch-mediated radioresistance in cervical cancer. This work, which we recently published in International Journal of Biological Macromolecules. Move on to the next topic, heavy ion radiotherapy. As many of you may not aware about this heavy ion radiotherapy, which is an advanced treatment module for treating the patients. Here, QST, which is the National Institute for Quantum and Radiological Sciences and Technology, which is situated in Japan, Chiba, formerly known as National Institute of Radiological Sciences, NARS. This is the first center where they started to treat the patients by using carbon ion beams since 1994. You may have a question, what is a carbon ion? The particle which are heavier than the helium, the carbon, neon, silicon and argon, these are said to be the particle beams or heavy ion beams. Here, the important thing is the amount of dose which distributed in the precisely at the cancerous region. That is the hallmark of this particular carbon ion beams. There are only 8 centers all over the world. They have this treatment facility. 4 at Japan, 1 in America, 1 in China, and the other 3 at European countries. The orange color, which shows the under construction of heavy ion beam all over the world. In Korea, they are setting up of 4 carbon ion beam facilities. It will be under operation from the upcoming month. Beam facility in the another 4 years. Calcutta Tata Memorial Institute, they are planning to construct this particular beam line. Here, this slide which shows the important characteristic futuristic of X-ray as well as proton beam and the carbon beam. Here, if you see the image, the x-axis which shows the body surface, the y-axis which shows the amount of dose, the green color, X-rays, as I told you that X-rays which are widely used for cancer treatment, the problem in X-rays are X-rays which deliver the high amount of dose in the normal cellular region, where an individual, they get the very lesser amount of dose at the cancerous region, but the normal cells will be get affected. But if you go for heavy ion beams such as proton and carbon, when the beam which reaches the normal surface of the skin, it delivers the very lower amount of energy and increases the higher amount of energy and delivered precisely at the cancerous region and deliver the maximum amount of dose and causes irreparable damage and a very good treatment module for cancerous patients. Here I wanted to share the slide.
To radiation accelerators used in physics. But the difference is that the carbon ions this machine emits are much heavier than electrons or hydrogen ions. The heavier particles have more energy, meaning fewer therapy sessions are needed for effective treatment. And the biggest benefit is that there are significantly fewer side effects. X-rays shoot energy over a large area. Then the energy gradually focuses in on cancer cells. Energy once they reach the cancer cells. Of the 10 balloons, if the 7th is the cancer cell, X-ray therapy requires the bursting of the previous 6 balloons to reach the 7th cancer cell. Whereas in carbon ion therapy, none of the other balloons are affected and only the cancerous 7th balloon is popped. But not this is a carbon.
I hope you all get some clear idea about the difference between X-ray as well as the carbon ion beams. And here it shows the advantages of carbon ion beams, which will be a less pain, which is highly suitable for elderly patients as well as curative in the early stage. This carbon ion beams, which is highly effective in X-ray resistant tumors, and the treatment time it will be a very short duration and it is feasible. And there are some of the cellular factors which are involved in cancer stem cell radio resistance, which are slow proliferation, increased DNA damage repair, low ROS levels, as well as the attenuated of pro-apoptotic pathways. And here we got beam time from the National Institute of Radiological Sciences, and we started the project to see the role of FTS by carbon ion beams, and we wanted to check the pattern. Interestingly, we identified that carbon ion beams increases the FTS expression, which the increase which shows, which is involved in the DNA damage repair and leads to a treatment resistance, where here we identified a set of markers and the Notch signaling molecules which are upregulated by the carbon ion beams. The Notch signaling molecule as well as the Notch target gene H1 and the gamma secretase complex persilin 1, persilin 2, nicastrin, as well as PEN2 were increased by carbon ion beams. And then we speculate whether silencing of FTS will have any effect on carbon ion beams. Interestingly, we found that silencing of FTS combined radiation treatment significantly reduces all the Notch signaling molecules as well as the gamma secretase complex molecules. And then we have shown the interaction between the set of markers by carbon ion beams, and further silencing of FTS greatly reduces the Notch1, cleave Notch1, as well as the Notch target gene H1. And then we checked the spheroid formation ability in Cascade as well as in ME180 cells. Carbon ion beams does not affect the spheroid formation as in X-ray, as like X-ray beams. But in FTS silence treated group as well as combination of FTS silencing, we identified a clear reduction of the spheroid formation as well as the cancer stem cell markers. And then which reduces the spheroid size as well as the cell viability. More importantly, which reduces the clonogenic cell survival in Cascade as well as in ME180 cells. This is the first ever report which shown FTS silencing combined carbon ion beams, which targets the Notch signaling and reduces the spheroid formation as well as the cancer stem cell markers. This work which we recently published in Frontiers in Oncology. And this is a slide which shows during my PhD and postdoctoral research, what are the works which I done by FTS in numerous cellular signaling. During my PhD, I studied the role of FTS on EGFR signaling by X-ray irradiation as well as cisplatin and Notch signaling by X-rays. And then further, I wanted to extend my study in the postdoctoral level by using carbon ion beams at Japan. I have been visited 14 times to QST Japan and stayed for 16 to 31 days per visit for utilization of this carbon ion beams. Our institution having 2 collaborative research grants at QST Chiba, and these are some of our publications by FTS. The slide which shows the projects which we are currently working on, the 17J120 and 18J333, where here we are identifying the radioprotective effect of peptides by Cryptosporidium parvum in normal as well as in the cancerous cells. This is the outline of the institution. This is the entry of National Institute of Radiological Sciences, where this is the beam facility. HIMAC, which is meant by Heavy Ion Medical Accelerated in Chiba. This hospital, which is running round the clock from morning 7 a.m. to 7 p.m., they will use to treat the cancer patients by using carbon ion beams. After 7 p.m. to the next day, early morning 7 a.m., the beam facility will be open for the international researchers. They will utilize the beam for their physics as well as radiation biology studies. This is the plan of beam line at NIRS, and these are the image when I got the beam time at QST. I have been irradiated the cancerous cells at using carbon ion beams at Japan. Here he is our Professor Woo Yun Park, my PhD advisor and my postdoctoral research guide. And the down picture, he is our collaborator, Professor Takashi Shimokawa. He is the coordinator of our biological experiments. And this is Professor Furusawa. This person involved in the construction of beamline since early 1990s. And here, Korea, they are planning to setting up the heavy ion facility at the Daejeon. Totally, Korea is setting up 4 carbon ion beam facility, 2 from the private and 2 from the government. One is from the Yonsei Cancer Center and another one is from the Hyundai Group. This is constructed by our government, and this project, which is the highest budget project from the Republic of Korean government. And this is our end of my slide presentation. And here I want to request some of the audience to take part in this special issue. I am serving as a guest editor. Whomsoever working in radiation biology or cellular and molecular immunology related research, please contact me and give an opportunity to publish your paper in these journals. And here I am serving as a president of Indian Student Association at Thanjavur National University since 2011. Our institution is signed a MOU with various Indian institutions such as Madras Christian College, Central University of Pondicherry, Satyabama University, and some of the private institutions around New Delhi. These are the pictures we taken during the MOU signing ceremonies. And my sincere thanks to this PONNAIYAH RAMAJAYAM institution for organizing such a wonderful event and giving an opportunity to interact with the young minds. And also I am giving my special thanks to the Indian Immunology Society for funded this international seminar and get into a successful way. Thank you.
Thank you, sir. Do we have any questions? Thank you, sir. That was a very wonderful explanation about cervical cancer. Thank you for your fruitful speech.
Thank you.
So shall we wind up the session, sir?
Okay, shall I sign out?
Thank you, sir. Thank you for coming.
Thank you, madam.
That was really truly a fruitful thing for us
Thank you.
about cervical cancer. Thank you so much, sir.
Thank you. If I get the chance, if I visit India, I will come. I will come and give my talk in person.
Sir, pardon, sir.
Yeah, when I visit India, I will come and give my lecture in person at the institute. I think some of the slides you may
Sure,
not be
sir.
visible.
You're
I
always
think
welcome, sir.
yes.
You're always
I think
welcome,
the screen
sir.
size is very small. I don't know how many of you get the important points from my slide.
Sure, sir. You're always welcome, sir. The university is always
Okay.
open for you, sir.
Thank you.
Thank you so much, sir.
Thank you.
So kind of you. The poster session is getting started. The first participant. Nadrajan Gopalan. Is anyone is willing to present first? Is anyone is willing to present first?
Am I present there? Can I
Yes,
present?
sir. Dr. Lal.
Yes, ma'am.
Sir, please start your presentation, sir.
Yes, ma'am.
Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am. I'll present, ma'am.
Ma'am, Dr. Lal is presenting. Please wait, ma'am.
Yes, ma'am. Yes.
Yes, sir, you can start your presentation, sir, please.
Yes, ma'am.
Yeah, it is visible, sir. Your presentation is visible. You can start now. Yes, thank you, sir. It is presented. Yes, sir, it is viewed, sir.
Good afternoon, everyone. This is Dr. Parallel, Department of Physics, Vanasli Vidyapith, Rajasthan. The title of my presentation: An attunement on ultraviolet gain characteristics of aluminum gallium nitride for disinfection applications in immunology. This is layout of my presentation: introduction, theoretical details, results and discussions, conclusions, and references. Now, introduction. First, we define heterojunction. Heterojunction means junction or interface between two dissimilar band gap materials, and the combination of multiple heterojunction is known as heterostructure. Heterostructure has minuscule dimensions is called nanoheterostructure. There are three types: Eastern SCS, Green SCS, and quantum SCS. Eastern SCS means step index nanoscale separate confinement heterostructure, and Green SCS means graded refractive index nanoscale separate confinement heterostructure, and QWSCS means quantum well separate confinement heterostructure. Now, theoretical details. This is optical gain equation. Here G is the optical gain coefficient as a function of photonic energy in terms of charge of electron, Planck constant, classification levels, temperature, and bulk momentum. Optical gain is measured in per centimeter. This is modal gain confinement factor. Here gamma is the ratio of modal gain and optical gain. It is always less than 1. And this is modal gain equation. GMS bar omega is the modal gain as a function of photonic energy. It is the product of optical confinement factor and optical gain. It is also measured in per centimeter. This is differential gain equation. G dash is the differential equation, and this is the differentiation of optical gain with respect to carrier density. It is measured in centimeter square. This is differential refractive index equation, and dash is the differential refractive index as a function of photon energy. It is the differentiation of refractive index with respect to carrier density. It has no unit. This is anti-guiding parameter. Alpha is the anti-guiding factor in terms of differential refractive index and differential gain, and K is the wave vector. Alpha has no unit. This is optical attenuation equation. A is the optical attenuation factor, and L is the length of optical fiber, and PO and K are the output power and input power. Now, results and discussions. This figure represents the behavior of ultraviolet gain with respect to lasing wavelength. The red graph represents the behavior of ultraviolet gain without radiations with respect to wavelength. The maximum ultraviolet gain is obtained at the 2000 per centimeter at lasing wavelength 254 nanometer. When neutron radiation are induced, then the gain is enhanced and it reached as the 3000 per centimeter. It is shown by blue graph. In the results, the maximum ultraviolet gain is obtained at the 254 nanometer. It is very useful in the disinfection applications like purifying of water, surfaces, and air to remove the bacteria and other harmful contaminants. Now, conclusions. In this study, an emerging cathode P theory has been utilized. The intensity performance of ultraviolet gain spectra with ultraviolet photons wavelengths of aluminum gallium nitride has been investigated and analyzed at neutron radiation influence of 5.6 into 10^11 centimeter square. Owing to the prevalence effect of neutron radiations, the potential height of barrier in scale layers is unhinged, hence leakage of electrons occurs diminished. As a consequence, intensity of ultraviolet gain is obtained in unhinged order. In the analytical results, the PRIST value of ultraviolet gain intensity 2750 per centimeter has been achieved at the ultraviolet photons wavelength of 254 nanometer. This ultraviolet light of wavelength 254 nanometer has also been used in the high purification to disinfect the surfaces, air, and water by eliminating the various types of viruses, bacteria, and harmful contaminants. In today's test, ultraviolet light of wavelength 254 nanometer can also be used for the biochemical testing, molecular detectors, and the DNA analysis. These are references. Thank you, ma'am.
Thank you, sir. That was really a wonderful presentation. Next,
Thank
I
you, ma'am.
call upon Dr. M. Murugan to present the slide. Murugan, sir?
Yes, madam, I'm here.
Yes, sir. Present your slide, sir. Murugan, sir.
1 minute.
Murugan, sir, can you hear me?
Yeah, yes, madam.
Yes, present your slide, sir. Your presentation is not visible, sir. Yes.
Not here.
Now it is okay, ma'am?
Yes, sir. Now it is viewed. Pyridol, kindly mute your mic.
Okay. Okay. Can I let me start
Yes,
the
sir. Your
presentation, ma'am?
slide is visible now. You can start now.
Okay, okay. Thank you very much, ma'am. Thank you very much. This is Dr. Murugan from Sri Kalishwari College, Department of Botany, Sivagasi in Tamil Nadu. My presentation title is Preliminary Phytochemicals Analysis and Antimicrobial Assessment of Leaf Extract of Dodonia viscosa. This is a very famous medicinal plants. In our area, this is plenty of plants are available. As my introduction, India has a strong wealth of medicinal plants which are used traditionally in Ayurveda, Unani, and our Siddha medicine also. Many healers and tribals also utilized their medicinal plants for their generation to generation. In approximately 25% of people, they were used the herbal medicine in the developed countries. The medicinal value of Dodonia viscosa lies the presence of very intensive bioactive molecules. That's why this is very useful to the protecting of a physiological effect of human body. Our study materials methods, the fresh leaves of Dodonia viscosa are collected from Mukutu Malay village. It's in a small hills located nearby Kovilpatti Taluk in Thoothukudi district, Tamil Nadu. The collected leaves are immediately dried in the room temperature under shade condition up to 10 days and then powdered with the help of a mixing grinder, and then 50 grams we are taken as a Tudonia viscosa, the dried leaf powder. Then cereal dilution, cereal solvent extraction methods we are taking with the help of 250 ml of 250 ml of different solvents like ethyl acetate, acetone, ethanol, and aqueous. With the help of a Saxlet apparatus, we get extract, crude extract, and then stored off for 4 degrees Celsius for further study. As our result, we got the crude part of we are testing for 15 testers. Among the 15 testers, 13 biochemicals are present, such as saponin, flavonoids, bitosterol, phenylicum, mucilate, terpenoid, steroids, carbohydrates, and amino acids are present in the particular leaf. This is our workflow. Just this is our area of plant collected, were located in Mukutu Malay. This is the leaf structure. This is an extraction unit. As for the second aspect of, we are going to analyze the antimicrobial activity. Here ethyl acetate having the candida albicans 14 mm zone inhibition is zone, followed by Staphylococcus aureus 4 mm only, and Bacillus tuberculosis 13 mm, Salmonella typhi 12 mm, Bacillus cereus 12 mm, and Enterococcus facialis 12 mm, Klebsiella pneumonia 12 mm, Serratum maracin 11 mm, and Escherichia coli 10 mm. From the ethyl acetate solvent extract, here inhibition zone Bacillus 11 mm and Enterococcus 10 mm, Stabilo 7 mm, followed by E. coli 7 mm and Klebsiella pneumoniae 7 mm. Whereas the acetone extract showed the higher inhibition zone produced by the fungus Candida albicans 11 mm, followed by bacterial produced the higher inhibition zone such as Bacillus cereus 14 mm, Escherichia coli 13 mm, Salmonella Staphylococcus and Aedes 9 mm, and Serratia americans zone is absent. The next solvent of acetone solvent, the higher inhibition zone showed by produced by Bacillus 7 mm, Escherichia coli 7 mm, and others are absent. The study of species found to great source of medicinal important bioactive compounds are present, and also this plant acts as a good nutritional sources. Then finally we got a good antimicrobial activity for the root activity. So we recommend for the conclusion of to use the plant in the coming future day, for recommended for the pharma company to take this plant. This is all our expression of figure chart, so that already I discussed in the detailed manner. So this is an wonderful medicinal plant. My suggestion and come to the conclusion about this plant, go for cultivation. The wild is now available, but the cultivation is an important for the conservation of particular plant because of a good antimicrobial activity and phytochemical analysis activity of the result. This is of some my reference. Thank you very much.
Thank you, Murugan sir. That was wonderful presentation. Next presentation was.
Ma'am, can I present?
Who's that?
Hello.
Ma'am, this is
Monica.
Dipanshu.
Yes, ma'am. Can I present?
Yes, ma'am. You can start now.
Okay.
Ma'am, next, can I present my people?
Yeah. Next, you'll be the next one, sir.
This is
Please wait.
Dipanshu Pandey. Please mention my name, Dipanshu Pandey.
Sure, sir. But they are also waiting at the same time, sir.
Ma'am,
Please.
kindly share the list.
Ma'am, are you able to see my screen?
Murugan sir, kindly. Yes, thank you, sir.
Hello.
Hello.
Monica, now you can start your
Hello.
presentation, Monica.
Okay, okay, yes,
Only
ma'am.
5 minutes. Only 5 minutes. Others also waiting. Kindly wait.
Okay, yes,
Only
ma'am.
5 minutes.
So the topic of my study is characterization of nanoparticles incorporated into lipid membranes. So initially, I will introduce you what are lipid membranes and the gold nanoparticles and the techniques which I have used to characterize the lipid membrane and nanoparticle system, that is UV visible, FTIR, dynamic light scattering and transmission electron microscopy. At the end, I will discuss the results
Your
and
presentation is
conclude.
not visible, Monica.
It's not visible? Okay.
Yes.
Let me share again now. Is it visible now, ma'am?
No, no, no. Yes, yes, it's now visible.
Okay. Hello, good afternoon, everyone. So today, the topic of my presentation is characterization of nanoparticles incorporated into lipid membranes. So the presentation is divided into four parts. First, I will introduce you about the lipid membranes and gold nanoparticles, then the techniques which I have used to characterize the membrane and nanoparticle system, that is UV visible, FTIR, dynamic light scattering and transmission electron microscopy. At the end, I will summarize my results and conclude. So the objective of my study is to form gold nanoparticles loaded lipoliposomes which mimic the lipid membrane and then evaluate their stability of the nanoparticles incorporated into these lipid membranes by UV visible spectroscopy, and the effect of size of nanoparticles was also checked on lipid membranes, and finally the lipid membrane was analyzed by Fourier transform, infrared spectroscopy, calorimetric studies, and fluorescence spectroscopy. Then the data was analyzed to evaluate the interactions of gold nanoparticles with lipid membranes. So this is the proposed structure of the DPPC lipid liposome interacting with the gold nanoparticle. It seems that the lipid liposome is having a head group, that is a round structure, and a fatty acid tails in inwards, and head groups are outwards. So the gold nanoparticles are interacting with the head group of the lipid membrane. So on the right hand side you can see the structure of the lipid, and the lipid which I have studied is DPPCs 1,2 lipid which I have studied is DPPCs 1,2 dipalmitoyls in glycero 3 phosphocholine. So this lipid has two fatty acid acyl chains, and this is a polar head group which is connected to the phosphate group and the glycerol backbone. So now you can see here on the left hand side the UV visible spectra of the lipid DPPC liposomes. Here there is a small interactions around small peak around 222 nanometers wavelength, and once we add the gold nanoparticles, 5 nanometer size gold nanoparticles into DPPC liposomes, so the transitions comes around 525 nanometers. So the stability of the gold nanoparticles have been checked. So this is the gold nanoparticles, the black one, incorporated gold nanoparticles into DPPC liposome. There is a broad transition, and once and after 1.5 hours also the spectra remains the same. It means the system is stable even after 1.5 hours. So to check the size of the gold nanoparticles incorporated into DPPC liposomes, the transmission electron microscopic study have been done. So you can see here on the top there are only DPPC liposomes which are having 0.29 micrometer and 1 micrometer, as they are of different size because they are multilaminar liposomes. They are not of the uniform size, they are multilaminar, so they could be of different sizes. So when the gold nanoparticles were incorporated into the DPPC liposomes in 1:1 and 5:1 ratio, all the DPPC liposomes, all the gold nanoparticles surrounded the DPPC liposome. They are interacting with the DPPC liposomes. As the interaction started, the black structure in the is the gold nanoparticles. So the clustering of the gold nanoparticles also happen because they have the self aggregation properties. So FTIR spectra has been checked. There are two main symmetric and antisymmetric stretching vibrations of the lipid: CH2 stretching vibrations and antisymmetric vibrations. And black curve shows only the symmetric CH2 symmetric and antisymmetric particles have been added in different concentrations of 1:1, 5:1, 10. The intensity of the peak has sharply declined, and it means the gold nanoparticles are interacting with the methylene chains of the lipid. That's why the intensity of the peaks have decreased significantly. So the effect of temperature also have been studied on this system, because the temperature, when there is increase in temperature, the fatty acid chains in the lipid starts to melt. So in the DPPC, 42 degrees centigrade is the main transition temperature, where it exists in the liquid disordered state. And here we can see, with the increase in temperature, the wave number increasing, it means the fluidity in the membrane increases. So the temperature have been studied at 2920 wave number, symmetric stretching vibration and antisymmetric stretching vibration of 2850. So both the stretching and antisymmetric vibrations, the wave number increases with increase in temperature, which makes the lipid more fluid in nature. So at the end, I would like to conclude that the gold nanoparticles incorporated into lipid liposomes of zero point 0.3 micrometer have been observed by TEM and DLS. So the gold nanoparticles are found to incorporated into liposomes are found to be stable even after 1.5 years of preparation, as studied by ultraviolet visible spectroscopy. So FTIR study shows that the wave number of antisymmetric stretching vibration has increased by approximately 10 wave numbers, and symmetric stretching vibrations has increased by 6 wave numbers at the main transition temperatures of 42. So this significant increase in wave number of CH stretching vibrations by incorporating gold nanoparticles clearly indicates the strong interactions between the gold nanoparticles and lipid membranes. Thank you so much. Any questions, I would be happy to answer.
Thank you. Thank you. Next, I call Deepanshu Pandey to present the.
Yes. Okay.
Pandey,
Yes, ma'am.
you ready? Yeah, you can start
Yeah,
your
yeah.
presentation, Deepan Pandey.
So it is visible now. My screen is visible, ma'am.
Yeah, yeah, yeah. Okay, it is visible. Your slide is visible now. But it is. Yeah, okay.
Okay.
One
So
Deepan Pandey, continue.
what happened, ma'am?
Yes, you are
Can I
audible.
start?
Yeah, yeah, you
Okay.
can start. You're audible.
Thank you very much. Good afternoon, session chair, faculties, organizing committees, and fellow colleagues. My name is Dipanshu Pandey, and I am here to present my topic: synthesize, characterization, and medicinal application of chromones and flavose derivative. Chromones is an organic compound with a molecular formula C9H6O2. It is derived of benzopyrene ring, which is a class of organic compound containing a benzene ring fused to pyrrole ring. Chromone is often used as a building block in the synthesis of a variety of organic compounds, including pharmaceutical, agrochemicals, and natural products. The chemical structure of now this particular slide is the chemical structure of chromone consists of six-membered benzene ring fused to a five-membered pyrrole ring. The benzene ring provides the compound with an aromatic property, which means it is a stable ring structure and is resistant to chemical reaction. The pyrrole ring, on the other hand, is responsible for the compound reactivity, and it is the site in many chemical reactions. So these are the reviews which I have done throughout my experiment. So the chromones is having a very privileged structure. Due to this privileged structure, it is basically the chromones reaction is basically a nucleophilic reaction in which the molecules that donate electron to form a chemical bond with a positive charge atom. Chromones is a strong electrophile, meaning it attracts electrons. Now, by bringing the biological activities at different particular sites, these are the sites of this particular chromones derivative. The biological activity of chromone molecule is largely determined by the presence and position of different functional groups. So hydroxyl, as it is mentioned in the slide, the hydroxyl is going to bring the antioxidant and anti-inflammatory biological activity. Whereas amino group is bringing is showing the antibacterial, antifungal, and anti-tumor effects. The methoxy group, when attached with on at 2, 4, and 6 position of chromones, it is going to show the property of anti-spasmodic and analgesic. The nitro group is going to show antimicrobial and anti-tumor. Carbonyl anti-inflammatory. And last but not the least, the sulfonamide is going to show antibacterial as well as anti-tumor activities. So, what are the synthesis path? So these are the synthesis path of chromone derivatives. Chromone basically is synthesized with the help of condensation of substituted benzaldehyde and acetone. Modification on chromones. So chromones, by bringing basic modification in the chromone structure, the chromones is going to show a different kind of properties and biological activities. For example, like methoxy, nitro, and amino can be introduced in the compound and with the appropriate reagents such as methyl iodide, nitric acid, and aniline. So these are the reagent which is used for the synthesis of the first synthesis process which I am taking right now is going to be like the protonation of carbonyl oxygen of acetyl chloride by H plus catalyst to form a helium ion intermediate. In this intermediate is very important because that intermediate is going to follow the nucleophilic attack on hydroxyl group. And due to that, we are going to get a product of orthohydroxyacetophenol on ethylene ion, forming a tetrahedral intermediate. Elimination of pyridine hydrochloride as a living group regenerate the pyridine catalyst. And due to that catalyst pyridine, the formation of C double bond O take place of the acetyl group. And last, the deprotonation of the intermediate by the pyridine catalyst, leading to the formation of the final product, that is 2-acetyl compounds which I have synthesized in the laboratory. The infrared spectra, the IR spectra denotes what? Now I am going to explain the interpretation of 2-methyl-7-hydroxychromone infrared spectrum. In this, a broad and strong peak around 3,200 to 4,200, 3,400 centimeter inverse, which corresponds OH stretching, vibration of hydroxide. Just last 20 minutes, 20 seconds, ma'am.
Come to
Yes,
the
ma'am.
conclusion, ma'am. Conclusion.
Yeah, okay. A strong peak around this particular compound shows the position of this particular chromones. Now, these are the things, the conclusion. The synthesized chromone derivatives are expected to have a wide therapeutic use. Therefore, most potent drugs will be synthesized which have diverse medicinal application and are helpful to the society. Thank you very much for listening my presentation, and I am open to take the questions. Thank you, ma'am.
Deepanshu Pandey. Next, Priyadarshini. You can start your presentation. Deepanshu Pandey, please put your mic off. Yeah, thank you. Priyadarshini is in line.
Yes, ma'am. Now I'm present.
You can start your presentation now. Next presentation was Vikas. After Priyadarshini, Vikas Kushwala. Vikas Kushwaha. Next to Priyadarshini, followed by Vikas Kushwaha. Yeah, your presentation is Priyadarshini. Put on your mic on. Priyadarshini, put on your mic.
Yes, ma'am.
Yeah, you can start your presentation. Your slide is not visible.
No, wait, ma'am.
Okay. Okay, start your presentation. It's now visible. Only 5 minutes. For time consuming, only 5 minutes.
Okay, ma'am. I am Priyadarshini from the Department of Biotechnology, Bonsai presentation. The title is Study on Synthesis of Iron Nanoparticles Using Cashew Nut Shell Oil. The basic information about cashew nut is like evergreen tropical tree. It is a evergreen tree to introduce in other parts of world by Portuguese. So cashew nut has two shells or walls, outer one being smooth and somewhat elastic olive green in color. The cardinal is important component of cashew nut shell liquid, can be extracted from the shells readily and inexpensively. The cashew nut is a agro-based waste. It is novel regarded as an important tool to reduce the destructive effect of associated with the traditional methods. And synthesis is commonly utilized in laboratory and industries. Next, we see the aim and objective of the study is
Hello.
yes, ma'am. Okay. Uh, Cashew nut shell is a crude extract. It is the major and cheapest source material. It is naturally occurring the methyl cordals, polymeric materials, and anacotic acids. The main objective of this presentation is collection of cashew nut shell villar colangari, optimize the synthesis of nanoparticles using ferrichloride to study the characterization of synthesis nanoparticles by UV spectra, FTIR, and SIM. Next, we see the materials and methods of my work. Collection of cashew shell is I will obtain the large quantities of cashew nut shells and then extractions of ferrichloride by using magnetic stirring for continuous 3 hours and use the ferrichloride chemicals. This is my work on the cashew nut collection, the cashew nut extractions of oils, and the synthesis of magnetic nanoparticles. The result, the FTIR is used for the analyzing functional group. I identified the emulsion peak at 3172 centimeter in the power of -1 region, that is hydroxyl groups and cardinal. The another hydroxyl group is identified in the small peak at 2595 centimeter in the power -1. The identified nanoparticles formed and strongly interacted with the components of cashew nut shell liquid. Then we see the scanning electron microscopy. The VO18 SEM electron microscope. The average size is 5 to 200 in nanometer. Some are aggregated between 18 to 100 in nanometer. The most of the particles are spherical in shape. That is shown that are absorbed in 20, 50, and 100 in nanometer. Individual particles in round shapes at 20 to 75 nanometer. The obtained ferric nanoparticles might be had significant antibacterial activity, minimum inhibitory concentration, and minimum bactericidal concentration associated with many diseases. Next, we see the GCMS analysis. GCMS also used for the identified biological compounds. For time consuming, I only tell two compounds, biological compounds, antibacterial activity. The another one is phenol 1,3 pentosidyl. It is for antioxidant, anti-inflammatory, and anticoagulant activity. Then we directly go for conclusion. It is cashew nut shell is a liquid is mainly used for the natural protection against insects and innumerable applications such as frictions, linings, paints, laminating resins, rubbers, compound resins, cashew nut cements, polyurethane based polymers, surfactants. The external applications for the leprosy and the ringworm. The nanoparticle, besides the medical applications and magnetic resources, images in anti-cancer therapy. The further study will be conducted for the patient. Thank you for valuable time to spending my words. Thank you, ma'am.
Ma'am, kindly allow me because I need to go. My college is gonna close.
After Vikas, you can present. Oh,
Okay, ma'am.
thank
Thank
you,
Ma'am.
you.
Priyadarshini.
Okay, ma'am. Thank you, ma'am.
Next, Vikas, Mr. Vikas, you can present. After that, Shaka will be presenting.
Thank you, ma'am.
Your time limit is 5 minutes. Mr. Vikas, are you in line? Okay, then,
Ma'am,
Shaka, Miss Shaka,
it's
you
not
can
here.
present.
Okay, ma'am. Wait a moment. One second, ma'am. I'm gonna share my screen.
Your screen is not yet visible.
Ma'am, may
Yes.
I present next? Ma'am, may I present next, Rashmi Singh?
Yes, ma'am.
Visible. Yes,
Okay,
visible.
ma'am.
You can start now.
Okay. Good evening, everyone. So I'm Dr. Shakha from JGM Science College, Jammu. So today I'm going to present my research topic that is on species composition and varietal susceptibility of mango leaf hoppers on different varieties of mango in Jammu region. As you all know that mango is basically a king of fruit and rich source of vitamin A, C, B6. And India is the highest producer, but the high production of mango in India is not because of the high productivity, but it's because of the large area under its cultivation. Otherwise, it's facing a lot of damage as far as the yield is concerned because of lot many number of pests, which are actually causing great harm to this fruit crop. The major pest of this mango is mango leaf hoppers. So in Jammu also, there are so many varieties of mango which are being cultivated like Bombay Green, Desheri, Langda, Malda, Chosa, Amrapali. And all these varieties are facing a lot of damage due to these mango hoppers. So objective of my study is to identify what are the different hopper species which are associated with mango cultivation. And second, which variety is more susceptible to these hoppers so that our farmers could come to know that which variety should be planted here which is less get damaged less by these mango hoppers. And our experimental area mainly include Udewala and Nagbani. These are the two areas which are rich in mango orchards, and they found at an altitude of about 300 to 320 meters above the mean sea level. So this is my research area, and as far as methodology is concerned, methodology mainly include the sweep net method and backtrap method, during which lot many number of hoppers they were collected and were taken to the laboratory for its count, so that we come to know that what are the varieties which are actually responsible for causing damage. And second one is counting the number month wise, fortnightly first, and then converting it to month wise data, so that we could come to know that which variety is harboring more number of hoppers on it. So during the study, two main different species of mango leaf hoppers were seen. One is the Idioscopus clypealis, and second one is the Myrtotus atkinsoni. Both these two species are morphologically different. Like Idioscopus clypealis is smaller in size, light brown in color with creamish colored scutellum, having dark spots on it, and it's usually however the leaf or the bushy part of the mango. Whereas Emratodes atkinson I mostly seen sitting on the stem, and it's basically larger in size, dark brown in color, and it is having a arrow mark on it. So this is the data which I collected as far as the species composition is
Ms.
concerned, and the
Shaka,
data clear. Yes, ma'am.
it's now clear.
Am I audible? Am I
Yes,
audible?
ma'am, you are audible. Please start.
Ms. Shaka, I think your connection is bad.
Yeah, I know, ma'am. It's J&K, so the network connection is a bit weak here. Okay, fine. Should I continue, ma'am? Should I continue, ma'am?
Thank you, Ms. Shaka.
No, should I continue, ma'am? It's not over yet. Hello?
Uh, is your connection okay? Because we are not
Yeah,
hearing
ma'am.
you
Actually,
properly.
No, ma'am. This is audible.
ma'am, is my screen visible?
Yeah, visible.
Okay, fine. Okay, let me wind up with the conclusion part only. Okay. So as far as the species composition is concerned, for the first 6 months, it's basically Idioscopus clypealis, which seen its dominance, whereas in the next 6 months, it's basically Myrtotus atkinsoni, which showed its dominance. So this is all about the species composition. As far as the yield is concerned, as well as varietal susceptibility is concerned, so this graph clearly revealed that out of all these 5 different varieties of the Sherry, Langda, Malda, Amrapali and Wild, it's basically Amrapali which is more susceptible, followed by Malda, followed by Wild, and the Sherry. Langda is the least susceptible variety of all these 5 varieties. Thank you. Thank you, ma'am.
Thank you, Ms. Shaka. Thank you.
Thank you, ma'am.
Next, Ms. Rashmi Singh, are you in line?
Yes, ma'am. Yes.
Okay, you can now
I'm just
present it.
sharing my screen. Oh, sure.
After Rashmi Singh, Manasvi Paul can
Ma'am, I have
present.
lecture at 4:40.
Okay, Ms. Rashmi Singh, please take 5 minutes. Rashmi Singh will be followed by
Ma'am, is it visible?
Manasvi Paul.
Okay.
Yes, yes, Rashmi, your slide is visible.
Okay, ma'am. Thank you. Okay. Myself, Rashmi Singh, and I am assistant professor at DB College, Bundelkhand University. And my presentation topic is study of brucellosis disease in cattle of Uttar Pradesh. The aims and objective of my work is to assess the knowledge, attitude, and practices of cattle keepers regarding brucellosis disease in India, where ruminant brucellosis is present at a high level, and to study the serological prevalence of brucellosis in India, to aware the cattle keepers regarding the cattle's health, common diseases, and available vaccines. India, as we all know, it is an agricultural country and it has the largest cattle population in the world. Almost 30.5% of the world's cattle population is located in India. As it is clear from this graph also, India has almost 305 millions of cattle. And in this scenario, the unawareness of the cattle keepers become a big issue, and brucellosis is a very common disease which is found in cattle. And it is a zoonotic disease which affect the livestock and people worldwide. Almost 5 lakh human cases are documented, making it one of the most severe zoonotic disease in the world. And now, what is brucellosis disease? Brucellosis disease has been identified as a neglected tropical disease by World Health Organization, and these are the contributors like Bernard Bang, Sir David Bruce, Sir William Brunet, who contributed in the discovery of brucellosis. It is an endemic in countries belonging to Mediterranean region, Asia, Middle East, Latin America, and Africa. This disease will also have many names like undulant fever, Mediterranean fever, Malta fever, Banks disease, etc. Brucellosis is a kind of reproductive disease of livestock, and it induces infertility and delayed heat, interrupted lactation, abortions, and loss of calves, and loss of meat production and milk. Human mainly acquire this disease by consumption of raw milk or unpasteurized milk or contaminated meat. And now, what is the causative agent of the brucellosis? It is a bacteria which belong to the genus Brucella, and it is a gram-negative bacteria. And there are several species of Brucella, like Brucella abortus. It basically infects cattle. Brucella melitensis, it infects sheep and goats. Brucella suis, it infects pigs. Brucella canis, and it infects dogs and cats. All these species of Brucella can infect human also. But the Brucella militensis is the most common human pathogen. And now, what is the mode of the transmission? Ingestion of unpasteurized milk and milk products, as I have mentioned earlier also. The cuts and abrasion in skin, inhalation, like aerosol infection is also very common here. Poor farm hygiene practices like discarding the aborted fetuses with bare hands without wearing gloves or wearing masks, and when the reproduction like semen of infected bulls, when they copulate with the cow, the chances of the transmission is very high, and so the artificial insemination is necessary for these cases. Unrestricted trade and movement of the animals also a reason for the mode of the transmission. This is the picture of the clinical manifestation of the brucellosis, and it affects many parts like ocular, pulmonary, gastrointestinal, cutaneous, skeletal, urinary, cardiovascular, central nervous system. Basically, it causes weight loss and arthritis in bones become very weak, and it require a long antibiotic treatment for the treatment of the brucellosis in human. The great part is the vaccines for the brucellosis are available for the cattle. So, Brucella abortus RB51 strain developed for the immunization of cattle, and S19 is also another vaccine for the cattle. Brucella melitensis Rev1 for small ruminants like sheep and goats
Dr.
available,
Rashmi, your time is over.
and these vaccines are administered. Ma'am, just 20 minutes, please give me. I will finish them. And this vaccine is administered to 4 to 8 months old female calves. And as we know, India has the largest cattle population in the world. So vaccination of each
Kindly
female calf
one
born in the
complete
country
as
is
much
very difficult
question as
task.
possible.
And oh, okay, ma'am. Okay. This is the methodology where we just collect the blood and screen the blood for the anti-brucella antibodies, and then we do the Western blot analysis, and we confirm the presence of brucella in the blood. And this is the little survey, ma'am, which we have done here, knowledge, attitude, and practices survey, where we found there was zero awareness about brucellosis disease in the people, in the cattle keepers, and about the vaccine also. Majority of the people drink raw milk, sleep in cattle sheds, and about almost 70% of cattle keepers apply intrauterine medication with their hands. And for the precautions, one can avoid consuming raw meat
Ma'am,
and
you
pasteurized
completed your
milk,
10 minutes.
and brucellosis vaccines for animals at the right time. Okay, I'm wind up with this. Thank you, ma'am. Thank you for listening me.
Ma'am,
Thank you,
can I
thank you.
present now?
Yes, yes, sure.
Present,
Please.
ma'am.
I've put the list in the chat box. Please refer to it.
I am the outside of the list, ma'am. Please check it.
Ma'am, kindly send the list of rest of the participants.
I am next, ma'am.
Sure,
Yes.
sure, we'll be sending.
Can I start, ma'am?
Ma'am,
Yes,
kindly
ma'am.
send the list for rest of the students, rest of the participants.
Good evening, everyone. Hello. Am I audible?
Yes, you are audible.
Yes,
Good
you are.
evening, everyone. So myself, Yashpol. My today's topic for presentation is survey on awareness among the people about immunity increasing ingredients of kada. So this is my abstract. So as we all know, world was facing COVID-19 and battle is still going on. To increase the immunity, we
Not visible,
started
sir.
doing
Slide not
home
visible.
remedy.
Slide not visible, sir.
It
No,
is
no,
visible.
slides are there, ma'am. Slides are there. We can see the slides.
Is it visible, ma'am?
Yes, yes,
Yes,
yes,
yes.
sir.
Okay. So the kada includes herbs like the turmeric, which is an anti-inflammatory and antioxidants, which is there is a black pepper as it lowers the cholesterol and blood sugar level. The cinnamon, which is an antioxidant and anti-inflammatory, and also improves the insulin level of our body. So the next is the ginger, which helps to improve the digestion and has ability to reduce the nausea. So tulsi is the main ingredient that is help to fight the respiratory diseases, as the giloy was the most famous ingredient at that time, which helps to boosting the immunity. So in methodology, I have collected the data from the 100 people on the survey basis. These all are the questions I have included in my questionnaire Google form. So from the observation and results, it shows that 84% people have kada at time of pandemic. The second chart shows that people had kada twice a day. More than people, 50% people had kada twice a day. Have trust the immunity boosting kadas properties of the kada. The second chart shows that the turmeric, cloves, black pepper, cinnamon, and tulsi are one of the main and the mostly used ingredients by the respondents to increase their immunity in kada. So the study concludes that there are many medicines in the market which increases the immunity, but India defeated the COVID-19 by the Asian knowledge of Ayurvedic kada. So these all are my references. Thank you.
Can I present, ma'am?
Thank you, sir.
I've
Okay.
posted the list in the chat box.
Is it visible, ma'am?
Yes, yes. You can present now.
Okay, okay. Good evening to everyone. My name is Uday, and my presentation topic is green synthesis and characterization of silver nanoparticles from bulb extract of Virginia indica. These are the contents. This is my abstract. Let's start with the introduction. So basically the particles whose size range between 1 to 100 nanometer are called as nanoparticles. And when we synthesize these nanoparticles by using living organism, we called it is as green synthesis method. Nanoparticles can be synthesized either through green synthesis method or chemical synthesis method, in which green synthesis method is cost effective and eco-friendly method. There are numerous metals which can be synthesized as nanoparticles such as gold, silver, zinc, copper, platinum. Excessive use of antibacterial drugs has created the problems of increasing drug resistance in bacteria. This problem can be overcome by using plant-based medicines or nanomedicines. The therapeutic property of the plant is due to presence of various phytochemicals such as alkaloids, flavonoids, saponins, phenolics, etc. The plant Argemia indica is a medicinal plant which is rare endangered Indian medicinal plant, belongs to Asparaceae family. It is endemic to some regions of India and Africa. It is also known as jungli pyaj. It comprises of about 100 species, of which 9 species are found in India. My work is focused on phytochemical screening and green synthesis of silver nanoparticle from bulb extract of Argemia indica. Botanical description of plant. So basically, the Argemia indica is look like onion. As we can see in this picture, it has a globular bulb, of which lower part it has fibrous root and upper part it has long lanceolate leaf, which is green in color. Then materials and method. First one is collection and identification of plant sample. The bulb sample was collected from hills of Ranjangaon district of Chhattisgarh, which was later identified at Botanical Survey of India. Then collected bulb samples were washed with running tap water followed by distilled water to remove all dust and debris. Then bulb scales were peeled off and crushed using mortar and pestle. Extraction of bulb sample was done by softlet apparatus, in which I have prepared 50-gram bulb sample with 250 ml of different solvents such as acetone, methanol and aqueous, which were further used for phytochemical analysis. Green synthesis of silver nanoparticle was done by the aqueous extract of bulb. For this, firstly I have prepared 0.1 molar concentration of silver nitrate solution, which was mixed with aqueous plant extract in 9:1 ratio to synthesize silver nanoparticle. Then extract were performed for phytochemical analysis for all three solvents such as acetone, aqueous and methanol. Results and discussion. Phytochemical analysis showed the presence and absence of different phytochemicals for in all three solvents, as we can see in this table. Then invisible spectrum in the range of 300 to 800 nanometer, in which absorption peaks were found between 300 nanometer to 500 nanometer, where maximum absorbance peak was found at 438 nanometer, as we can see in this graph. After analysis showed the peak at 3318.32 centimeter inverse for NH stretch, 1639.18 centimeter inverse for CO stretch, and 670.27 centimeter inverse for CH stretch. Time analysis showed the particles were spherical in shape, whose size ranged between 9 to 30 nanometer. Then conclusion. The plant Argina indica is a rare endangered Indian medicinal plant, and it has various types of secondary metabolites that can help in reducing metal ion into nanoparticle. The nanoparticle can be synthesized either through green synthesis method or chemical synthesis method, in which green synthesis method is cost effective and eco-friendly method. These are the references. Thank you.
Can I present next, ma'am?
Am I audible?
Yes,
Yes,
sir.
yes,
Audible.
you're. Thank you. The next person.
And can I present, ma'am?
Yes, please present.
Hello, ma'am. Can I present, ma'am?
Yes, Mr. Ashish, you can present.
Can I present, ma'am?
After Ashish, you can present, ma'am.
Ma'am, is my screen visible or not?
Yes, sir, your screen is visible.
But only the Google Meet.
Is my PPT visible or not?
Not now. It was visible earlier. Yeah, now please, yes.
Hello.
Turn it, yes, it's visible.
My name is Ashish Verma, Department of BTech
But
Biotech. I'm a
can you
student of
please
third year.
can you please turn the slide over? मतलब your mobile phone, rotate.
Okay, ma'am.
Put your mobile in landscape mode.
My topic today I'm here to present my topic effect of sting protein in cancer. So
Mr.
now
Ashish,
we
can you present your slide in landscape mode?
okay, ma'am. I'll try.
Yeah, enable rotate on your mobile phone. Enable rotate screen.
Okay, ma'am. Okay, I'm trying. Okay, okay, okay. Nice. Okay, ma'am.
Yes, it is.
Yes, just go for slideshow mode.
Okay, ma'am.
Yes.
Now I will start by the introduction. The stu- sting, also known as the transmembrane protein 173 and MPYS, MITA, is a protein that in humans is encoded by sting one gene. In human, it's found in the chromosome number 5 and in mouse in chromosome number 8. Produced when cells are infected with the intercellular pathogens. Sting protein works as both a direct cytosolic DNA sensor and adapter protein in type 1 interferon, signaling through the different molecular mechanism. Now, I will discuss about the structure. It made up of the 379 amino acids and it has a 4 transmembrane and the C-terminal domain. And C-terminal domain, it about the 138 to 379 amino acids and contain the dimerization domain and carboxy terminal domain. Tell ma'am. The sting protein forms a symmetrical dimer in the cell. Sting dimer resembles a butterfly with a deep cleft in two protomers, ma'am, as shown in the structure for transmembrane and the dimerization domain and CTT, ma'am. Now I will talk about the functions of string. It provides the antiviral immunity, defense against the intercellular bacteria, pathogens, and role in the host immunity. Here are the mechanism of the sting. Ma'am, I'm audible, ma'am.
Yes, Ashish, you are audible. Please continue. You
Now I
have
will
only
talk.
1 minute.
Now I will talk about the sting signaling mechanism. Sting mediate the type 1 interferon immune response by the functioning as both a direct DNA sensor and a signaling adapter molecule. Upon the activation, sting stimulate the TVK first activity to phosphorylate IRF3 or STAT6. Phosphorylate IRF3 and STAT6 dimerase and then enter nucleus to stimulate the expression of gene involved in the host immune response, such as interferon B, CCL2, CCL20, etc. It works as a cytosolic tensor, signaling adapters, and others, ma'am. Now the CGS sting mechanism, ma'am. The CGS.
Limit.
Okay, ma'am. I will end after this conclusion. Studies are being carried out to evaluate the therapeutic potential of the sting agonists in the combination with the next generation cancer immunotherapies. Basic studies that will elucidate the influence of such compounds on various cell subsets they are present in the tumor microenvironment are of of research can be useful in selecting patients that would benefit the most from sting agonist treatment as well as for the development of most effective combination treatment. Thank you.
Thank you, Ashish. Next,
Should I
Dr.
present next? Yes, ma'am.
Dr. Podamekala. Are you in line, Dr.
Ma'am,
Podamekala?
can I present?
Yes,
Next.
yes, you can present now.
Ashish,
Ma'am,
please stop presenting. Ashish, your screen.
hello, ma'am. Hello, ma'am.
Yes.
Ma'am, when we are going to get the souvenirs?
Yes. Okay. You'll be getting it in the PDF form, I guess.
Okay, okay. Thank you very much.
Dr. Podamekala, you can present. Your time limit is 5 minutes. Dr.
Yes, ma'am.
Podamekala,
Yes, ma'am.
are you online? Please present
Yes,
your
ma'am.
screen.
Okay. Good evening, everyone. Are you audible?
Share your screen, ma'am.
Okay. Okay. Yes, ma'am. Today we are going to discuss the topic is immunology of infectious diseases. So, immunology is the study about the immune system, which is responsible for protecting the body against the pathogenic diseases. The immune system have two types of immunity that they are innate immunity and adaptive immunity. Pathogens or causative organisms of the infectious diseases. So now the pathogens like viruses, bacteria, fungi, and nematodes. And these pathogens enter the body through mechanical or wounds and environmental factors and cause harmful activities. When a pathogen enters the body, automatically immune system recognizes the foreign particle and responds to eliminate or kill it. So immune response depends on the type of the pathogens and location of the infection. But this response was mediated by different components of the immune system. So including WBC such as T cells and B cells and macrophages. So here T cells one type of white blood cells then can immerse and destroy the pathogen. In addition, the immune system depends on some signaling molecules such as cytokines. Here are a few examples of the importance of immunology. So, for medicine, agriculture, and biotechnology, environmental health. And now, importance of immunology. So, infectious diseases mainly. Correct. Immunology is the branch of biochemical science that focuses on the study of the immune system and how it functions in both health and diseases. The immune system plays a crucial and elimination of abnormal or cancerous cells. So, in immune diseases, the immune system is either overactive or underactive or leading to a range of disorders such as allergies, autoimmune diseases, and immunodeficiency disorders. Immunology also studies the immune system response to pathogens, how immune cells recognize and respond to infections, and the factors that can influence immune responses such as genetics, environmental factors, and age. In summary, immunology plays a crucial role in understanding immune diseases and developing therapies to prevent or treat them. By understanding the complex mechanisms of the immune system, immunologists can develop new ways to harness the immune systems to power to fight the disease and improve health outcomes. Finally, immunology of infectious disease is a complex and dynamic active research field, understanding the immune response and discovering the effective vaccines to cure different infectious diseases. So, infectious diseases are illness caused by microorganisms, virus, bacteria, fungi, and parasites, etc. So, these microorganisms can enter the body and cause a range of symptoms from mild to severe, depending on the type of infection and the individual's immune response. Some examples of infectious diseases include common cold, flu, tuberculosis, HIV, or AIDS, malaria, measles, and Ebola. So these diseases can spread from person to person through direct contact, such as airborne droplets or contaminated surfaces. So preventing the spread of infectious diseases may involve the use of antibiotics, antiviral medications, or other medications to manage symptoms and support the immune system. In some cases, hospitalization may be necessary to provide more intensive treatment and prevent the spread of the infection. So, final conclusion is infectious diseases remain a significant public health challenge worldwide. They can cause a widespread illness, disability, and death, and can have significant impact on social economic well-being. However, with advance in medical research and public health measures, many infectious diseases can now be prevented or effectively managed. It is important, it is including vaccination, practicing good hygiene, and following public health guidelines. By working together, we can continue to reduce the burden of infectious diseases.
Dr. Bodha Meghala, I think
Yes,
it's time up.
overall
Uh,
health and well-being.
please come to the conclusion. There are lots of persons are waiting. Please come to the conclusion.
Yes, ma'am.
Next, I call Gaurav Pratak.
Yes,
Patak.
ma'am.
Now you start your presentation.
Thank you, ma'am. Thank you.
Thank you, Bodha Meghala.
Dr. Podamekla, you can end your presentation.
Deputy,
Yes.
good afternoon. My topic is evaluation of antidiabetic activities of selected plants using Drosophila melanogaster as a model organism. Diabetes is caused due to deficiency of insulin and loss of insulin action, genetic factors which cause physical activities enhance and major threatened factors for development of diabetes. According to the WHO, by 2014, incidence of diabetes had risen 8.5% in incapacitating diseases with a long and protected progress. Doses, including medicinal plants, are very effective to treat the diabetes, like moricana and cyclone and almond leaves. These plants have very good phytochemicals and antioxidant activities to treat the diabetes. In all the plants have the maximum quercetin is the most important constants which found in these plants to treat many diseases like anti-inflammatory activities, anti-cancerous, neuroprotective, immunomodulatory, and anti-infective activities. We choose Drosophila as a model because Drosophila have a short life cycle, short lifespan, and is less ethical concern and easy to handle. So we use Drosophila as a model to treat the diabetes. Drosophila has approximately 75% similarity with diseases causing genes humans. Numerous studies have shown very effective to treat diabetes by the Drosophila by using the different plant extracts. We use three plant extracts mainly, moringa and almond leaves and cyclic cumini, which all are the very effective to treat the diabetes because all the plants have many phytochemicals like phytochemicals like quercetin, algic acid, or they have alkaloids, flavonoids, and saponins. Cyclic cumini is rich in anthocyanins, which are very effective to treat the diabetes, yeah, neural diseases, and other diseases. The presence of these bioactive compounds diminishes like oxidation of proteins and lipids and increases the level of antioxidant enzymes like SOD, catalase, and reduce the glucose level in Drosophila. A material method. We use Drosophila strain and the culture conditions. We grow the wild type of strain Drosophila used to study. We collected the Drosophila nearby area, and we uh, we give the standard diet like corn flour, sugar, yeast, agar agar, water to grow the Drosophilas, and we give high sugar diet to induce diabetes in Drosophila. Then media cooked slurry were poured into the bottles and vials to be allowed to solidify before transfer to adult flies. And the flies were allowed to the grown in 25 degrees Celsius with the 60% humidity for 12-hour day-night cycle. We prepare the plant extract by the standard protocols. The experimental design. A base group is there. In base group, the Drosophila flies were administered on the standard Drosophila full medium alone. In the test group, a certain amount of plant extracts were weighed and then added to the heated standard Drosophila medium. After that, different concentrations varying after 1 to 10 milligram per ml. Yeah. We use different assays to check the diabetes in Drosophila or art treatment. Is climbing assay, then longevity, we can check longevity, we can check weight of flies, and further antioxidant assay, which determine the antioxidant activity in Drosophila, and the super oxide dismutase activity determination. The discussion is the present study found that the supplement herbal plant significant prolonged mean lifespan Drosophila. There are two improve the physiological effects compared to the flies, and because of this plant extract feed, flies climbed a greater height as compared to the flies not fed with the plant extract, because the Drosophila has a negatively geotrophism, negatively gravitotrophism. In summary, antioxidative and anti-diabetic effect of Drosophila were analyzed. It showed that it is able to reduce oxidative stress, enhance the immunological responses by increasing the level of antioxidant enzymes like SOD, catalase, research, etc. Thank you. This is my references. Thanks all.
Ma'am, can I present?
Thank you.
Ma'am, can I present my presentation?
Ma'am, I am ready. Can I present, ma'am?
Ma'am, I am ready.
Anita
Yes,
Palani
Devagutta.
Is Anita Palani Manikam online?
Yes, ma'am.
No,
Yes,
ma'am.
ma'am.
Yes, you may start your presentation. Your time limit is 5 minutes.
Yes, ma'am.
After that, can I share my presentation, ma'am? Yes.
I'm asking Mr. Vismana.
Yes.
Yes, ma'am. Your screen is visible.
Thank you, ma'am. Thank you, ma'am. Good afternoon to all. My topic is comparative analysis of COVID vaccine. Nivovaxide, Moderna COVID-19, Sinopharm COVID-19, Sinovac Corona. As of March 2023, India has administered over 2.2 billion doses overall, including first, second, and precautionary booster dosing. 74% of the Indian population has received first dose, and 69% of the Indian population has received the Second, overall 80% of the people total vaccinated is more than 12 billion. Study is compare the analysis of the Covishield and Covaxin vaccine. The developer of the Covishield is Oxford AstraZeneca vaccine, manufactured under the license of the Serum Institute of India Private Limited, and Covaxin Bharat Biotech is developed in collaboration with the ICMR and NIV. And the medicinal product of the Covishield is chimpanzee adenoviral vector CHA-DOX1 and containing the SARS-CoV-2 structural surface glycoprotein antigen gene is a spike protein of the COVID-19. For a pharmaceutical form of the Covishield is off-white and pleasant and of the suspension. Then the therapeutic indication is both below above 18 years old. Then the method of the administration is two separate doses of the 0.5 ml through the intramuscular injection.
Ms. Anita, kindly come to the conclusion.
Above 77.8% of the vaccine efficacy against the symptomatic COVID-19 diseases. Through 130 confirmed cases, 63.6 production against the asymptomatic COVID-19, 12% experience in commonly known side effects, and below 0.5% of the subject feeling serious adverse events. In Covishield, 67% of the overall efficacy against the symptomatic COVID-19 after more than 14 days after the second dosage. After the second dose of the vaccine, almost all recipients developed the antibodies. Conclusion: COVID-19 vaccines are designed to induce the antibodies. It's extremely rare that health problems are found to be caused by the both of the vaccine itself. In the rare cases where a genuine adverse reaction was reported. Thank you to all.
Can I share my presentation?
Next.
And can I
Oh,
share?
okay, sure, you can.
Okay, ma'am. Yes, ma'am. My screen is visible.
Yes, it is visible.
Okay, ma'am. Good evening, everyone, to ImmunFest community and all participants. My name is David Vasni from Agra, School of Life Science, Khandari campus, Dr. Bhimar Uttar Pradesh University. So, without wasting my time, I'm started. So, this is my abstract, and after that, this is my introduction. We all know about the tuberculosis. Tuberculosis is called by mycobacterium tuberculosis, which is a major public health problem that affects the people all over the world. Mycobacterium tuberculosis, the bacterium that cause the tuberculosis, typically lungs, result the pulmonary tuberculosis. They are airborne bacteria that can spread from one person to another. After that, exosomes are the 30 to 100 nanometer vesicles that is rapid and responders secrete early during simulatory and or microenvironment changes by most cells, including B cells, macrophage, dendritic cells, Schwann cells, and are found in many fluids, using and fluid, including blood, urine. After that, microRNA, the family of small non-coding RNA, involves an important role in regulators of gene expression. Uh, in recent years, increasing the role of miRNA as a diagnostic biomarkers has been reported by the several studies. Plasma-derived exosomal mRNA has been investigated as a potential diagnostic biomarkers for the various types of cancers: epithelial, ovarian cancer, non-small cell lung cancer, and cholangiocarcinoma and gallbladder carcinoma, hepatocellular carcinoma, cancer, lung cancer, breast cancer. This is my objective to investigate the expression profile of candidate mRNA in serum exosomes of patients with pulmonary TB, healthy human using real-time PCR. They study the role of deficiency expert mRNA in pathogenic of tuberculosis using in silico analysis method. Material, we collected the serum from the following steps, and after that, we extract the exosomes by the SBI system bioscience kit by using these methods. Isolation of isolation of exosomes by this method, exo quick solution. This is the mercury LNA plate. With the help of this plate, we find out the mRNAs, MIs and his positions. And next, the expressions. Expressions, we check the expression MI script small nuclear RNA, U6SN RNA, SNOT 388 and SNOT 49, the spike control targets of the mRNAs. And this identification of protective sequence method, these all are uh, these all bioinformatics analysis occurred by the MI work and
Come to the conclusion.
yes, ma'am, and Sinigo. These are the results of the healthy control and the PTB, PCR. After that, these are the markers CD63 and 29s. Protein markers. After this, these are the result. HSA MI 7 5P is the upregulated and all are the down regulated. That means HSA MIR 7 5P is the more efficient. So this is a graph, volcano plot. In the end, identification of the mRNA by the these biological pathway. These are the find in the plasma membrane boundary neurogen projection, cell projection, neuron projection development, cell morphogenesis, and etc. etc. After the conclusion, the present study identified the several differentially expressed mRNA in serum exosomes of pulmonary TB patients and highlights that HSA-LAT7 will have ability to distinguish the significant active TB from the healthy individuals. The findings present in this study primarily based on the limited number of clinical samples. More confined research needed to be present findings before making any final conclusion. Thank you. And I am last end. I am working on the working on tuberculosis stigmatitis. Uh, and this study conducted in the ICMR lab NJL in OND, Tajganj, Agra. Thank you.
Thank you, Mr. Gupta.
Ma'am, can I present?
M- Ma'am, can I
Ma'am,
present?
can I present?
Myself is
Myself
Krista.
is the
Next, Mr. Kushwan Nandan. You may present. Mr. Kushwan Nandan.
Yes, ma'am, presenting, ma'am.
You may present.
Hello. Good evening, everyone. My name is Kushwan, and I am presenting my presentation on the topic the complex role of the gut microbiome in inflammatory bowel disease and immune homeostasis. Inflammatory bowel disease is a chronic inflammatory disorder of the gastrointestinal tract that includes two main subtypes, Crohn's disease and ulcerative colitis. These conditions can cause symptoms such as abdominal pain, diarrhea, rectal bleeding, and weight loss. The exact cause of IBD is not fully understood, but it is believed to involve a complex interplay between genetic, environmental, and immune system. Overview of the gut microbiome is a complex ecosystem of microorganisms that inhabit the human gastrointestinal tract. It play a crucial role in human health and disease by performing various functions such as aiding digestion, producing vitamins and other essential nutrients, and regulating the immune system. The gut microbiome is also involved in developing and progressing many diseases, including inflammatory bowel disease, obesity, and diabetes. Understanding the gut microbiome comparison function is important for developing new therapies and intervention to improve the human health. Factors that influence the gut microbiome, including diet, genetics, and environment. Diet, the type and amount of food that a person eats can affect the composition and diversity of the gut microbiome. For example, a diet high in fiber can promote the growth of beneficial bacteria in the gut. Genetics. A person's genetic makeup can also influence the composition of their gut microbiome. So they have shown that certain genetic variation can be associated with a specific microbiome profiles. Environment factors. Such as exposure to antibiotics, pollution, and stress can also affect the gut microbiome. For example, antibiotics can disrupt the dysbiosis and its role in IBD pathogenesis. Dysbiosis refers to an imbalance in the composition and diversity of the gut microbiome. It is believed to play a role in pathogenesis of inflammatory bowel disease by contributing to chronic inflammation and tissue damage in the gut. This process can lead to a decrease in beneficial bacteria and increase in harmful bacteria, which can trigger an immune response and promote the development of IBD. Studies have shown that IBD patients have different gut microbiome composition compared to healthy individuals with decreased diversity and increased pro-inflammatory bacteria. Therefore, targeting these biases may be a potential therapeutic strategy for IBD. Metabolic diversity and its association with specific microbiome in IBD patients. There are there are differences in metabolite between IBD patient and non-IBD control due to the IBD microbiome. Different computational methods were used to characterize the IBD
Krishnan,
microbiome and
kindly
their
conclude your presentation. Krishnan, kindly conclude your presentation, please. For the time consuming.
I will come to the therapeutic to modify by the gut microbiome in IBD. By the probiotics, prebiotics, antibiotics, fecal microbiota transplants, or by the anti-inflammation or other therapies.
Thank you. Thank you. Next, Mamta. Mamta Bayal, you can start your presentation. Mamta Bayal. Devra. Yes, Devraj speaking prepared. Devraj, kindly mute yourself. Devraj, Devraj, kindly mute yourself. Next, Mamta is presenting his presentation, poster presentation. Mamta, Mamta Bayal. Yes, you have only 4 minutes. Kindly make it fast.
Remaining participants for presentation.
Is Mamta Bayal online? Okay,
Can I
Miss
present, ma'am? Archana.
Miss Isai Madhivanan, are you online?
Yes, ma'am. I'm here.
Yes, yes, ma'am. You can start your presentation.
Okay. Very good evening to everyone present here. I'm here to present a review on advances in molecular epidemiology of infectious diseases. This is the gist of my presentation. And let me give a definition of molecular epidemiology. Molecular epidemiology is a discipline of diseases in human populations and veterinary animals. Our understanding of epidemiology of infectious diseases has evolved with the technological advancement made in molecular biology that refine our perceptions of the identity and dynamics of microorganisms. According to Mosner and Bond, they defined that the study of the distribution and determinants of disease, and injuries in human and non-human animal population using molecular microbiology methods. These molecular epidemiology studies can be descriptive or analytical. In general, infectious disease epidemiology deals with the descriptively and quantitatively characterizing the following concern. Number one, disease concern and distribution. Number two, the reservoir of infectious agent. Number three, modes and patterns of disease transmission. Number four, the setting of disease transmission. Number five, pathogen related biological factors that influence transmission. Number five, host related factors such as demographic, behavioral, clinical, genetic, and microbiota that also influence transmission. Number seven, environmental factors such as socioeconomic, anthropologic, and ecological factors. And the final one is assessing the etiological role of a microbe that to a newly recognized diseases or a disease previously not recognized to the associated with an infectious agents. And this slide shows the differentiating molecular epidemiology from taxonomy, phylogenetics, and molecular evolution. In infectious disease molecular epidemiology, the genotyped microbes are also compared to other microbes such as microbe A and microbe B, but the microbial relationship data are linked to the population of host from which these microbes are isolated from the particular
Madam,
environment
can we come to the
or
conclusion?
transmitted. Yes.
It is not moving, ma'am.
Yes, ma'am. Yes, ma'am. Um, and what are the ways to address the molecular epidemiology? Number one is nucleic acid sequencing, then electrophoretic band pattern, and later will be introduced PCR technology. And coming to conclusion, molecular epidemiology is now an established branch of epidemiology. As with the other tools such as biostatistics, molecular epidemiology evolves constantly as a new analytical and quantitative tools are developed. Each step of technological advancement contributes to further refinement and new ways to conduct analytical epidemiological investigations. Thank you.
Archana Verma, thank you so much. Next, start பண்ணுமா? Archana Verma, you can start now. Archana Verma is here. Archana Verma.
Archana,
Archana
Archana
Verma is
ma'am.
here. Okay, no problem. Dr. Archana Gupta is here. You
Yes,
can
ma'am.
start
Yes,
your
ma'am.
presentation, please.
Okay, ma'am.
Gupta.
Okay, ma'am. Okay. Yes, ma'am.
Yeah, you can start it now.
Okay, ma'am.
No, it's not
Okay, ma'am.
visible.
Thank you. I'm Dr. Archana Gupta, Assistant Professor in
No, your
Department of
presentation is
Zoology and
not.
Paleontology.
Your presentation is not visible, Archana Gupta.
Is it not visible? Okay, ma'am. Let me try. Okay, okay.
Okay, next Akshaya is here. Akshaya, dear Akshaya, you can start your presentation now, Akshaya. Archana Gupta, just wait. After Akshaya, you can start.
Okay, okay, ma'am.
Now Akshaya is presenting the poster presentation.
I am D. Akshaya. The topic of the presentation is a study on anti-cancer activity of Pharmatroma perlatum lichen with liver cancer HG2 cell line. Lichens are abundant growing on park leaves, mosses, on other lichens, and hanging from branches, living on thin air. In rainforest and in temperature, outside solid rock growing between the grains. It estimated that 6% of Earth's land surface is covered by lichens. Of lichen used as a spice in India. They are also used in vegetarian dishes. It is the fungal and where they have a very light fluffy texture. The lichen was used for antibiotic, antioxidant, anti-ulcer, antimicrobial, antifungal, and anti-cancer activities, etc. Liver cancer. Liver cancer is a cancer that originates in the liver. Aggressive tumor in frequently and the seventh most common cancer in females is the third leading cause of cancer related to death worldwide. Objectives. The present study. Ma'am? Ma'am, I am audible?
Yes, you are audible, ma'am.
Yes, ma'am. Thank you, ma'am. To collect Formoterma perlatum from Ayurvedic shop, to prepare ethanolic, methanolic, and aqueous extract by study of qualitative analysis, to prepare methanolic extract by study of antioxidant activity, the methanolic extract by study of GCMS analysis, in vitro anti-cancer activity of selected lichen Formoterma perlatum using liver Hepg2 cancer cell line. Materials and carbon method, antioxidant activity done by DBPH method, GCMS analysis, cytotoxicity study, MTTSA. Result and discussion. Phytochemical analysis. The present study was carried out on the Formoterma perlatum lichen extract for the presence of medicinally active constituents. Phytochemical characters are investigated by alkaloids, flavonoids, phenols, saponins, etc. Antioxidant activity. The present study will have evaluated free radical scavenger activity of Formoterma perlatum lichen, presence of high amount of phenols. A concentration dependent scavenging activity was observed in methanolic extract of concentration of the lichen had relatively higher reducing power than other lichen due to the presence of high phenolic compounds. The present study examined and revealed active compounds in the study was observed dose-dependent manner when treated with various concentrations. The maximum cytotoxicity 90% was observed in Hepg2 cell line, treated with 390 mg/ml of extract, whereas 90% of cell viability was observed in Hepg2 cell line, treated with 390 mg/ml of extract. The following reports supported the present study. The Formoterma perlatum bioactive compounds which might disturb the mitochondrial assembly, which resulted in the increased cytotoxicity of Hg2 cell line. Photomicrograph 20x represent morphological changes in Hg2 cells, such as shrinkage, detachment, membrane clubbing, and a disordered shape induced by sample Formoterma perlatum treated. Compared with the control. Control showed a normal intact cell morphology that imaged by light microscope. This is the image of cell viability of Hepg2 cell line. Cell viability concentration of different concentration level. Summary and conclusion. The present study shows the Formoterma perlatum has increasing high nutritional value and most antioxidant activity of compound is present against cancer diseases. Ayurveda and Siddha medicine has been recommending of Formoterma perlatum plant for centuries to treat specific ailments. The lichen is used to alone some areas. It is mixed with other species in various places, thus used as some domestic spice. The phytochemical analysis was carried out on the Formoterma perlatum lichen extract revealed the presence of medicinally active constituents. The nutritional potential, especially because this can be used as a food source, given the protein lipid contents using Formoterma perlatum lichen spices, results indicate that probably the higher amount of phenolic compounds in the lichen extract are responsible for encouraging antioxidant properties. Resulted indicated that probably the higher amount of phenolic compounds in the lichen extract are responsible for encouraging antioxidant property. The lichen was playing an important role in health and disease prevention. This important lichen aspects such as medicine, foods for commercially purpose, inhibitory effect on various cancer cell lines, the mechanism of liver anti-cancer action.
Akshaya,
Yes,
this
sir.
is Dinesh. I will be handling this session hereafter. So, can you go to your conclusion and results and conclusion section? It's almost 3 minutes over.
Okay, sir.
Can you make it faster?
Hence, the
Because other
selected
participants are waiting.
Yes, sir.
Okay, yeah.
Hence, the selected Pharmochroma bellatum lichen possesses significant anti-cancer activity against liver cancer. The further study will be carried out for isolation of bioactive compounds which responsible for liver cancer pharmaceutical application. That is all. Thank you, sir.
Thank you so much. Uh, yeah, yeah, you can start. Please limit your presentation for 3 minutes only. Okay, there are so many participants waiting. Yeah, you can start.
Is my slide visible, sir?
No, no, it's not visible.
Hello.
It's not visible. Your
Sir,
slide.
is my slide visible, Dr. Archana Gupta?
Actually, please mute your. Yeah. Dr. Archana Gupta, are you there? I think. Okay. The next one, Durga Devi. Can you? Oh, sorry, sorry. Archana Gupta is still there.
Yes, sir.
Let her complete.
Sir.
Go to presenting mode. Like, share your screen. Archana Gupta, are you there?
Yes, sir. Yes, I'm trying to present. I'm trying to share my screen, sir.
Shall we move to other participants so that you can start later on? Is it okay for you?
So is my slide visible?
No, no, madam. It's not visible.
Okay, sir. Okay, sir.
Okay, let me go to
Good
Durga.
afternoon, sir. Shall I? I am Dr. Viranagendra Kumar. Shall I present my?
Okay, okay. Carry on, carry on. Yeah, you can.
Thank you, sir. Thanks. Thank you a lot.
Welcome. Please limit your presentation for 3 minutes.
Okay, okay, sir.
Okay. Yeah, you can go on.
Sir, it is visible my slides.
Yeah, yeah, it's visible. Go
Thank,
ahead.
thank, thank you, sir. First of all, thanks to Department of Biotechnology, PRIST University, for
Yeah.
giving this opportunity.
Welcome.
Now, I am presenting my research topic: phytochemical screening and antibacterial activity of Phyllanthus neruri in methanolic and ethanolic and aqueous extract. The medicinal plants represents an enormous reservoir of potential phytochemical compounds. They could be useful as an alternative and to allopathic drugs are being used to develop pharma drug. In this study, we are using the Phyllanthus neruri. It is belongs to Euphorbiaceae. This plant's pharmacological properties are gastrointestinal disturbances, so it prevents the cough and fever and malaria and like that. In this study, we take two studies. One is phytochemical analysis, and second one is antimicrobial property. We are using for the phytochemical screening in this brain study three different solvents. One is ethanol, another one is methanol, and the next one is water. These three extractions. For this phytochemical staining, we are testing the alkaloids, tannins, saponins, quinines, and reducing sugars and resins also. And next study is antibacterial activity. Uh, for the antimicrobial activity, we have chosen the five bacterial strains: Staphylococcus aureus, Bacillus subtilis, Pseudomonas aeruginosa, and Bacillus cereus. Uh, Antimicrobial spectrum of different extracts of various concentration by bacterial zone of inhibition. The maximum zone of inhibition activity is 30. We are using the standard drug than the streptomycin. In this slide, we observed in ethanolic extract all these phytochemicals are present. In methylation extract, except anthraquinones, remaining all phytochemicals are present. If you take aqueous extract, anthraquinones and resins are absent. Remaining all phytochemicals are present in aqueous extract. This is the first study. The second one is antimicrobial activity of Phyllanthus neruri, different solvent extract. One is methanolic, another one is ethanolic, and third one is aqueous extract. In the Antimicrobial activity Phyllanthus neruri alkaloids which are responsible for the strong antiviral activities. This antibacterial activity of Phyllanthus use the ligands, filamentous, hypophyretin flavonoids, and titernite glycosides and tannins in the plant extract. Finally, the higher activity of the methanol extract may be due to higher solubility of the active compound in their solvent. Methanol had a higher power to extract the active bacterial compounds. In our study, two extract methanolic and aqueous extract showed better inhibitory effect of Salmonella aureus. The ethanolic extract produced better inhibitory effect of Escherichia coli rather than Salmonella aureus. Staphylococcus aureus, sorry. So, the finally we conclude from the results we obtained Hence, quantitative estimation of total phenol, tannin, and flavonoid. These results suggested that ethanolic, methanolic, and aqueous extract are used for the isolation of novel bioactive compounds in econo-medical development potential drugs. Different concentration of ethanolic, methanolic, and water extracts of Phyllanthus neruri possess antibacterial activity. Finally, it may be used as an antibacterial agents. Thank you, sir.
Thank you. Thank you, sir.
Sir,
The
shall
next,
it start, sir?
yeah, go ahead. Fine.
Sir?
Yeah, you can go
Can
ahead.
it? Okay, sir.
Yeah, it's visible. Your slides are visible. You can go ahead.
Sir, is my slides visible, sir?
Yeah,
Mr.
yeah, it's
Archana
visible.
Gupta.
It's visible. Go ahead. Go ahead.
I'm Dr. Archana Gupta, Assistant Professor, Department of Zoology, MDBM College, Muzaffarpur, Bihar. My topic is a review topic, helminth and immune responses.
Uday. Good evening. Baunava.
And, so, helminths are the worms which are
చేయట్లేదే ఉదయ్ అని నేను మళ్ళీ డిస్టర్బ్
medically very important because they
తిరుపతి
cause
విన్నావా
disease in different
ఏమంట
organisms, including the human. Basically, helminths are nematodes, fatty helminths, and there is a
తెలుసు
strong
మన
equilibrium
జాకోబ్ సార్
between the host and the helminth parasites and
Sorry for the
enzymes.
interference. Sorry. Someone is not muted. So, Veer Nahan. Yeah. Now it's okay. You can go.
So, shall I continue?
Yeah, you can continue. Yeah.
Yes, so can I continue as well as the immune evasion of the parasite? The immune responses of the host to helminth infection are generally characterized by TH2-like responses. Helminth develops several means of escaping these immune responses, known as immunomodulation, and this immunomodulatory form lead to interaction in inflammatory and immune mechanisms and other vaccines and allergic autoimmune reactions. The host produces several effective mechanisms against helminths and also their regulation. And basically, these responses involve TH2 response as well as T regulatory cell responses. And basically, these responses depends upon the localization of the element. So accordingly, effective mechanism against tissue-dwelling parasites, effective mechanism of localization of human objects, and their regulation is in the effective mechanism against the tissue-dwelling parasites and escape mechanism. The first diagram shows the effective mechanism of host where two main immune mechanisms are directed, like during early phase TH1 activity is shown, which induce nitric oxide production, which is toxic for food, and during chronic infection, release of intermediates, which causes TH2 type regulation, anti-regulatory cells, which include several cytokines, and these all causes the host responses. And then there are some escape mechanisms which is done by the parasite, like radicals, and then it cleaves the IgE and IgG, which is involved in ADCs, so that the host responses not so effective, and also it produces the blocking of antibodies IgM and IgG. Similarly, effector mechanisms in human, especially the larva forms, also show they are involved in blockage of their migraine. Immunoglobulin A also neutralizes the metabolic activities which interferes with the worm. Then there are several regulatory immune responses also which are involved. It's protective and immunological effects of the immune response against the habits are present despite the long infection of active immunity present, pre-immune and partial immunity. In pre-immune, the host is prevented from the reinfection by the same species, but in case of fistula, it is seen that it doesn't prevent the infection from different species, and also it causes the partial explosion of the adult worms. So it causes partial immunity also.
Dr. Archana, are you here?
Yes, sir. Yes, sir.
Sir, your time is up, so please make it fast.
Okay, sir.
The time is 3 minutes only. Okay, so
So,
please go to your conclusion section.
yes, sir. So, Helminth and co-infection, they are immunomodulatory okay, activity which is Th2 and T regulatory type of cells, and it causes several studies have shown that Helminth can influence vaccine by modulating the host immune responses. Immunomodulatory functions of helminths and the products can also be used in anti-inflammatory drugs. Like Trichuris Suis has been recently tested to treat patients with inflammatory bowel disease and Crohn's disease with success. So, in conclusion, alien species have co-evolved with the host for a long time, and this involves Th2 type response as well as the T-regulated types of responses. And helminths are strong immunomodulators and interfere with immune and inflammatory mechanisms induced by themselves and co-infecting pathogens, inflammatory disorder, and vaccines. Immunomodulatory products from helminths are probably the anti-inflammatory molecules of the future. The effect of helminths on host immune system are not properly known, so much more studies in this direction are needed. Thank you.
Thank you, Archana. The next,
Sir,
Durga.
shall I start, sir?
Yeah, you can go on.
Okay, sir.
Yeah, please limit your presentation for 3 minutes. Okay, it's very tough to limit your
Sir, can I,
entire.
sir, sir, can I present next, sir?
I have already mentioned
Sir, I'm
presented.
waiting for a long time, sir.
Sir, shall I
Okay, let
start,
her
sir?
finish. Let her finish. You can move on to you.
Sir,
Yeah,
mushroom pleurotus oysters using beetroot as substrate in span production. Introduction is a mushroom is a high nutritional food and secondary food. It contains the high nutrients, and most of the pigmented mushrooms are implementing the vegetables, nutrients, and pigmentation properties in mushroom, and also to increase the hemoglobin content. The beetroot has chosen as a substrate in span production to produce a pigmented mushroom. Beetroot contains nutrients and calories. It reduces blood pressure. It can improve athletic
sorry,
performance. It
sorry,
may fight inflammation.
sorry to disturb you, Durga. Your slides are not visible.
Sir?
Your slides are not visible.
Okay, sir.
Yeah, please make sure that
Okay, sir. One నిమిషం, sir. Sir, it's sir. Sir?
it's not visible.
1 minute, sir. 1 minute, sir. Is it
Yeah,
visible, sir?
no, not at
Oh, and then click the slide and then share.
Yeah, present now. There is an option. You could see present. Yeah, yeah. Now it's visible.
Okay, sir.
Go ahead. Go ahead.
Sir, start பண்ணுமா சார்?
Yeah, yeah, you can start.
This is the picture of pigmented mushroom. Objective, the main aim of this study is effect on a comparative study of normal and pigmented mushroom on yield, nutritional and antioxidant activity. Hence, the present study has been aimed to mad investigation with the following objectives. Identification and cultivation of Purotus oysters, mushroom of the same species, to study the yield and production of pigmented mushroom, to study nutritional analysis and antioxidant activity of the Purotus. Materials and methods. Collection of sample, mycelium culture preparation, span preparation, nutrition analysis, antioxidant activity. Result. Effect of pigmented mushroom on the cultivation parameters. The highest growth rate was observed on pigmented mushroom. Nutrition analysis. Estimation of crude protein by Lowry's method. Protein content of the most mushroom have a high protein content, usually around 20 to 30%. Dry weight. The pigmented mushroom has increased content of the crude protein or present compared with normal mushroom. This is the graph diagram of protein present in the both pigmented mushroom and the normal mushroom. Carbohydrate. Total carbohydrate were observed to be highest values on the pigmented mushroom, or the lowest values were obtained from the normal mushroom. This is the graphical diagram of carbohydrate present in the both mushrooms. Lipids. Lipid contain normal mushroom increasing range comparing to pigmented mushroom is low concentration processing. It is the graphical diagram of lipids present in the both mushrooms. Minerals. In my study, macro elements, sodium, potassium, magnesium, and calcium, and the microelements, manganese, Fe, zinc, and copper, were determined highly in pigmented mushroom compared with the normal mushroom. Graphical diagram of minerals present in both mushrooms. Antioxidant activity. The antioxidant activity in the fluorescers extract were evaluated by determining the IC50 values of DPPH radical. Lower IC50 activity, the highest IC50 values of the study, normal mushroom was obtained, while the lowest IC50 values represent the higher antioxidant activity were obtained for pigmented mushroom compared with the normal mushroom. Grown in the padista and the mushroom grows very well, and weight is 0.258 gram. The well-grown oyster mushroom taken and cut the internal tissue and put it into the mycelium culture. In that culture, the mycelium was grown well. The mycelium was inserted into the sterilized beetroot beads and kept in for incubation. After a few days, the pigmented spawn were produced in the spawn are used for the pigmented mushroom cultivation, in which padista acts as substrate. The pigmented mushroom appropriate are very good analysis in pigmented mushroom. However, also a good and high contents. Antioxidant test shows that the OD value than oyster mushroom. Summary and conclusion. Usually non-veg foods like fish, meat are rich in protein and nutrients. Non-veg eatable people should take the protein and nutrients, but vegetarian people are not able to eat. And in olden days, the mushroom are not ready to taken by the people, but nowadays everybody takes the food. In the study, normal edible mushroom are common one, but the study shows the vegetables beetroot also produces spawn and the grow in the mushroom. Normally, the spawn are produced from the corn residue, but the stores that the edible vegetables are produce the colorful mushroom. The
Thank you.
mushroom, pigmented mushroom red color are attractive to children and they are eagerly eat. Also, beetroot pigmented mushroom are rich in hemoglobin. It is also good for women's health. It is not only possible in beetroot, possible in all vegetables. In my study, the edible vegetable beetroot changes its form and produces a colorful healthy food. Research can be shown that pigmented mushroom is a good source of health.
Thank you, Durga.
Thank you, sir. Thank you.
Next,
Sir, shall I present, sir?
yeah, go ahead.
Thank you.
Sir, after Durga Devi, I am in the queue.
Sarit, hello.
Yes, sir.
Am I audible?
Yes, sir. Audible.
Yeah. I think as per I
Myself,
mentioned, you are
Subhasis Das.
okay. Yeah. You can go ahead.
Okay, sir. Thank you.
Yeah, you can go. And Dr. Santosh Kumar, please wait. Let me call you upon once these people finish their presentation. I think Das and Swati. After that, you can present.
Okay, sir.
Good evening, sir.
Yeah,
Today
you
is my
can. Please keep in slideshow mode. Okay.
okay, sir. Today my topic is about comparative analysis of antimicrobial activity of wheatgrass extract against drug-resistant bacteria. Nowadays, here is the content: introduction, review of literature, aim of the study, plan of work, and future prospectus, along with references. Nowadays, the herbal medicines are more popular because they have no side effect, and if we take antibiotic, then it should have also cure the pathogens which are helpful, and also the disease causing pathogens both are death by the antibiotic. So the natural product nowadays are more popular. The wheatgrass should have laxative, antioxidant, diuretic, astringent, and antibacterial property. So the for antimicrobial property, mostly the flavonoid should responsible for this as per the several literature survey. Then the objective, we should find the antimicrobial activity and the phytochemical enrichment fraction of wheatgrass. The working, first we should grow the wheatgrass, then the development and growth of this, then we should prepare the different extract, then phytochemical analysis and perform qualitative and quantitative test for phytoconstituent present, and then we should find the maximum zone of inhibition. The from the shop, I should collect the wheatgrass seed, and then it should be germinated for 7 days. In soil, the germination should be high, and the yield is also maximum. Then several type of extract we should prepare, and the extract is mostly acidic in nature. As a result, different type of test from Mayer's reagent and foam test, several phytoconstituents are found. After that, the aluminum chloride colorimetric method, from which we should find the result of the detection of flavonoid maximum number. Here is the result for uh, flavonoid detection in 540 nanometer of UV. Then we should check on this on TLC plate, and on TLC plate we should consider as juice, wheatgrass juice, ethanolic extract, acetone extract, and chloroform extract, along with petroleum ether extract. On wheatgrass juice and the ethanolic extract, the separation is better, and the presence of flavonoid we should found. Then we should perform the preparative TLC, and we collect the scrap. This is the preparative TLC, and after that several types of color changes occurs, from which we should collect the scrap, and then uh, UV analytical method performed. This is the color graph, and we should found the several types of peak. In light green we should found more number of flavonoids. Then this should be my future plan of work. After that, we will perform the HPTLC and the antimicrobial assay. Here is the reference. Thank you, sir.
Thank you, Das. Next presenter is Swati.
Sir, silent.
Yes, sir. I will present.
Yeah, you can go ahead. Swati, are you there?
Sir, my slides are visible?
Yeah, keep it in slideshow mode. The slides are visible, but keep it in. Yeah, fine.
Okay, sir.
Go ahead.
So good evening to all. My presentation title is Evaluation of Antioxidant Activity of Cysium cumini in Parkinson disease using Drosophila as model organism. So what is Parkinson disease? It is a multifactorial disorder that includes both the factors like genetic and environmental, which affect the multiple signaling pathway in different cell types, and the major oxidative stress and aging cause the neuroinflammation that involves so many factors like interleukin 6, 1, beta, nitric oxide, TNF alpha. TLR4 is mainly responsible for the deposition of alpha synuclein, a protein that are accumulated in the brain cells between the two neurons actually. According to World Health Organization, 80% population relies on herbal or medicinal plants to cure their disease as they contain bioactive compounds to elevate the level of antioxidant enzymes. And why we choose a Drosophila over mammalian model? Due to their low cost, small size, easy quantifiable phenotypes, and it shows 75% disease related conserved genes which are homologous to our humans. Genetic manipulation is easy. It is also used to check the efficacy of drugs. Now, next is my plant name is Cysium cumini, which is rich in antioxidants, and which is the seed contain glucose, malic acid, quercetin, myricetin, flavonoids, anthocyanin, polyphenols, tannins, and so many compounds. The presence of these compounds diminish the oxidation of protein and lipid, and increase the level of antioxidant enzymes for the betterment of transmission of nerve impulse. Although the role of Cysium cumini against neurodegeneration is not well understood, so we investigate the supplementation of Cysium cumini extract to the Drosophila against neurodegeneration and against neurodegenerative diseases. So material and methods. First, we take a wild type strain of Drosophila, which is collected from nearby nursery through net sweeping method and identified. After that, we prepare the standard diet of Drosophila, which consists of corn flour, sugar, yeast, agar-agar, and water. To prevent the bacterial growth and fungicidal, we add sodium benzoate and propanoic acid. After that, the food was prepared and it is poured into the bottles and was allowed to solidify before transfer to adult flies. Next is preparation of Cysium cumini extract. White test plant was collected and the seeds were grounded and aqueous extract was prepared. Now we design the two groups, base group and text group. Base group, in which Drosophila was administered only on standard Drosophila food media, while in text group, plant extract Drosophila media. Longevity, to check the antioxidant effect of plant on the lifespan of adult fly, was studied by feeding the DM flies to normal food and the food mixed with plant extract. Next is fecundity. Fecundity is also used to check the antioxidant response. We place a 6 days old female and male in a one vial for around 12 hours or 24 hours, and after that male was removed. Now the eggs lay on the food placed at the bottom of the oviposition chamber were counted, and then it was transferred daily into the new chamber, and this phenomenon is repeated for 15 days, and it is a mean time for the laying of eggs in Drosophila. Climbing assay next is to access the antioxidant effect of plant extract. So we generally tap the flies to the bottom of the vial to stimulate a negative response, and the height of fly was measured. Next is viability. Viability is the percentage of surviving pupa in F1 generation. So we place 3 pair of male and female into a vial containing standard food along with different concentrations of plant extract, and the number of surviving pupae was calculated per vial. Next is we give the exposure of paraquat so that relish get activated and translocated in the nucleus, which cause the decreasement in antimicrobial peptides, which increases the sensitivity to bacterial infection and reduce survival, mobility defect, and loss of dopaminergic neurons. So Cysium cumini is one of the plants that are able to combat these types of defects. Next is results. Effects of Cysium cumini on longevity, fecundity, and viability. As you see here, the concentration 7 mg per ml is the best suitable for their fecundity, for their lifespan, and for their viability. Next, we perform an assay, superoxide dismutase activity, which is an
Swati,
Hanji sir.
can you go to your conclusion section?
Yeah,
It's
sure.
almost
Okay, sir, sure. This is my discussion. Last slide. The present study found that Cyzum cuminis prolong the lifespan and the beneficial effect are due to increased SOD activity and the protein content get decreased. Present study found that fly supplemented show improved physiological effect which were not given to plant extract. It climb a greater height. It show it is helpful to reduce oxidative stress and enhance the immunological responses by increasing the level of antioxidant enzyme like SOD catalase. These are my references. Thank you so much.
Sir, shall I present it next, sir?
Actually, Dr. Santosh Kumar. Are you there, sir? Dr. Santosh Kumar? I think he's not available. Yeah, you can go ahead now.
Thank you, sir.
Yes.
Sir, in the
Yeah, your slides are visible. Yeah, you can go ahead.
I'm Hemamali, Department of Biotechnology, Bonsex College. Progressive neurological disorder affecting patients non-graft over the world. The Parkinson's disease is chronic degenerative disorder of the central nervous system that mainly affect the motor system. The symptoms usually emerge slowly as the disease awareness non-motor system becomes more common. The symptoms of Parkinson's disease: tremor, rigidity, slow of movement, difficulty with walking problems, also arising in the behavior, sleep, sensory system. The dementia becomes common in advanced stages of Parkinson's disease. The Parkinson's disease other name is idiopathic primary Parkinson hypokinetic disorder syndrome, paralysis shaking palsy. The illustration of the Parkinson's disease shows head injuries. Mirror should be consumed on a daily basis. The health benefits: enhance cellular activity. It is used to treat paralysis, relieving joint pain, improve your immune system, manage your weight level, enhance hemoglobin level, boost system. The color and quality: the rice white color in which no fine quality. The height 1 centimeter. The crop duration is 1 or 4 days. The normal cultivated disease Samba July 15 in January 14. Ilpungu Samba is highly cropped. Aromatic substance improve the efficiency of body major organs. The objectives: the collection of traditional rice Ilpungu Samba, the analyze the cooking of traditional rice, to isolate from analyze the compound disease, to examine the Selvine. Materials and method: the collection of materials. The rice is Samba. Organic varieties are collected. Organic land cultivated Karur in Tamil Nadu. The Ilpum Samba brown in white color. The slide was obtained body turning in room temperature and end of the analysis. The estimation of carbohydrates. The estimation of protein Lowry's method. The conduct the compound of nutrition present in Ilpumbu Samba. Following result in observed. So in table and figure, the carbohydrate content in Ilpumbu Samba traditional rice 60 to 40 gram. The protein content in Ilpumbu Samba rice is 6.47 gram. Next one, cooking properties. The cooking properties Ilpumbu Samba rice can take optimum cooking time 1.15 minutes. The Ilpumbu Samba slender rice took minimum time for water uptake ratio Ilpumbu Samba is 1.5. Yes, not bold rice. The elongation ratio of height about 1.6 because it's slender rice. The Ilpumbu Samba has low yes and breadth, and because slightly round comfort. The barrel loss is Ilpumbu Samba 3% of contains high starch. Next, examine of L-DOPA by thin layer chromatography. The yellow color developed by thin layer chromatography.
Can you go to your conclusion section? It's almost 3 minutes is over.
Okay, sir. Okay.
Yeah.
The RF value 0.7 similar to standard, and unless of bioreactive compound of GCMS, the table information. Bioreactive compound present in traditional rice Ilpumbu Samba. Bioreactive compounds taken from Biotechnology IDH compounds, cytotoxic entity aside. The antioxidant compound and anti-cancer active were present in Ilpum summarized in GCMS, so the sample poses significant cytotoxic activity relying in the low concentration. Summary and conclusion. So, the rice is one of the most important variety of rice grown commercially in the world. It is the staple food of the 3 billion people, mainly the Asia. It is the food more than 3 contain flavonoids and phenolic compounds, inhibit movement, detection by cell damaging, free radical development, and Parkinson disease. Thank you. Thanks to all.
Thank you. Thank you. Dr. Santosh Kumar, are you there? Dr. Santosh Kumar?
Sir, can I proceed, sir?
Yeah, you can go ahead.
One second, sir. So, visual, sir, now?
Yeah, it's visible. You can go ahead. You can proceed.
Thank you so much, sir. So,
Welcome.
today my topic is the role of diagnostic and antibiomedical studies in the implementation of the rapid molecular infection disease diagnostics. Abstract. New rapid molecular diagnostics are technology for the infection the disease of the all of the more accurate and the microbiological diagnosis to be made very quickly. However, the diagnostics of the antibiotical strategies that require the ensure that these technologies are conserved rather than they consume the additional healthcare as a resource of have the greatest possible impact on the patient care. The diagnostic service that require the implement the appreciate the clinical testers and the testing towards the appreciate the patient. The antibiotic situation that require to ensure they promote the appropriate the clinical actions, the order to the translate the foster diagnostic test. The result is the laboratory in the improve the beside the outcomes. The diagnostic revolutions underway is the clinical microbiology laboratory, by providing the more rapid, robust microbiological diagnosis. The new microbiology diagnosis the technology have the potential to transform the modern biotechnology. The laboratory and the care of the patients with the suspect of the infection. This may overview of the rapid molecular infection disease diagnostics. The clinical take the 3 basic truths of the clinical microbiological laboratory. So first one is the whether the patient is infected. If so, with the second one is if so is with what, and third one is the what will treat it. In the windows between the specimen the collection of organisms the identification is the antibiotic assessment of the determination. So next one is the overview of the rapid molecular infection and the disease of the diagnostics. The bacteria, viruses, the fungi is the parties that cannot be repeatedly identified using the molecular method. To the diagnostics, because the agency infection of the bloodstream they represent to treat the urinary. The new technologies used to treat rapid diagnostics. The technologies include the nucleic acid-based diagnostics. So this may feature directions. Or the rapid molecular infections disease the diagnostic evaluation is the ability of the microbiology laboratory to more rapidly provide the 3 basic truths of the trachea of the cleanliness. The current technologies are excellent of the formally answering the questions whether the patient is infected and this is over there. What is the identification is organism directly from the specimen using these enemy panels. This my conclusions. Current technologies are excellent or the answer is the questions whether the patient is infected. However, the technologies currently cannot have the perhaps will be have the able to provide the if the negative results rule of infections the critical to the success. The practices and the regularity the authorities which is can be challenging of the fact the phase of rapid growth. The emerging the technologies can be over the regulatory landscape. So thank you so much for giving the opportunity, sir.
Thank you, Dr. Santosh Kumar. Thank you.
Sir, can I present?
Thank you, sir.
Yeah, Sankita, you can go ahead.
Thank you, sir.
Shall I present, sir, right now? Hello, sir.
Yeah, you can go ahead.
Shall I present?
Yeah, you can present. But slides are visible now. Yeah. Sankita Devi?
Yes, sir. Yes, sir.
Yeah, you can present.
The topic is biological active component of Tinospora cordifolia and their biological function in controlling the different diseases. Tinospora cardifolia is a deciduous climbing shrub which is commonly known as a gilloe, belonging to the family Verismepodiaceae. A variety of biological active components A variety of biological active components are present in these plants, which are mainly used for the treatment of oxidative stress related disease because it contains the albinogalactam and epicataceae, and also it is used for the treatment of the pains, diarrhea, respiratory infection, cancer, diabetes, and gastrointestinal disorders. The different components present in this plants are mainly uh, mainly belongs to the alkaloids, glycosides, lactones, steroids, and other compounds. These compounds are mainly obtained from the stem, root, leaves, and various parts of this plant. So we can say that the whole plants are very useful for the treatment of the various diseases. The reported biological effect of these plants are showing here. The anti-cancer, it shows that the anti-cancer properties, it also can help in the anti sorry sir. The varieties of the biological effect shows by these plants are it is used as an anti-HIV, anti-inflammatory, anti-diabetic, anti-cancers, and antimicrobial activities. Thank you.
Thank you, Sandeep.
Shall I present right now, sir?
Yeah, Rashmi, you can go ahead. Sankita Devi, can you?
Yes, sir.
Yeah.
Stop sharing. Thank you, sir.
Welcome. Rashmi, are you there?
Yes, sir. Yes, sir. Is
Yeah,
my
you can.
slide still visible, sir?
No, it's not visible. Sankita Devi, can you mute your?
Yes, sir.
Is it visible, sir, right now? My slide still visible?
Yeah, your slides are visible, but keep it in slideshow mode.
Well, thank you for the opportunity. Myself, Rashmi TS. Today I'm going to present topic phytochemical analysis and evaluation of anti-urolithic and biostimulant and modern properties of a banana pseudostem extract. Straight away, I will go with the materials and methods because we have collected the raw pseudostem
and that from the Badravati 20 on Karehavi, and we extracted the pseudostem juice also. Along with that, we carried out the phytochemical analysis by the standard methods. Then we also done the evaluation of in vitro anti-urolithic activity of a banana pseudostem extract by a titrometric method. Exactly weigh the 1 mg of a calcium oxalate and 10 mg of our extract are packed together in a semi-permeable membrane, and culturing the method, this was allowed to suspend in a conical flask containing 100 ml of zero point 0.1 molar phosphate. Another group served as a positive control, which obtained 10 mg standard needy drug and 1 mg extract. All these three conical flasks are placed in a incubator, and it is as usual titrated by the 2 molar 1 normal sulfuric acid, and it is titrated against zero point 0.1 normal KMnO4 till the light pink colored end point has appeared. So the percentage of dissolution of calcium oxalate by our various groups are calculated. And also we studied about the banana pseudostem sap as a biostimulant and its influence on a seedling growth of Arachis hypogaea, that is a groundnut, and the Macrotyloma uniflorum, that is horse gland. So in this study, the banana pseudostem extract checked for its biostimulant characteristics and as growing media. Then it influences on seedling growth of several seeds such as groundnut, sweet corn, half gram, as well as the chana. But we observed a good result for the groundnut as well as for the half gram. Further study is concentrated on that. So this is how we first selected the seed treatment method for all the 4 seeds. And after the observation, we go with the good result for the Arachis hypogaea as well as for the horse gland. And also this pseudostem sap extract is a good mordant source for the natural dye. So the Vixa or Elena are the natto seeds are collected and crushed and mixed with the water as for the extraction of the dye. Then that is treated with the 10 ml of margarine, that is nothing but our pseudostem extract of the banana. That is going to give the good result. The material were dried in a hot air oven with 60°C for a half an hour. Then the dyed material is washed with the detergent and determine the retention of the color. So coming to the total result of our experiments, collected banana like tannins, phytosterol, proteins and amino acids, tannin, resins, triterpenoids and the phenolic compounds too. Along with that, this study evaluated the anti-urolithic activity of the aqueous extract, and here we find a good result. 38.6% of calcium oxalate dissolution was observed in this extract. So when compared to the needy standard drug, so we found a good result with the banana pseudostem extract for the dissolution of that calcium oxalate, which is going to be the part of that kidney stone. Pseudostem sap is a good source of beneficial secondary metabolites and nutrients for plants. The smaller concentration of the sap showed a higher percentage of seed germination compared to control, while the higher concentration inhibit the germination because the sap may also contain the unknown allelic compounds like abscisic acid and pathogenic microorganisms, which could inhibit the germination rate.
Rashmi, sorry for the interference. Can you please wind up your presentation?
Yes, sir. Yes, sir. I will go to the conclusion.
Yeah, thank you.
The phytochemical constituents, as we already discussed, many groups of flavonoids present in the sap known to have an anti-urolithic property and various bioactive compounds present in the sap, and the various, although the quantification of secondary metabolites has not yet been done, future research on the quantification of beneficial phytochemicals will lead to a better understanding of how banana pseudostem sap can be applied in a various fields such as agriculture and medicine in an efficient, economical, and eco-friendly manner. Thank you, Ananda.
Thank you, Rashmi. Next presenter, you can Jyoti Gupta. Are you there? Is there anybody is ready to present now? I think almost all the participants have presented their work. Shall we wind up this session? Dear participant, please respond. Shall we wind up?
Sir, can I present, sir?
Yeah, you can present.
Yes.
Balaji, your voice is not audible. Please make sure.
Yes, sir. Is my voice audible, sir?
Yeah, now it's audible. Please go ahead.
Is my screen visible, sir?
Yeah, it's visible. Go ahead.
Good afternoon, everyone. This is Balaji, studying BTech third year in Biotechnology at CBIT. Today, I am going to present about vaccine pros and cons, navigating the benefits and risks of COVID-19 vaccines. The COVID-19 pandemic-19 vaccine is complex and it is essential to consider the pros and is to explore the benefits and risks associated with COVID-19 vaccines, including their efficacy, side effects, and potential long-term impacts. By examining the available evidences, it aims to provide a comprehensive overview of the benefits and risks associated with COVID-19 vaccines, empowering individuals to make informed decisions regarding health and well-being. Now we will see the pros and cons of various types of vaccines. Coming to whole vaccine, it is of two types: inactivated vaccines and live attenuated vaccines. This can be available in the form of Covaxin, Sinopharm, etc. And the pros will be suitable for people with compromised immune system, no live components, and there is no risk of getting diseases. And the cons will be booster shoot may be required. Next, coming till Janssen, etc. And RNA vaccine is available in the form of Moderna, Pfizer, etc. And pros of this is immune response involves B cells and T cells. There is no live component, so there is no risk of vaccine. Relatively easy to manufacture, but when compared to cons, some RNA vaccines require ultra cold storage and booster shots may be required. Coming to viral vector-based vaccines, it will be available in the form of Johnson & Johnson and AstraZeneca. The pros will be stronger cellular immune response by T cells as well as the production of antibodies by B cells, and cons will be the previous exposure to the vector could reduce effectiveness. It is relatively complex to manufacture, and protein sub vaccine will be available in the form of Novavax, suitable for